Biodiversity, organized by Universidad Javeriana in the framework of the COP16. We are honored to have here experts dedicated to key roles of the somic science to investigate the molecular composition of foods and take so that we can make decisions based on science around the food security in this country. We start with the national anthem of Colombia and Valle del Cauca. Please stand up. Thank you very much. You may sit. Now I give you Dr. Ingrid Schuller, Vice Rector of the Universidad Javeriana, who is going to give us a welcoming words. Good morning, everyone. On behalf of Father Vicente Dron of the Compañía de Jesús, director of the community and all the whole community of the university, I want to welcome you to this symposium that gathers us around the molecular characterization and scientific evidence around all the living things. Biodiversity gathers us here. We are so joyful to have these sessions, which are endless if we want to talk about biodiversity. Facing this is facing a huge challenge for a country like ours. This invites us to have academic sessions, scientific sessions, social, political sessions, because around biodiversity there's a lot to talk about. But I also want to to remark how we are fortunate we are to have this and connected to what we listened yesterday in Simposio. It is paramount for us to keep continuing the genetic richness that we have in all the living things and knowing how we are going to leverage that. I want to highlight, even though that we are in the 21st century, the scientific leaps that we have given in the last century. Science has contributed to the knowledge of the living things. I cannot ignore efforts all around the world that have reached Latin America and our countries to leverage those scientific advances and use them in living things in our region that belong to a region that we are lucky to be so biodiverse in Colombia. Back in the 90s, many genomes of living things were sequenced. Plants, animal microorganisms, which have been the source of knowledge to keep controlling and leveraging and working improve our food, our health and genetic richness. The use of tools have been transforming. Working in molecular characterization is very different to work in a morphologic characterization system. And that has led us to talk in a responsible and ethical way that reaches the whole community, not only the scientific community, but also society in all the countries, because there's a lot of efforts around this, a lot of investments in human resources and technology. And if the society don't know these advances, might reject this, and that has a huge cost for a society and for an area like science, technology in all these tools. We have talked about knowing the human genome. That is the more question one in this tradition of knowledge, of molecular knowledge, with huge advances. How are we not going to know what is the footprint that we have in our genes to improve our health and to overcome nutrition problems, behavior problems that are linked, as we talked about yesterday, with genes, with genetic information, and with an environment. Gene transfer was one of the more questioned technologies, and we worked around that. We worked on that in a way, knowing the good facts and not ignoring the facts that science has not strayed from nature, has respected nature process and based on them to be more accurate and to know what we are dealing with how we are using that. We are also talking about other sciences and technologies. We moved from genetics to get to know what is expressed and how they express it. We are talking about omic sciences, transcriptomics, metabolomics, and many others that are going to appear because living things not only have static sequences but they can modify on time in aim for a necessary response for society. In plants and crops, we have struggled to to get cleaner and healthier foods. We know that in harvesting human foods, there's a lot of plagues and illnesses that we have to overcome, and we have to reach better practices so that the consumer is is sure that what he's eating is not the product of a moody management, but a responsible work to achieve better foods for the environment, for the consumer. We also found that this is a value chain. Not only do we have to know what the living things that we are dealing with, but also to know what are the leftovers that we leave. Those byproducts are object of the scientific knowledge so that we can achieve not to impact the environment, and so that we can recycle of those byproducts so that they can be used in a better way. I invite you to keep having those conversations and to learn with the presenters that we have today. I want to point out that very proudly, in an honest way and humble at the same time, I want to recognize the effort of the Universidad Javeriana in Cali through the ómicas Institute that has taken that leap in science and technology. I want to invite you also to bring all our partners and stakeholders who are not only giving their funds but also helping us to give formation to the new generations and create capabilities. Thanks for the Omicas group led by Dr. Jaramilla, who are in the state of the art of what are the application of all these technologies for humanity. Let's keep knowing, preserving, and using biodiversity with new tools. Thank you very much. Thanks the Vice Rector for her words, and now we have some announcement by Andrés Jaramillo, Director of Ómicas Institute. Good morning, everyone. Today, as the Vice Rector was mentioning, to whom I thank her words and her presence here, we are going to have an event very much in the framework of the COP16, which is going to be held here in Cali on starting on October 21st. Why is it important? This particular symposium, it has a lot to do with a topic which is very familiar to Ómicas Institute, and this is food security. We're at a breakpoint. We're close to a breakpoint in which the population, the growth, is going to exceed our ability to produce foods. We're also in a privileged country facing what we know biodiversity, and we are going to see through different talks. We have a very good set of talks today, which is going to lead us to know how to measure that biodiversity in edible, and we are going to understand its composition in all its dimensions, not only nutrition dimension, foods, plants, what plants produce, is a range of phytocompounds and molecules that have utility beyond human consumption for nutrition. And that's where we are going to have to make a huge effort to recognize what Colombia has by the privilege of having this geography that we do by our geographical composition. In that sense, we are going to see how, as Dr. Schuller was saying, we have a very particular genetic expression of organisms in a context of a variety of conditions which well preserved, which allows that diversity. And I want to talk about a convergence that we have here, because this is not only studying living things from biology. You're going to see that characterizing these living things, the ones we use as food, is very difficult as how we can extract that composition, how we can analyze that composition to understand the function of each one of those molecules, right? So our invitation is that we have here that a moment of joy. We have very good set of presenters. We are going to be during the COP16. We are going to have an open house. 25 and 30th of October, we'll have an open house, and we are going to be participating in zones south and zone green. For the ones who are interested to have the tours that we have in the open house in the ómicas, please register yourself. Our pages are open in the ómicas or in the university are already available for you to sign up. So I want to, with this, welcome you to today's event, to this symposium, and I want to go straight to present one of our first speaker, Mauricio Díaz Granados, who accepted our proposal, understanding that Mauricio is very, very busy. He is the current Chief Science Officer and the Dean of International Plant Science in Jardín Botánico de Nueva York. He led the initiative of things based on Nature in the Q headquarters in London. That's an initiative that aim to solve some challenges like climate change and the loss of biodiversity. He was the science director of the Jardín Botánico de Bogotá, where he led the biggest greenhouse in South America. Mauricio is a biologist from University Javeriana in Bogotá. He has a master degree in bioethics in the same university and PhD in St. Louis University in the United States. Without further ado, I want to welcome Mauricio and I want to thank him for being here. And that's the best thing that we can do. Your talk is going to be the best first talk. Good morning, everyone. Thank you, Dr. Jaramillo, for this invitation. And the best regards to Dr. Schuller, Vice Rector, and all the organizers, colleagues, and friends that are listening to us. I'm going to share my screen with you to start presentation. Please let me know if you see my screen. Yes, we are. Thank you very much. As Dr. Jaramillo mentioned, I am honored to be from the Universidad Javeriana. I was the director of the herbarium for 7 years, and I did my doctorship in University of San Luis. I was there for 6 years, and after that, I had the opportunity to go to my postdoc in the National Museum in Smithsonian in Washington, Natural History Museum in Washington. I was the scientific director of Jardín Botánico, the Q, for 4 years. I was lead researcher for the Q for 7 years. And this here is my new house, the Royal Botanic, the Botanic Garden from New York. I want to show you a few slides. It's one of the biggest research centers at a global level. We have the second or third greatest herb collection. It has over 8 million species, so it's 275 bigger than the one we have in the Javeriana. We have the biggest library, botanic library. We're saying the numbers. We have 750,000 volumes, printed volumes, and 13 million files. So if we gather the next 4 libraries in Bogotá, Javeriana, and Cali, it's around 730,000 volumes. So it's a similar quantity, although all the books that we have are about botanics. And we have some of the more spectacular labs, more complete labs, where we can do a lot of research from the molecular level to the ecosystemic level. We have a program of genomics, a very important program. We're going to talk about that. Our scientists discover many species, many plant species. We have fungi, a cordyceps killing an ant. So we have a very important activity at a global level in new species publications. In average, we are talking about 40 new species per year. But also we have a program of environmental action where we work with restoration ecology, with communities. We deal with about 300 gardens, urban gardens in the city of New York, and we work in several countries in different continents. We have 79 projects in 81 countries. In this network, I want to show you our collaborations based on a sample of publications, recent publications. So you can see that the colors we have that show the diverse geographic areas of our colleagues. And in my case of science directors, chief science officer of the Jardín Botánico, I'm in charge of the three centers, facilities, central resources in science, the herb library, the labs, and three centers, biodiversity and evolution, plant center, people and culture, and the center for conservation and restoration ecology. So we also have a program of graduate students. We are linked with 7 universities, six of which are in the area, Cornell, NYU, Columbia, Portham, etc. And we have collaborations with Florida International University and with a university in Germany, where we give, alongside the titles of doctorship, we have 25 doctorates. We have many more students in master's degree, and we have some scholarships also. Regarding uh, food science, we have several lines. Here we have the five main lines. We have some underutilized plants or not known. We have a program, species related to crop wild relatives. We have a resilience line of food plants and sustainable practice also, and food plants and bioeconomy. Regarding neglected and underutilized species, NUS, I continue with my line that we started in the Q garden, where we started working with a global list of edible plants. But we also have some smaller approaches in Colombia, for example, Mexico, Chile, Ethiopia, and India. Regarding crop wild relatives, in our lab we identify, screen, and we also do some research on potential gene for domestication. We also have a genomics program where we evaluate genomic-based and metabolomic expressions. We also have sustainable practice through the Centro de Plantas, People and Culture, where we evaluate the efficiency of the mixtures of plants, crop plants, and some mixtures of grain species that come from ancestral traditions, many of them from Africa or Mexico, compared to the monocrops. And we also have some projects around conservation through use. And finally, we have a line we want to develop even furthermore, is the food plants and bioeconomy. I do it from an initiative that I started in Q through the network of natural ingredients in Colombia. And the idea is to give tools that foster the development of bioeconomy, local bioeconomy. So I'm going to tell you about my trajectory in terms of edible foods. When I came to Q, I got in charge of managing the base of useful plants of the world checklist of useful plants. We started with a publication in 2020 that gathered information about the state of plants and fungi in 2016. And that database was the first input for the report of the state of the world's plants and fungi back in 2020. And papers like this, in which we proposed of having a description in several aspects on the edible plants, curated with names that exist in the world. And in that paper, in that article that we published in Plants, People, and Planet, we checked the phylogeny and the related uses, which were the most important families for the food security. What other uses could those plants have? But also we looked where that wealth of edible plants were concentrated and the state of conservation of those plants. On that first article, we managed to match the names of about around 7,000 plants around the world. The list keeps growing. We are talking about 25,000 species, and I want to tell you very soon about a new publication that is about to come up, about 25,000 species that we now know today. But with the information that we had at that moment, we managed to get some approaches to understand how that distribution of plants can provide or can allow us to create some solutions for food security. In this article that we published in Nature and Plants, we looked what countries have deficiency or have a deficit of different types of B vitamin versus the wealth of species that could supply that lack of vitamin. You can see here the maps where we can see the opportunities to use species that can have high content of those vitamins and could create solutions, local solutions. A few months ago, we published this paper, which was the cover of Science, where we have the global distribution of plants used by people, by humans, where we identified the richer areas and the more endemic areas and the places that require most conservation areas in terms of useful plants. And only 10 days ago, we published this article in Nature Sustainability. We transferred the focuses on protection of conservation areas to apply them to preserve the agrobiodiversity, and we propose a framework. We propose a framework, a theoretical framework where we can implement these conservation areas that allow us to protect species that sometimes are outside of the conservation structures that we usually use. But now we're going to talk a bit more about what we have learned in Colombia. We start from the when I was working in Q Gardens in the project of useful plants and fungi in Colombia. This was a huge project that involved 119 researchers from 26 institutions, including Q, 24 of which are in Colombia, many universities, and in that project, which lasted for 3 years, we published 5 to 12 reports, 3 papers, 4 applications, 9 videos, several portals in which we have the call plant Coplanta.org and Colfunky.org, which have a wealth of information. In total, we have information about 160,000 species, including 36,000 species combining plants and fungi, and you can see some of the maps in there. And as part of that, we have two of those five books. We have the Catalog of Useful Plants of Colombia of and the Catalog of Fungi of Colombia. These books, and as they are available for free in this, we have several repositories. These are the links for them. But now I'm going to focus on the work that we started doing with Benedetta Gori, my master degree student, to understand the distribution patterns of the edible plants in Colombia. This was published in scientific reports, among other publications. So we are about 30,000 species of plants, including the non-native plants, which is a huge wealth. And we identified that there are some problems that are still remain, as you can see in the figures of food security in several of the regions. There have been some recent news in places like Guajira, in the peninsula of Guajira, but we also see that the consumption of native plants has decreased significantly along in time, and that we have some gaps in information. So we wonder how that diversity exists in Colombia, and what are the distribution patterns in the regions. We start from the source, from the catalog, including the non-native species. We have so far 30,159 species, of which 7,205 have reported uses, and we have 3,864 species which are edible to date. The list, every time you publish a catalog, it's immediately outdated. What we are talking about, 4,000 species that are edible. There are going to be more for sure very soon. Compared with the global base, only 17% of the useful species are edible in Colombia. 53% of them are edible. So we can say that we are a store repository for the world. However, only 198 species are part of natural products in Colombia and have a tax registry. Only 5% of the total diversity can be commercialized in a market like a supermarket like Carulla, because the rest of them do not have a tax code. 90% of the natural ingredients marketed in Colombia are imported, which is unbelievable. Regarding the taxonomic diversity in Colombia, here we have the wealth by family. The base of legumes are the most important, followed by the palm trees, the grains, the coffee, and other families. Here you can see the number of species by family, and here you can see the most important genus. Genus, potatoes, Sapotaceae, Brocchium, Burseraceae, etc. And here I want to tell you what is the diversity of edible plants in Colombia. We have 230 species of passifloras, all of them eatable to a certain degree. Only 7 are commercialized in the markets. Regarding guamos, Inga genus, we have 145 species in the country, but very few of them are commercialized, and it is very strange to find them in market. Maybe a little bit more today, but normally to buy guamo you have to be very lucky. And here we have some other promising species like chontaduro, amaranto, bataco, cresta de gallo. Inchiocacae, onchos, etc. We could mention a wealth of species which are important, and I hope to have time to show you one of them at the end of my talk.
Hey. So, okay. Now I can show you an introductory video in order to fill out this survey, and the purpose is to gather information regarding where people are consuming, in what part they didn't get the product, so the process of the product. Well, we see, for example, in the coastal region, they consume a lot of potato, which is coming from the inner side of the country, and a lot of products that are not familiar that we do consider that can substitute the consumption of carbohydrates. Those consumption patterns, okay, to be applied for Colombia. In that survey, so the people have been filling out the information. What do they consume? What do they eat? How do they prepare that, etc. And here we have a barcode. All of you are welcome in order to take part and participate in this project. Okay, of those edible, okay, products. And also we have a conservation through use. We do consider that this is a good way of that conservation through use in order to assure not only the appreciation for these species that are underutilized and unknown for many, but also the conservation of those. So what we have done, okay, let me explain what I was mentioning is, well, I think that is one of the alternatives that is less destructive and with the highest potential. But what we've done so far is that we have performed a series of programs. We have disclosed, we have done food workshops, we have invited chefs, we have done plenaries and talks with the Humble Institute, and we have published a lot of information on the media, and we have invited well-known chefs in order to explore with native species from Colombia that are very little utilized, and they have developed new dishes that are international or Colombian, or you can find them in Minimal restaurant in Bogota and Celelele restaurant in Cartagena. The idea is to enrich the composition of the community in Colombia. And finally, in order to finish this, and then to continue with question, I want to show you one of the species that it is utilized and that I consider that there is potential, and there would be other species, is Guaymaro, red nut tree, proximal alligastrum, and the Moraceous family. You can see on the right how Guaymaro is. These are gigantic trees from the forest, tropical forest, as you know. So the dry tropical forest has been destroyed in the country. Only 5% of these forests, and Guaymaro, the breadnought tree, is an emblematic, is key of the dry forest. Is a kind of an umbrella in order to have the interaction of the forest. It's kind of a species that is well recognized of the dry tropical forest. And the Guaymaro is an edible species. We can see the seeds of the Guaymaro that through centuries they have been consumed by natives, by the local communities, indigenous communities, as well as farmers and peasants. But Guaymaro is also, as you can see, is so gigantic, bulk, so they have been cut out in several regions of the country. So we have very few of Guaymaro trees or dry tropic of these breadnought tree in terms of consumption. The forests of Guaymaro are known as food forests. When the Guaymaro tree and they bloom their seeds, and you can see that these seeds are spread all over the ground or on the soil, it attracts a lot of animals. And also the community do it for hunting. So you can see the potential of these forests, not only for the fauna, but also for the local communities. The seed of the Guaymaro tree, it is clean, it is dried, and then it is munched, and you can make some kind of hot beverage, but instead of that of oat, it looks like oat, you can have Guaymaro powdered. In the Perijá Sierra, it was an area that was affected due to the armed conflict. So we did a testing and a pilot project with these edible species in order to supply alternative solutions for additional income for the communities, and also problem solution in terms of inequality, social conflict, and all those things. So what we did with them is that we designed in terms of co-working and co-designing. We made these traps to capture the seeds. The people from the local community, they place them in order to catch the seeds before going to the ground. We mark the trees, we measure the productivity, the quality of the seeds. We taught the communities how to collect the seeds, how to weigh them, to measure them, to assess their quality. And also we taught them how to make a primary transformation in situ to show them and the proper classification, separation, as well as a secondary transformation in situ, in the region. So in Becerrillo, we have implemented a system which is in two other areas in the country where they do it by themselves. They prepare the product. It is sent to Bogota, where we have reached different organizations. The product is packed. You can see the Guaymaro powder or flour, kind of flour. So it is distributed in different restaurants, such as restaurants in Cali, Bogota, Medellin, and Cartagena. And if you go to the Salele restaurant, You can find the desserts cooked or made by Waimaru or flour. And this is a way of aiming those sustainable uses of the species, edible species that we have in our country. This has been very useful that along with the community they have seen that instead of cutting out the Waimaru tree in order to expand the mining borderline or the cattle raising, it is also important to have good conversation of our forests and also restore. We have a restore program of Waimaru tree. We can see the plants, and with the community on the left, we have identified several places where it is very important to expand and to do restoration process and also the seeds for these Waimaru trees. And we hope that with this project we can save, restore, and protect this dry tropical forest that we do need. With this, I want to show you that it's a huge diversity of edible plants. We know very little, and this is an invitation for you in order that we do some research about those species that are unknown, that it has, of course, a future for our country. I want to thank you, and I will be willing to answer any questions or any comments that you have. Thank you. Thank you. Thank you, Dr. Diaz Granados. Now the audience is open for questions. We have our people with the microphones for any questions. Good morning, Doctor. Thank you very much for your speech. So I have a question. In that exploration of new food supplies that you have used the keyword which is edible, what was the criteria that you utilized in order to define that a new plant that has not been well known, that it doesn't have a historical background but any community and is new in the sense of being discovered? What is the criteria they utilize that you consider that is an edible species? That is a very good question. I want to thank you for that question. This is a key question that we can also transfer to our useful plants catalog worldwide. This is a question that I've received all the time. And if we think about most of the plants are useful, they may be useful, of course. I can, for example, I can take any branch and maybe make a relevant utilization to make fire or whatever. But the definition of useful or edible plants is a plant that has a cultural use that has been transferred generation after generation, and that also has been captured in terms of literature. So through, of course, through a research that has been published. So we don't do the identification in situ or in the field. We don't ask the communities or the local communities what are they consuming, and we record. What we do is that first we do a general review of the information that already exists, that is already published. We make a comparison with the presence of the species that are already listed, and from that, or from those sources, we record if there is a reported or edible utilization that belongs of the servo-cultural of the communities. For Colombia, we have reviewed around 1,400 sources, maybe. It was more, if I can recall, okay, it's more than 1,400 sources of literature that was published. So I'm not talking about a plant that is only is a fruit and a person that can taste. So there is a consumption tradition in Colombia through generations. Another question, please. Good morning, Dr. Professor Mauricio. I have a question in regard to, so I saw a graph that you mentioned about the species of the Menostomataceae family. However, it was not mentioned as an important species, but I would like to know if maybe if you know if there is any edible plant that belongs to this family. Thank you for your question. This is an important question. Melastomataceae family, we have some of them that have consumption by indigenous communities. Possibly there would be other kinds of, so we're talking about Myconia, is one of the ones that we have, the biggest one that we have. But I can't recall at this moment if we have a species from other kinds. But of course, this information is available online. And here, in the article that we published, let me check. Here in this article, you can see all the relevant information, supporting information included the edible species catalog, okay, that you can find it there. The article is available online. You can take a look at it, and you can find the supporting information. You can download the catalog, and you can find all of the species for each of the families, okay? Thank you, Professor. A particular question here in Colombia about, in terms of ambitious projects for recovering of plants for common utilization. I like what you mentioned about the Guaymaru tree, also in the Llanos Orientales area, in Puerto Lopez area, all of the plantations of the cacao and the sachains for the oil obtention. So we have oil sources very good. Maybe in the short future we can replace the olive oil or other oils. What project or real? Because I feel sad because in Colombia we don't take mamey, guamo, no piñuelos, no zapotes, all those fruits. All the time we are having pears, Chilean pears, and grapes. And in terms of sovereignty, and this is tons of carbon that it is waste. That in terms of production is to be closed. But what do you know about these ambitious projects in regard to recover the consumption of traditional products that maybe we are not aware or we don't know? Well, I share that kind of sadness or pain in regard that you miss, for example, that I am in the region when I visit Colombia and I want to drink a natural orange juice. They only say that is soda or just, yes, or canned juice. Yes, I feel that pain. That so maybe the not to know that richness, that potential amounts of products. I think that the country slowly or gradually is evolving to that kind of recognition, but we have to break those traditions that have been imposed. In fact, when we think about a typical dish of Colombia, la bandeja paisa, for example, many of the ingredients of la bandeja paisa dish, they are introduced. We're not talking about native species. What I mean is they start as a starting point of not knowing the potential of our native species, and this is something, a wake-up call in order to start our diet. I think that there is a huge opportunity. I know that there are institutions, or many institutions, that they do have the same purpose, intention. There will be a lot of meetings during the COP16 from the Botanic Garden from New York. The idea is that the garden can be a leading in terms of global conservation for edible plants. So let's see that very soon you will be hearing the good news. And of course, there's so much work to do. Currently, we are having meetings with Trust, and we did a workshop about the roles of the botanic gardens in terms of conservation and research of edible plants worldwide, because sometimes the plant sectors that are harvested, or the crops, they are focusing on those harvested plants or crops, but they don't know about the unknown plants that we have. Thank you, doctor. Yes? Yes, doctor? Maybe something to add, doctor? Yes, I just wanted, we are short of time, but I would like to thank you. That question is a valuable question. Thank you, doctor. We want to thank with a round of applause. Before continuing with I want to thank you to the people who have organized the support of the Alliance Biodiversity, CIA, American Heart Association, Rockefeller Foundation, and of course, Universidad Javeriana, Cali. Now we have the same speech in terms of the utilization and tropical genetic. Dr. Monica Carvajal, she's leader of the Digital Gene Bank in Alliance Biodiversity International, NCIAT, and she works in the development of a gene banks and digital gene banks in order to make conversation and the proper utilization of biodiversity. She's a biologist from the Javeriana University and virologist from the Abel Wildens University in Germany. Her career has focused in the investigation of virus that is affecting crops and also surveillance in terms of agricultural diseases. Good morning, everybody. Thank you very much for the introduction. As you can see, I have changed in terms of areas of research, and for more or less about 5 years ago, or more than 5 years, I have been working with the genetic resources from the Alliance, and I'm scientific from the Biodiversity Alliance and International, and the International Center for Research, the CIAT, and the presentation is related to digital information, digital sequence information, and how this has been supporting the conservation and also promoting the use of genetic resources, the ones that we conserve in the herm labs in the Alliance. According to the organizing body, the International Treaty about the plant genetic resources, up to date there has been an agreement of the definition of the terminology of the digital information. However, for this presentation, I will refer to digital sequence information or data or genetic sequence data derived from the material that we have and focusing on the information of genetic sequence from DNA. This digital information can be stored, you can have access and share in an electronic way, and will allow the researchers and scientists to make use of the genetic information in order not to have access to the biological material. So I want to share with you some of the examples of the work that we have performed and research in the Alliance. But before that, I want to tell you that the Alliance and the germ plants banking belongs to an international red of germ plants banking, that this is a consortium in agricultural. So this network is composed of 11 search plant banking that are located strategically in biodiversity centers. You can see, for example, in this map in green, we can see the countries that are the ones that are called house or greenhouse areas for this. The network store more than 700 materials in terms of grains. We have tropical forage and other centers in terms of conserving diversity of trees, in terms of roots and banana and tuberculosis. There are 2 banks, one in Belgium, and conserves the diversity of banana crops. And we also, in the Palmyra, in the CIAT, we have a conservation in terms of global biodiversity of 3 crops. We have cassava. We store more 6,000 materials or genetic material that are coming from several countries in vitro. Another dark collection of genetic fascioles from the bean. We have 38,000 genetic material coming from 100 countries. And also tropical forage, we have 23,000 genetic material coming from 75 countries, and this is a diverse collection, and it has a higher number of species. This is important for food, animal food, and also for human food. So at the moment of composing the gene bank or the germ plants in banking in Palmyra in the 70s and up to date, we have distributed more than half a million of samples to more than 140 countries. You can see the countries that have been the beneficiaries and receptors that we keep here in Palmira, and to conserve and distribute the material and the diversity of the crops that I mentioned before. This has been possible thanks that the procedures and operational procedure for the storage of those seeds and also the disclosure of these plantals has been working for several years or 10 years. There are other effective ways in order to get to know what is the better way and to transfer the benefits of the genetic materials from our country to the different modern varieties, but it's insufficient. So this is due to the lack of information that it is associated to those genetic material that we have in our collection. So this brings about the potential of these varieties, of the biodiversity of these crops. It is underutilized in several cases. Luckily, there have been progress in terms of areas of knowledge, in terms of the DNA sequencing, and which allow us to utilize the gene, okay, blend in terms to utilize it of the germ plants in the banking and also to close the gap between the crops and improvement programs. So this image on the right, you can see, okay, the greenhouse that we have since 2029, the genetic data. This is called Future Seeds. It's located in Palmira. On the B line going to Palmira, this is more than a building. This is a building which is eco-friendly with the environment. And as I mentioned, more than a building, this is an initiative that we have been working in 3 thrusts or main pillars that you can see in the circle. The first pillar is conservation and distribution, because we have to continue conserving and distributing in an efficient way all of the genetic resources that we keep. Also, we have an innovation pillar, because we also want to take advantage of the new technologies that are in terms of genotype and digital and information and technological information in order to improve the conservation and enable access to these genetic resources. And the third pillar that we have involves in order to commit and engage. It is important to engage the society about the importance not only to conserve but also to know and to use the genetic resources.
I have chosen the digital bank, which is one of the innovation areas, and this digital bank is consists of 2 areas: an extraction lab, a DNA extraction lab, where we run all the processes that are summarized here, that involve all the processes that are related to the tissue collection, DNA extraction. We sequence outside with a third-party sequencer to obtain the DSI. I told about that. We have a lot of materials about the collection. We have a 66,000 materials. We are creating a data bank. And we have an open office area with data scientists supported by servers who connect the different types of data that we get, especially the sequence data with other kinds of information like evaluation data, passport data, and other kinds of evaluations. I'm going to show you some examples of some specific cases of how we are working and how we are facilitating the conservation and use of genetic resources. So far, what have we achieved? We have been able to create DSI of 3 collections. The cassava collection, by being the smaller one, we have been able to achieve not a complete genome, only for some materials, but for 100% of the collection, we have genomic representation of the materials of the collection, of the cassava collection. Beans, we have been able to sequence, we have obtained sequences of 10,000 materials of the collection. That is a small sample that represents the diversity of collection. That is a huge collection. And of tropical forages, we have focused on those genus that are important because there is an improvement program. That is the most diverse collection, and we have to select with which groups we want to start. This has been only possible. The costs of sequencing are lower now, but it's still very costly, and it has been possible thanks to the institution that I have here in the bottom of the screen, and they have been funded through projects that aim to do some research to find solutions for adaptation to the climate change, the increase of nutritional value of the crops. And in that role, we have been able to learn a lot about the diversity that we preserve. I want to show you some specific cases of how we are understanding and exploring the resources that we preserve. And let me start with the cassava collection, which is the smaller one. The smallest one is the most costly to preserve. It's in vitro. Most of them are local varieties, 82% of them. It's 8% of wild species related to the species Manihota esculenta, which is the one that people crop here. And we have some improved lines. Most of the diversity we come from Colombia, Brazil, and South America, which is the place where we believe it was domesticated. With the genetic information, we have been able to see the relationships between the materials that we preserve. This is a graphic representation of the genetic distances. Each point represents a session, and the closer they are, the most similar are the materials. The farther they are, the less similar they are. We have a blue cloud, which is Manihota esculenta variety, and improved varieties, and the green points represent the wild species that we preserve. There are some groups, the closest to the blue one, to the blue cloud, is subspecies related to the most closely related to the harvested species. With this information, besides knowing the composition, another of the priorities for us is seeing how different the preserved materials are, because we want to preserve diversity. And as I mentioned, the cassava collection is kept in vitro, so that is a way in which we harvest it, not by seeds. So this is the most costly collection. About $92 is the cost of preserving an accession, and we are wanting to preserve that forever. So we have to identify the duplicates or the redundancy is very important for us because it can help us optimize the physical space, reduce costs, and optimize the efforts of characterization. And at the end, it's going to help us make the collections more accessible to the public. We had a survey in which where we studied the genetic distances as a baseline to tell from this baseline everything below it of this genetic distance is redundant and above is considered unique. Defining this line is not easy. After several attempts and after using different techniques, we have reached the conclusion that the difference of the distance of 1.5 allows us to identify unique redundant material. To our surprise, we found that there is a high level of redundancy in the cassava collection. It's about 50% of genetic redundancy. This is very important because this allows to optimize the conservation, reduce costs, make decisions on what to do with the materials that are redundant, and helps the curators of the collections to select the materials for other preservation activities. For example, if they have to, due to the regeneration programs, select only the ones that are unique, and also prioritize the cryopreservation efforts. We published this work at the beginning of this year. I was telling you that there are 11 centers that preserve diversity. There is another one that preserves cassava in Nigeria, that is the International Institute of Tropical Agriculture, and they preserve the diversity of African cassava. The yucca started in South America. It was taken to Africa early on, and Africa nowadays is the main producer of the world. Nigeria is the first producer in the world. And leveraging that these advantages are also characterize their collection using the same platform as we did, that allowed us to take this both sets of data, co-analyze them, and to understand how how is the collection, the composition of the two collections. We see this diagram. This is a representation of the genetic distances in orange. We have the African ones in green, the one here in Colombia. We can see that both collections are complementary. There's little redundance between the two collections. It's about 4% of genetic redundance. We think that this distinction that exists within the collections is because aiming for re- resistance, they they mixed with another wild species that has rendered the materials to be very different to the ones domesticated here in America. We are still working with ETA to understand both collections, and understanding this information, this DSI allows us to do everything that we have shown you, but we also can do mining to find genetic variables, to find solutions to different problems. I want to share with you a master degree thesis. As I told you, or cassava in Africa is very important. It is affected by viruses and illnesses caused by these viruses. The brown stripe illness in cassava, searching for solutions for that illness, they asked us for us to some for 235 accessions. They took this material to Germany and they analyzed that, and they identified that out of those 235 there are 16 that can tolerate and are resistant to that illness. Out of those 16, 70 were not couldn't resist the viral infection. These are the symptoms. This is a sample of the study of this independent, and it was published back in 2019. So leveraging those phenotypic data reported by this group and the genetic information that we have from the collection, the student Jessica did some association with this, and he identified 35 associations related with resistance. With the functional notation, in the context of the genome of these variants, we found that they were inside genes which are recognized with pathogen recognition. But we didn't want to get there. Our interest was to promote the use of the resources that we preserve. So what we did was selecting the association with more percentage of phenotypic identification, identified the responsible allele of that resistance. And we found in all the accessions, we don't have the phenotypic data, we have the genetic data, but not the phenotypic. And we found 35 materials that have a high frequency of those variants that we have found. And we propose a list of these 35 materials that we predict that can be tolerant to this illness. So that is a way in which we can facilitate access those 6,000 materials to a smaller group with desirable features. Last week, this work was accepted and is going to be published in scientific reports. Another example, not about illnesses but climate. We want to provide solutions for the climate change, which is affecting each time more and more the farmers. And there is an initiative that includes our center. We are doing allele mining to look for climatic adaptation. We are working with 5 collections, 5 crops. We we provide mandioc, cassava, and bean in local varieties. This is the composition of the collection. We can see in green the local varieties, which are the most of the collections, and the dark green, you can see the ones that have geographic coordinates where they were collected. So we started. We think that the local varieties were domesticated here and adapted to the cultural part in a given environment. So if we have the coordinates, we can extract climate variables. historic climate variables, and with the genetic data we can associate in an environment fashion. We have done that for cassava, and we're doing that also for beans, and we have used 37 environment variables, and we have found some very significant variants which are associated to high temperatures, to receive high temperature and low rainfall. So as I told you in the last example, we have made a prediction of what materials can be within that the collection, high temperature resistance and low rainfall resistance. We have that included in the program so that we can introduce those materials to the EDIT lines to find if that molecular markers are already markers fixed in the improving processes. Another case is our collection of forages that is the most diverse and the most challenging. We have more than 700 species, most of which are wild species. Most of them are leguminosae. That is Stylosanthus is the genus which more representation. 7% we have Graminea, and I'm going to go very quickly to focus in a case in which we are helping clarify the taxonomy point to understand the collection also. That is Stylosanthus amata, which is the species, the third most abundant Stylosanthus species in our collection. Amata is a species highly distributed in the Caribbean, and it's known. We have diploid and tetraploid form. The tetraploid is the one that is most used as a forage. We have an accession preserved in the collection, which was collected in Venezuela. When we collected it, we thought it was Stylosanthus amata. This accession punctually has been used because it can resist soil with low fertility. In Australia, this accession was registered, recorded as a cultivar summer in verano, and it's known that this is a variety or a tetraploid form of amata. Recent studies show that it is all allotetraploid, so it became tetraploid because it had two parents, one is Stylosanthus amata, another one is Stylosanthus humilis. The authors of these papers propose that these allotetraploid forms can be separated as independent species, but the morphologic part that doesn't clarify that. So our purpose and to know better the collection that we preserve, we want to identify the accessions that are tetraploids and be able to classify them to ease to have a better management of our collection. I want to be even faster now because I'm running out of time. We have identified a group of materials that came from different countries, which include the Stylosanthus amata and some other species, to organize better the analysis and the grouping, the genetic grouping. We introduced two controls because we have evidence of two accession tetraploid and diploid accessions. So we have three approaches. We are working with genotype to group to do some analysis of groupings. We are doing some analysis to see the morphology of tetraploids and diploids, and we are doing some flux densitometry to be the prevalence of these species. Out of that collection, these are the results of the genomic analysis. We can see that there are two groups in gray, one above which is closer to Stylosanthus escava, and the other one which is more related to Stylosanthus humilis. When we look at each one of the groups, we see that in the groups there is one that groups with diploids and the other one which groups with tetraploids. So we take this control, we run the flux densitometry, and we check if the probability is different. We can check that there were different patterns. Some of them are diploids and the other ones are tetraploids. And we select those materials of which we don't know, but are grouped with the ones that we do know, and we find that there are some groups that correspond to different ploidies. We are going to do this for the whole collection, which are 365 materials, and we expect this to be useful to work with this species. And lastly, I want to talk to you about Graminea, which is another part of the collection for which in the Alliance we come with some improving program. We have three improving programs. Two of them that is Urucloa species, and we are working to know the diversity, the genetic composition of the collection. This is a collection of almost 20 sessions, 21 species, and most of them are represented by Grisanta. Well, I'm going to be even faster because I'm running out of time. I want to tell you that we have done the diversity analysis. We can see the different species. This is a phylogram. Most of them in red, we have highlighted the Brisanta, which is the most represented species. The other colors represent other species. Let's say that we have found groupings similar to the ones reported in the literature. Apomixis is a characteristic, a very important characteristic for the improving program, and that is the capability for asexual reproduction through seeds. This improves the improving programs and is also connected to an increase in productivity. That is a very important characteristic. We have found that there is a marker to be able to find with PCR methods the state of the collection. If you amplify the product to that pometic, if it doesn't amplify, there is asexual. So we can say in this new bar that most of the collection is apomictic, and this is information which is useful for the correction. I'm going to continue. Okay, I'm going to change subjects very quickly in these 30 seconds that I have of time. I want to tell you that these are some examples that we have done with DSI. I want to tell you what we can do in training. We know that this is a very important topic. DSI is not the same in different countries and in different gene banks. So alongside with CIMIT, which is another sister center, we got together to establish a connection of banks which are interested in using DSI to know better their genetic resources and give them better use. We have done a survey to know what are the necessities, the priorities. We have had a response from 14 countries. I'm going to move forward, and we have identified the priorities regarding DSI information. And we have worked for 3 years. We have organized some training sessions like practice community. We are going to do the third one in person here this year, and we are created data with the national banks of other countries for 5 crops that we share, and we are with them evaluating in a collective manner and comparing the collections. I think that is a unique opportunity that we have not explored. Many times that information is used but in a local level, but not often that information is shared and understood in a collective way. Understood in a collective way. This image shows the being group. This is plants from people from different countries who are working in the interpretation of the genetic data. In summary, we have shown you some study cases of we can use DSI to get to know better your collections, empower their use, and collaborate with other countries in the region. Thank you very much. Thank you very much, Dr. Carvajal. I'm going to open the auditorium for a couple of questions, please. Hi, thank you. Your talk is very interesting. I wanted to ask you about the study that you did to determine the illness in cassava. Does that kind of cassava, my terms are not the best, but that type of cassava is it different from more than 1.5% in its code gene, in the gene code to others that resist the illness, that is not affected by the illness, and in that bias of 1.5% to determine the genetic redundancy, is it not possible to fall for that genetic variation that helps avoid the no, you're mixing two concepts. One study is the genetic redundancy, in which we use the 1.5 difference to tell if there are sessions that are fewer than 1.5. We say that they are duplicates or genetically redundant. For the ones that share less than 1.5, then they are unique. The study with the illness, on the other hand, is something different. Yeah, I am aware of that, but what I mean is that if I take the study of the illness and I analyze it, and I analyze the genetic information of the cassava that resists the the illness, is it? Yes, yes, yes. You mean if the materials that get sick and the ones that don't are different? Yes, yes, they are. They are different in more than 1.5. Yeah, different, definitely. 1.5% is is very little. It's very, very little. Hi, thank you for your presentation. I would like to know if there are some strategies like genomic selections and if they have been used in these crops and how have been the results. Do you use all the markers instead of only a few of them to select the best types or to improve their reproductive values? We are working alongside with improving programs on different crops because what usually happens is that they have been worked on in an independent way. So the improvers have their own markers for each genomic, for their own features, which are nutrition markers, resistant markers, and we use other markers which are not that specific, which are more oriented to explore the diversity. So we have found different markers that might be useful for them. So the idea is to coordinate, and we are coordinating so that we can talk the same language with them. Thank you very much. Any additional questions? Okay, we thank Doctor. Thank you very much. Our next talk is virtual. We welcome Alejandro Cifuentes with alimentomic and biodiversity. He is teacher of research in CSESA Madrid. He is also alimentomics, metabolomics director and the founding director of the research in food in CSSI. He has been specialized in developing advanced methods for alimentomics and food security and the utilization of bioactive compounds. He defined for the first time in a magazine, SCI, the new discipline of foodomics, and he has been included in the 1% of the best scientists of the world by the ranker of the Stanford University. Thank you very much. Can you hear me? Thank you very much. Good morning here in Cali. Good afternoon from Madrid. Well, the first thing I would like to do is thank the invitation that we received by the Universidad Javeriana, especially from my colleague Andrés Jaramillo, to be part of this symposium. And well, I have to tell you, I'm going to share my screen with you. Here we have it. I want to tell you that when I received this invitation, maybe I was on vacation or traveling, I don't know. And I didn't realize that the very interesting title, it was Foodomics and Biodiversity, when I started preparing my talk. I say, Mamma Mia, food omics and biodiversity, it would be something like the book of the famous book by Douglas Adams, who wrote the graphic statistics. He had another book called The Life, Universe, and Everything Else. So it would be like foodomics, life, universe, and everything else. So it has been very interesting to see how I can board this interesting topic. And this is the first slide I want to tell you. This is me saying how the topic is difficult to board, to approach. And this is my colleague, Andres Jaramillo, seeing my efforts. But I hope that it can take us to a nice end. Biodiversity is a very complex topic. It's a very, very complex topic from the point of view that there are many, many aspects. So we have biodiversity, food security, authenticity, processing, bioactivity, and of course, everything that has to do with sustainable production of the food industry. And mainly, as we have shown in the to preserve food security and biodiversity, it is going to be necessary to have a displacement to a diet based on plants and the dimension of industrialized food and the use of resources which we are not going to be able to afford when there is going to be a number of inhabitants in the earth of 10,000 of 10 billions and everyone wants to eat protein. That is going to be complicated.
Also, in this graph, you can also see the different dimensions of food safety and food security depends on biodiversity. And safety and security is related to stability of foods, the availability of food, and the proper utilization and access to these foods. If we are not able, for example, in order to put on the table to what we have in order to keep the biodiversity in our planet, these four aspects associated. The food safety will be too much. So the topic today, this is something that we may say complex. In terms of biodiversity, and we think about it, it refers of all of the variety of living species on Earth that includes plants, animals, bacteria, and fungi. So the global food system, it is the first on the primary driver that make lose biodiversity. So this is a continuous harm to biodiversity, the food system. And as you can see, there are several aspects that still require and that they can be very useful in terms of preserve biodiversity. For example, more than 50% of bacteria species, they remain unknown for many people. We do not know yet, not even the entire composition. So maybe something that I don't like it. So sometimes we make up the dark matter of the food. So maybe we don't have the analytical tools powerful enough in order, or maybe we haven't employed enough time in order to do this research. So or there is lack of awareness of our analytical tools, or just so this area has been studied yet. In terms of food safety and food security, requires more sensitive and analytical methods, and every time we will continue focus on new sources of nutrients. So there will be a need of a better characterization. So we have to be more focused in order to obtain proteins from insects, from algae, about microalgae, and etc. And we will also need that characterization in order to sustain and maintain this biodiversity. Besides the food, as we know, up to date, they can improve our health, of course. And the issue is that we don't know all the biological mechanisms, how they act. One of the ideas Foodomics study is that the reduction of the food waste is a stage that it is quite important. You can see, for example, here on the presentation, 1/3 of the food produced around the world is wasted or is lost along the food supply chain. So it is waste through the food supply chain end to end, from primary production to the consumer. 1.3 billion tons of byproducts and waste are generated per year. So there are waste that are generated by, and are around 14% of these food waste are coming from fruits and the vegetable production. So here, what we have been doing in the lab is that we have drawn up valorization strategies and biorefinery and green process technologies in terms of healthy with the environment, and also to obtain bioactive compounds. So I want to show you how biodiversity is important. We are biodiverse lab. In the last few years, we have had more than 150 students coming from 30 countries. And this is an example of how enriching is how biodiversity is. People that are coming from the different continents. This is very enriching and fulfilling. And also, our lab is open for new students. And by the way, for Colombian students, they are very well known in our lab, people that are dedicated, hardworking. So congratulations to those students that they are performing a good job. With all of this, a few years ago, we defined in terms of foodomics as a discipline that studies all of the food and nutrition domains by means of application of advanced omics technologies in order to improve well-being, health, and confidence in our consumers. So you can see that it has an impact. So several books have been published. And also, now there is a journal that is a baby. It's called The Exploration of Foods and Foodomics. This is so the idea is in progress, and now all the scientific community, okay, they are addressing to this and looking for information. In the Foodomics, we apply tools, and there are applications, and also we can see the genomics and epigenomics, metabolomics. It can be metabolomics, proteomics. So what we do is that we apply in order to study aspects related to bioactivity, safety, quality, and traceability, and authenticity as well. Here, for example, in terms of proteomics metabolics, what we have in the lab is in terms of the high resolution and metabolomics. For the sake of transcriptomics, there is a sequencing. We can see the NGS, and through years has been utilized microarrays, and different techniques have been applied. Also, around the systems biology, this is, for example, in terms of a structure, is not strong. The idea is to combine the relevant information that we obtained and be able to extract the essence, all of the biological information. And this is something that we have to improve. And above all, I always say to the young people, if you want to find a good job or get a good job, we have a lot of biotechnologies, bioinformatics, all these things. It's very important. Everything related to the effects. These are the main objectives and goals that we have in our mind when we are working in the lab. We have to demonstrate the potential of this discipline of foodomics in order to solve of the challenges that we have in the 21st century in food science. We also have to give support and foundations for the generation of scientific evidence that is consistent in order to develop healthy food products and healthy diets, that we can have a scientific evidence, very supportive information. There is a lot of yari data that people just says don't think that the companies are concerned about health. Some food companies or companies, they are concerned about money. A few companies about sustainability, but also here we have to change to push to the development that every time can be organized in terms of multidisciplinary and integrative approaches in order to generate and to foster a new generation of integrative researchers. When I say that they can have the opportunity to handle all those situations. Normally, you can work in a small workstation and be the expert around the world, but it has a value. There is no doubt that it's significant, but to be able to move in those areas where you are combining biological, chemical, and medical terminology, biostatistics, etc. To move there is complex. We need people to be able, with the relevant knowledge. This would be a typical workflow what we have in our foodomics lab. So we have the food with the byproducts. We have work with algae, microalgae, and food byproducts. We make use of extraction or green extraction processes which allow us to enrich extracts that are characterized through advanced analytical techniques, and then we can see the bioactivity through in vitro assays or in vivo assays, and these trials are obtained, and from these trials we also obtain the RNAs, proteins, and metabolites, and then through transcriptomics, proteomics, and metabolomics studies. And I can assure you that this is the less complex part. Everything that is complex is about data processing, statistical analysis, and also the identification of biomarkers, also the biological process, the understanding, all of that information. I want to show you this is work performed in the lab. And here I want to show you some of the topics that we do with bioactivity, colon cancer, leukemia, and Alzheimer, topics related to safety, about transgenic foods, related to contaminants and the new ingredients, about food qualities, and also traceability related to foods and ingredients. For example, in regard to colon cancer, this is something we have been working on it for several years, more or less 10 years. So these are the main steps of the whole work done. We have developed, green extraction process to obtain bioactive compounds from different natural sources, alga, microalga, food, byproducts, plants. And also, we determined the anti-proliferative activity of the new extras against different in vitro models of human colon cancer, and we have developed analytical approaches, including metabolomics, comprehensive, and for the chemical characterization of extracts. We have performed studies of toxicity by using in vitro models and metabolomics. And also, we have performed the identification of genes, proteins, and metabolites differentially expressed in cancer cells. And you can see why these active compounds were reduced, the proliferation of the development of that cancer, and the development of algorithms for the analysis of these MS-based data sets. And also we have carried out the correlation of the anti-proliferative activity of the extracts in vivo. This is an example about the development of these. We can see the advance. We are biodimensional LC times LC and liquids. And in order to analyze procyanidin coming from grape seeds, so this is a byproduct from the processes. This procyanidin is kind of complex to analyze because these are polymers and biopolymers in galette, and they give you a huge amount of compounds, and this is the chromatography that is obtained bidimensionally by combining the separation in helix and reverse phase, and by combining the spectrum ultraviolet and the fragmentation of the mass or fractioning there is a spot, we are able to identify this compound. We can see here the different fragments. In a similar way with apple polyphenols, several types of compounds, etc. The amount of the work that we have published for the molecular characterization of this biodiversity, I want to tell you something. Most of these natural sources and the samples, they are, so the students come with some samples. So we have studied a lot of samples from the different continents. Some of them have few studies in their countries, but we do a lot of work for that. For example, one of the dissertation or thesis or doctoral thesis, a student from Colombia and from the Javeriana University in Bogota, and we study a byproduct, food byproduct, mango, the calyx, the uchuva, and also the seed of the curuba, the passiflora mollisma. So the green extraction, characterization, and then anti-proliferative activity, and the foodomics assessment and evaluation. So just to have a general view, an overview, that it was interesting in terms of the activities and the result that we obtained from colon cancer. So we can see from this, calyx is enriched in different compounds with ethanolites, and these have a cancerogenic activity for colon cancer. This has been published. If somebody is interested in about these studies and research, no problem. Okay, feel free to let us know. Other lines that we have worked and we continue working is the Alzheimer. Alzheimer, as you know, is for the elderly, and of course every day we are getting older, and we will suffer from dementia. So now the situation is that this dementia is a new neurocognitive disorder and affects the cognitive function, affects the thinking abilities, and also affects your daily activities. So from this dementia, from 60 to 80% of the dementia cases, this is related to Alzheimer's disease. So there are two main pathologies of one related to the senile plaque, and also the metaboloid from the neuron, and this one neurofibrillary tangles within these nerves that you see in these cells. The Alzheimer's disease is affecting. For example, you can see that is something that it comes through the years, but that we know this is a disease that is affecting in terms of related of cytotoxicity, in terms of neurons, so this is in fact a complex disease, a multifactor. So there is no healing for this. There is no cure. In Europe, we have a discussion about if this antibody is being approved. It will be tested in Europe. So the treatment is 25 $1,000. So this can be useful for a 10%. Sí, no, ahí está bien, para subirle un poquito el volumen. Apologies because I'm doing my best to listen to him. Okay, yeah. Okay, we'll troubleshooting an issue here. Okay, gracias. Okay, okay, perfect. Gracias. So they take measurement and studies. In our case, for example, bioactive compounds that we may obtain from different sources. There may be a neuroprotective activity. For example, here, in fact, in order that you can see the work that we have performed with food byproducts and waste. Se va el sonido. Olive oil, the olive tree, the famous calyx, the seeds of the passiflora. Okay. And in Colombia, also people are interested in the studies of different plants, Carla showed, and also byproducts for the industries. Also we have different microalgae, cerulean, schistochrystis, and also applying the green extraction process. These analytical tools base in these studies. So we do, these allow us to characterize these compounds and also be applicable to analytical, and we apply in vitro bioactivity testing with different in vitro testing for neuroprotection, anti-flog, oxidant activity of nitrogen and oxygen, and also studies in terms of the blood-brain barrier, able also, but going through so well. By combining all of these data and this analysis in vitro testing, in the end, we match this in order to characterize this compound. In spite of all of this, we were able to do attentive identification, and in the end, there are some standards, and then confirm the relevant identities, etc. These are examples of compounds that we obtain from the seeds, the passiflora. This is a polyphenolic compound. These are terpenoids from the waste, on the olive waste or olive residues, or other compounds. We obtain incredible results and outcomes. It was loaded from the Alzheimer and related to the oils. Carotenoids. We can also see other that are very rich. We can see other phenolic compounds and also related to the falsification. This is more or less a summary. Apologies for the sound because it's kind of difficult to hear. And then a toxicity study was performed with the selected extract, and then the byproduct from the orange. And in terms of toxicity, in lines of, we can see, for example, here from cells, human cells, and better extracts and all the selection, then perform an in vivo trial. Then we did an Alzheimer's study in an in vivo trial with a transgenic, and there is a particularity, but this is a model a study accepted for the Alzheimer's study because this transgenic, the betaminoid for the mobility, everything related with atrophies, how mobility can be recovered and motion can be recovered. So they did a similarity study with humans. What I can say here is that the outcomes that we obtain with the orange extract, it was very interesting in terms of recovery, in terms of mobility. And of course, if we see the positive control that was up to date in natural extract, ginkgo biloba extract, and this is the recovery with the analysis with an extraction of 100 micrograms per milliliter, and the recovery is very useful by obtaining extracts of 50 milligrams, and we can obtain these extracts are quite interesting. So now we can see the positive effect. Just simply, so the idea was to understand the C. elegans when we gave them testing to eat, okay, the orange extracts. We make use of transcriptomic techniques in order to study. So there was a modification by these extracts. And also with the extract of orange in terms of lipid profiling, okay, 161 lipids. At the moment that they were consuming this extract, and this would be in terms of metabolomics, primary metabolism, in terms of helic acid, we saw 232 metabolites that were identified. I think that it was not modified. In total of lipids, 561, and the total of metabolites identified were 232 through the extract. Just you can see all the chemistry here. All this family of compounds. This would be the total of metabolites. We can see the number of liquids, 818 metabolites. And when we do the whole study, you screen and you filter all the information of metabolites. So at the end, it allows you to combine all this information and cryptomic information, and this you can obtain an antioxidant effect, an anti-inflammatory. some compounds, terpenoids, carotenoids, that they have direct effects through the ROS generation and then through indirect hormesis. So this improve the lipid metabolism that in the end improve the cholinergic transmission. And okay, you can see all the studies and what has been published. Maricas. Puta. No alcanzas a escucharle.
So to finish and to conclude with the aspects of foodomics, in this work we have this foodomics is a very useful tool that might help to solve many of the challenges that we have to preserve biodiversity and our global food system. Those foodomics can help to identify microorganism species, to better characterize the composition of foods and new foods, to warrant new sources of nutrients, to decrease food waste, and to understand the binomial food and health. Okay, thank you very much. Again to Universidad Javeriana and to all of you. Thank you very much. Thank you, Dr. Cifuentes, and we'll start with the Q&A. Dear Alejandro, Andres Jaramillo here. Andres, I'm very well. For a moment, I thought there was no one there. For a few seconds, I thought that I was talking to no one. Thank God you are there. Likewise, likewise, delighted to be here. Thank you very much. Here we have a group of people who are attending your talk, and I want to ask you something to a question like igual like the one I you again facing foodomics. I'm going to find a rope to hang myself. Um, we have had some talks by Mauricio Diaz Granados and by Monica. Taking given a look to genetic part of this topic, one of the difficulties that we face regarding food is how determine not qualitatively but quantitatively if something is useful as a food from the metabolomic standpoint. That is impossible to is that something impossible so far? Do we have a map of over 26,000 compounds out of and only a fraction is notated? So the question is, of this wide range of options that nature gives us, how can we detect that something is useful from the food standpoint to board a bit how to widen that basket of edibles that we have been reduced by the culture of that we are forced to eat but as you tell food that we are sold by money, not by nutrition. So the question is there a group that is working on metrics that help us to in a quantitative manner determine if an organism, if a living thing or a plant of the fruit of a plant have those characteristics that render them edible? Maybe metrics based on metabolites, maybe on lipids, maybe on proteins, something that in general terms tells us it can be eaten despite being some compounds that might not be good for the individual. The question is, I'm sorry, but the sound in the translation booth is not very good. we are missing some words. First, we have to make sure that the sources are not toxic, and after that, the quantitative aspect is very important, of course. Oftentimes, I give the example of garlic, the garlic, and then you have a study about it, about the compounds that come from garlic, and how thus enrichment of those compounds end up being toxic. So it's based on the dose. That is the first thing that we have to take into account. The second thing is something more related to a nutritionist than for an analyst like me. The tools that can be used to quantify that is that quantification is an aspect and the nutritional aspects are something else. Take a look at the proteins. Proteins, vegetable proteins. There are several amino acids which are essential to us, and you need them in a lower quantity. There are different aspects, which that is a question more for a nutritionist than for a chemical analyst like me. What we have is a lot of tools, and take a look. I think that that would be the typical problem that it would be very interesting to state in a project for an AI project, for the complexity that it has. I think that it would be the idea of a project for an AI, and we would need a lot of money to do that. Yeah, that's a good idea because nowadays by proximals we cannot get there, and I think that the quantitative aspect is very important. Yeah, you're sure. So you're going to go to an AI project if it's rendering very good results. We're going to topics that go beyond what I do. Alejandro, thank you very much. Thank you very much. I'm going to give space to other people who want to ask questions. Take care. Bye bye. Hi. I have a question. I think that you choose extraction by supercritical flux over the microwave wave. Is that so? It depends on the material, the subproduct that you are evaluating. No, it depends. For example, in the case of carotenoids, it works with supercritical fluids because of the polarity. But I can say that the supercritic fluid can give you a very clean extract that can be useful, but on the other side, the performance is not very good. So the requirements are better for pressurized liquids. We usually use water, ethanol to be compatible with our inventory, but we can do it using microwave, ultrasound. We are even using the combination of different techniques. There are many ways to do it, and oftentimes the decision is related to the connections that give this track with most activity. We can use it with supercritic fluids. We can use micropressure, expanded gases, and so on. Do we have any other question? Hi Alejandro, thank you very much for your talk. It is very interesting. I have two questions. The first one is, when we look at the effectiveness of a specific compound versus the extracts that have a combination of many elements, that might, in a way, boost an individual compound. What is your vision regarding the experiments that you have run and the characterizations of what could be more efficient? Dedicate a complete experimental approximation to find a precise compound, or see or look at what the extracts do without going to the detail of what there is in there to boost an effect. So we research in the sense that we try, but I always say that both approaches are okay. All the science, all the traditional science, all the natural resources, all the work that we have done to find new compounds, it has been done with the approach of trying to find a determined compound, any given compound. And that's what we have done, and it has worked. To extract a compound, purify it, characterize it, until you name it, you put a last name to it, and then you have all the information to know about that compound. That is a standpoint from pharmacology. But our approach is a little bit different. That pure compound is not what we aim for, that is the mixture of compounds. For example, the Alzheimer extract, we have multifactorial pathologies, and we are interested in those mixtures so that we can attack different aspects of the illness. For me, both approaches are valid as long as there is some solid foundations behind it. If we have very powerful analytic tools that allow us to study several compounds, always knowing that a natural extract is one of the biggest challenges for an analyst. There is, I always wonder if there is a compound or a concentration below the pressure limit, maybe it wouldn't have any effect at all. So that is something that practically we have to live every day, the sensitivity to different compounds. Thank you very much. The other question is, taking into account everything that you have shown to us, working in different compounds, different techniques, in that regard, in the institute, we are starting to characterize foods, aiming to do that in an efficient way. We would like to know if there is a person in charge of everything, or each project has a group of people. No, what we have is a structure in the group. We are a big group. We are a big group. It's about 30 people. I don't do anything because I'm the boss. Just oversee the work. The merit belongs to the other ones. But basically, what we have is specialized people in each one of the compounds that we have. There's people from the expansion line, cell line. We have other people in the ómicas line. So in that way, each one takes care of their job, and after that, what we do is gather with some frequency and have some meetings as executive as possible, short meetings. But it is very good to when we have those big groups, some it is very important to meet each other, to have some collaborative work. That is the dynamics of the work, because people come up with a with the people have their own networks with people that they have not met in different countries, and it is very interesting to connect with the others, and that is very healthy, and that I think that is very healthy. Dr. We thank Dr. Alejandro with a round of applause. please. Thank you very much. I love Cali, by the way. Let's go to a short break, 10 to 15 minutes, and come back for the next presentation. We apologize for the translation, but the quality of the sound in the translation booth is not good. Apologies.
Ahí yo estoy hablando. Ahí yo estoy hablando. ¿No me escucha? ¿Sí me escucha ahora sí? Listo. Gracias. Gracias.
at the ómicas Institute, or ómicas Research Institute at the Pontificia Universidad Javeriana in Cali. This is a relatively new institute focused on the idea to understand how to assemble materials, matter in general, from the constructive elements of matter, and that's atoms. Once we understand how that assembly process can be executed from the bottom up, we would start solving a lot of the problems that we have in general as human beings. Now, again, learning from nature, we are interested in looking at how omics science in general, by characterizing an organism, how we can understand it better in terms of how it interacts with our own tissues. One particular I'm very excited about is what we call the PTFI. We are a set So PTFI is bringing a lot of this, futuristic idea of really worrying about what we eat and about the idea that food is health. We're making very good strides to analyze local foods that are indigenous. We're also making good strides to normalize the characterization protocols that we use internationally to understand the composition of foods. So I welcome everybody to visit us at the Javeriana, at the Institute, and to continue doing good science.
Buenas tardes, les agradezco tomar asiento y...
Good afternoon. Please take a seat and mute your phones.
We'll continue with research on cadmium in cocoa, what role for soil amendments by Miriam Pullerman, who is a senior scientist at Biodiversidad International, SIAD Alliance, and Wageningen University and Research in the Netherlands, with expertise in soil management and sustainable agricultural systems. Her research focuses on improving soil health, enhancing functional biodiversity, and promoting agricultural practices that help mitigate climate change. She also works on regenerative agriculture and cocoa quality assessment and food security, with an emphasis on sustainability of agricultural systems in tropical regions. Miriam, can you hear me?
Yes, I hear you. Do you hear me?
Yes, so it's all yours.
Okay. Thank you very much for this introduction, and good morning, everybody. I'm very sorry that I cannot be there personally. That would have been much more fun, I guess, but thank you for giving me the opportunity to present virtually from the Netherlands. So as already introduced, I will talk about our research that we do at the Alliance of Biodiversity and SIAD on cadmium in cacao, and this is based on 3 different projects that we are implementing in Colombia, Ecuador, and Peru. The Clima Loca project, the STDF project, and the Regenerative Agriculture Demonstration project funded by Rockefeller Foundation. So for those of you that are not familiar with the cacao sector maybe, or with this problem of cadmium, so cadmium is a trace metal that originates from the Earth's crust and is naturally present in the environment, so also in soils and in water. So this means that cadmium can also be taken up by plants and also animals and humans. It does not have any known biological function in living organisms, and it's potentially toxic. In humans, the accumulation of cadmium over time has been associated with different negative health effects. Smoking and food are the main sources of human intake, and especially rice, wheat, and potatoes, because this is what we consume in large quantities. And Theobroma cacao is it? Yes. You don't see me. Oh, okay. Just a second. Let's okay. Yes, I see the problem. And you should. Yes. But do you also, yes, now it's the slide. Okay, thank you for that. Okay, so cadmium accumulates in cacao. So cacao is this capacity to take up significant amounts of cadmium. And this is why chocolate can also contribute to cadmium exposure. So yeah, cadmium present in the soil can be relatively easily taken up, is relatively mobile, can be taken up by the cacao plant, is distributed within the cacao plants to the leaves and the beans, or to the leaves and the fruits, and also accumulates in the beans. And then also during the cacao process, post-harvest processing process, where the different steps are followed to produce the chocolate. The cacao beans are mixed, and in the end, that determines also the final cadmium content in the chocolate, also depending on the amount, the percentage of cacao that the chocolate contains. So because of this toxicity of the prolonged human exposure to cadmium, food safety regulation has been introduced by different cacao importing countries, including the European Union, the one that you can see here on the left, but also in the US, California has a regulation on that limits maximum content of cadmium in cacao products. So the regulation is on the products that are sold to the consumers. And the difference is in Europe, there is a maximum limit. And beyond that limit, the product cannot be sold in the market, basically to protect the consumers. And in the US, it can be placed on the market, but it has a label to warn the consumer that it contains relatively high levels of cadmium. So because of this problem, so the first regulation was introduced in 2019, but already years before the countries knew that this was coming, and also knew that there is relatively high amounts of cadmium in cacao from Latin America as compared to cacao from other parts of the world. So with the Clima Loca project and the STDF project, we started mapping cadmium in cacao. And also we started mapping soil properties. And what you can see here on the right-hand side is the map with all the points that we have sampled so far in the project across the 3 countries where we are working. And why are we doing this? Why is this mapping important and understand where the cadmium is occurring? So we can calculate how many farmers are potentially affected and where they are, but because the cadmium is not everywhere affecting cacao production equally. This helps the countries also to develop policies and planning. For example, expanding cacao production in new areas where very high levels of cadmium are found would not be like a very good idea, probably. And also we want to understand better the underlying relationship. So what is the relation between geology, soil formation, relief, landscape features, and the cadmium, where the cadmium occurs, so that we can better predict where cadmium problems may occur, and also to target mitigation strategies. So what could be the best strategies to mitigate the high cadmium levels in different locations? So this mapping, so we have in the project, we have collected data partly from existing databases in the 3 countries, and also a lot of new sampling, especially in Colombia, because that is the country where hardly any data were available. We sampled 4,400 different locations in Colombia, Ecuador, and Peru. And then overall, we found that in Ecuador, about 35% of those sampling locations had more than 0.8 ppm in the beans. In Peru, this was about 32%, and in Colombia, 48% of overall. And why 0.8 ppm? So we know that the regulation is not on the beans, the regulation is on the final product, but 0.8 ppm is like more or less a value that we can consider, is considered acceptable by most of the clients that would buy the cacao beans, but that of course also depends on for what kind of chocolate the cacao beans are used in the end, if it can be mixed, or whether it's especially chocolate that cannot be mixed. So with this, we are making maps. We are making digital soil maps of soil properties, and we are making maps of cadmium in the cacao beans. As you can see here, examples for Ecuador. We are still working on the one for Colombia. So this one will be released in we hope in early 2025. Sometimes people also ask, where does the cadmium come from? There is like a concern that the cadmium in cacao could come from fertilizers, basically from rock phosphate fertilizers, but also organic fertilizers. This can be a source of cadmium, but what we have seen so far, looking at all the data on the cadmium distribution and where it's dominantly a natural problem associated with variation in geology and soil formation. And here we see an example of the red. This is the data for Colombia. We have not mapped this as such, but you can see the hotspots here. So the red areas is where the highest levels of cadmium in beans and soils are found. And this is also where we see that according to the geological map of Colombia, these areas often have sedimentary rocks, shales, that are naturally high in cadmium. And also this is very likely the source of cadmium in the cacao in those areas. So we have also looked at the socioeconomic impacts. We have done a study in the 3 countries, and the 3 countries, because the cacao sectors differ very much, the value chains differ very much. The impact, socioeconomic impact on cacao exporters and also on farmers are different. What we have seen is that not all farmers are affected. Not all farmers are aware of the problem either, no? But we see severe localized impacts on farmer incomes and also on the exporting companies in areas where production of specialty cacaos coincide with high cadmium content. So in those areas where mixing is not really an option. And we also have reported increased cost of analysis of cadmium, especially by companies, cooperatives that export the cacao, and reduced. So in Colombia, which is the country that exports least of the lowest proportion, let's say, of the cacao produced, the effects overall are relatively minor because there is not a very high export to Europe. But we do see that in areas with high cadmium content, there is a reduced investment in strengthening of the cacao sectors, and that's of course going to impact those areas. We also see that with time, similar regulations are also established by other countries, and adoption of those new regulations will further limit the adaptation capacity of the actors in the sector. So solutions. So what can we do about this? So basically two main strategies. One would be prevention and the other one mitigation. So prevention means like using the improved maps that become available. So those identifies those areas with high risk of cadmium and low possibilities also for mixing, and prevent further expansion of cacao in those sectors and in those areas, and look for alternatives. And then mitigation. What can we do in those areas where there is already cacao production, but the cadmium accumulation in the cacao is relatively high, which is definitely not everywhere the case. So measures could be taken potentially at the pre and post-harvest stage. So pre-harvest, of course, like we can try to find low cadmium accumulating genotypes, both rootstocks or science, or the combination of the two. And this is what our partners in Klimaloka are also working on. We also work with manipulation of soil properties. So especially through the use of soil amendments, we try to change the properties in the soil that affect the mobility of the cadmium or the uptake of the cadmium by the plants. And this is where we'll go a little bit more in depth in this presentation. Since I'm a soil scientist, this is the part that I'm mainly working on. And then there are also other organizations and companies as well that are looking at fermentation processes, for example, temperature or the inoculation with certain microbial strains, if that helps to reduce cadmium. There are also strategies related to agroforestry and phytoremediation, but those, let's say, probability that this can be effective are basically relatively low. And then the last one is also post-harvest, just like the fermentation one, but this one is about the mixing, what I already mentioned. So we also need to consider that the solutions should be integrated solutions. We should consider that looking for new genotypes can also mean that other properties are affected. So the same with the soil amendments, we change soil properties. So this can reduce cadmium, but it can also affect the productivity or the quality of the beans in other ways. And all of these strategies can have their trade-offs and synergies. And of course, we need to understand those and look for the synergies. Yes, so regarding the manipulation of soil properties, we need to have a good understanding how cadmium behaves in the soil plant system to develop targeted, like, let's say, hypothesis-based solutions, or test hypothesis-based solution. Part of the problem here is that the behavior of cadmium in soils plant system is complex, and relatively, there is still a lot of processes that we do not fully understand, let's say. So what we do know is that cadmium in the soil is present in 3 different pools. So we say the directly available pool, which is the cadmium in the soil solution, which is normally a very small amount. And then the reactive cadmium pool, this is the pool that is potentially available, that is bound, that is absorbed to the reactive surfaces in the soil, organic matter, clay, metal oxides, and that can be exchanged with the soil solution. So when plants take up the cadmium, it will be there will be new cadmium coming into the soil solution. So the movement from the reactive to the available pool depends on soil characteristics, including the pH. And sometimes we use the transfer factor, which is the cadmium content in this, this is the other way, right? The cadmium content in the beans divided by the cadmium content in the soils, as a sort of, as a measure of how available that cadmium is in the soil. And this is generally, this transfer factor is generally higher in low pH soils than in high pH soils, but not always. And plant uptake from the soil can also be affected by the availability of other elements in the soil that share similar transporters, for example, zinc and manganese, iron, and there can also be competition with calcium. So we are testing different kinds of soil amendments that change the soil properties in a way that we could expect that the cadmium availability in the soil would decrease. And some of those amendments may also have positive effects on the productivity of the cacao. For example, with liming and acid soils, we increase the pH, we hope to reduce the cadmium availability, but we also hope to improve the availability of important nutrients, for example, phosphorus for the plants, and that way we can improve the productivity. So we are testing different kinds of amendments in this kind of controlled field trials with different blocks, and we are doing this in different locations across the 3 countries where we are working. And this is just one example in Colombia where we are doing this. This work is ongoing. What is very important about this work also is that we don't monitor only cadmium, because in the end, to come up with solutions for farmers, we also need to understand these trade-offs and synergies that I mentioned before. So we're also looking, how do these amendments affect soil health? How do they affect cost benefits for the farmer, but also the productivity? And we are now also starting to look at the quality of the beans, how it's affected, because this in the end all affects the profitability and environmental sustainability of the cacao production systems. Here are some fresh results from one of the trials in Colombia. As an example, we can see here that different kinds of treatments are being tested, including liming, different kinds of organic matter to all with the idea to improve the soil fertility, but also to reduce the cadmium availability. So different combinations also with fertilizer applications and a control without any applications. So we're working in plots where we collect every 6 months. We collect soil data, leaf data, and bean data. We do soil characterizations, including cadmium. We do foliar analysis, nutrients, and trace metals in the leaves. We also look at cadmium in the beans, and also we measure yields of the cacao. We record the production cost, and this trial has started in December 2022, and we have now data completed until April 2024. So this is total duration of 60 months for this trial. So here are some preliminary results, and you can immediately see very high error bars. I have to say these are standard deviations. Yeah, but still the errors are high. We see on the left-hand side the average, the total cadmium in the soil, and on the x-axis we see M0, M1, M2. Those are the different sampling rounds. So every 6 months we did a sampling. And you can see that within the treatments, the total cadmium in the soil doesn't change, but there is quite some spatial variation in the cadmium content. But this is normal. Total cadmium doesn't change because that is the largest pool, and what we are interested in is in the pool that is actually available or potentially available. We also have looked at the data of the cadmium in the beans, and basically we look on the differences between the control, which is C0, and then also the differences over time. We see that that in most treatments there tends to be a decline over time, but when you look at the difference with the control, there is no significant differences. This is then the data for the transfer factor that I mentioned. So the cadmium in grain divided by total cadmium in the soil, and also we see it's very variable in time, also in space, but we do not see significant differences with the control after 16 months of trial. So if we look at the results that our colleagues in the other countries are obtaining, and also their trials have were already established earlier, we see that in general it takes quite a long time before results of soil amendments really become prominent in the beans of the cacao. But we also see consistently that there is a very high spatial variation, and that there is often quite inconsistent results, like some treatment works in one place but not in the other, and sometimes that is easy to explain, but sometimes it's not. We see, for example, that in very acid soils, then application of lime over time starts to show good results, but for some of the other treatments, for example, organic matter, and the results are not very consistent. So based on this work that will be continued, of course, for a longer period of time, but we are also really looking for ways like how can we better predict cadmium availability and better explain the underlying mechanisms and processes to not, be testing strategies for a mitigation that are a bit like a black box strategies to some extent, and a bit of a trial and error. And so one of our PhD students at Wageningen University, he is really looking more mechanistically at the behavior of cadmium in the soil plant system, using a combination of measurement modeling and isotope tracing studies. So basically, what is his PhD thesis about? So, well, we know that cadmium, total cadmium, is not very sensitive to changes in soil management, and it's also not very strongly related to cadmium uptake in cacao. So what we are really interested in is the bioavailable and the reactive cadmium pools in the soil. So we can measure those, but that is relatively costly, and also let's say that's relatively costly to measure that, and there are different methods, and there is not always consensus about which are the best methods. And so one of the questions is like, how can we best measure it? And another question is, can we also apply geochemical models that describe the cadmium behavior in the cacao soil system? And could that help us to explain the results or to predict results of treatments with soil amendments on the availability of cadmium in the soil and the accumulation in the cacao beans? So basically, Wietz, together with Orlando Idarraga in the lab at the Alliance of Biodiversity SIAD, they measured reactive cadmium in the soil with different types of simple chemical extractions that are commonly used for measuring cadmium in soil. And also Wietz implemented an isotope dilution experiment with cadmium 111 spiking to determine the exchange of cadmium between the reactive pool and the available pool, and that way he calculates like an E value that gives us information about cadmium availability as a sort of a reference value. So they did this with database of cacao soils in Colombia from 34 farms in 6 different departments. And they selected the samples from the mapping exercise, and they selected them to be very diverse in pH, organic matter, and total cadmium. And for all those sampling sites, we had cadmium in soil, cadmium in plants, cadmium in the leaf, and cadmium in the beans of the same tree. So with that, they tested whether with these different approaches for measuring cadmium availability in the soil, they could predict the cadmium in the leaves and in the beans of the cacao plants. So, and then they also did that also for the E value based on the isotope method. So what you can see here, maybe a little bit small, is that the different, so the different rows show the different pools and the different types of methods to quantify the cadmium in this pool. And then in the 2 columns on the right, you see the R square, the correlation with the cadmium in the leaves and the cadmium in the beans. So for now, let's focus on the cadmium in the beans. So you see that if you have data on total cadmium in the soil measured with agua regia, you can, the R square, so 49% of the variation in cadmium in the beans can be explained. If you look at available cadmium, so cadmium in soil solution, we see that the correlation is 7, or the R square is 0.7, meaning 70% of the variation in the cadmium in the beans can be explained. However, measuring cadmium in the soil solution is you need ICP-MS method that is not available in most of the labs, and it's more costly, so that's not really feasible to do that across like all the sites. So when you look at the reactive cadmium, the different kind of single extractions did show a little bit better prediction than the total cadmium, and especially the E value showed that. We also saw that the best performance was with the nitric acid method and with the Melis 3 method, and the latter one especially in higher pH and low soil organic matter soils. So what about modeling? So WITS has also been using models that predict the cadmium in the different pools depending on the quantity of reactive surfaces and the adsorption strength of the elements that we are interested in this case, cadmium. So he does that using a multi-surface model with sub-modules for organic matter and oxides. With that modeling, he found that the bioavailable cadmium model, as predicted with the model, so not measured in the lab, explained 74 to 77% of cadmium in the beans. So basically, the conclusion from this work is that we can explain cadmium in beans using empirical models, but that requires quite a bit of data, particular total cadmium, soil organic matter, and pH. We can explain quite a bit of the variation. And if we use geochemical models, we can also explain that amount of variation. But with the biochemical models, the geochemical models, we have also the advantage that we can predict the bioavailable cadmium and the speciation, or in other words, what surfaces the cadmium is adsorbed. And that gives us insights on the mechanisms of cadmium adsorption in different soils and different places. And that can also give us more clues about how to target the solutions. Yes. Okay, thank you for that. Well, I was at the conclusion. So concluding is, my conclusions are that cadmium regulation has strong socioeconomic impacts on the cacao sectors in Colombia, Ecuador, and Peru, but the impacts and the solutions are highly context specific. The science is incomplete, but we are advancing step by step. And when we think about solutions, we really need to consider the trade-offs and seek synergies between cadmium mitigation and other objectives that farmers have, other challenges that farmers have to develop feasible solutions for the smallholder cacao producers. So we need to consider soil health, productivity, profitability, and then also the product quality. Integrated strategies are needed. So just one strategy will not work everywhere. So prevention and mitigation, including the pre-harvest and post-harvest processes are needed. And then also the last point is the question for what about other crops, which is coming up recently. And then, and what about omics? So as I said, we are now starting also to look at the bean quality. So how do mitigation strategies for cadmium affect bean quality? So we're looking, we have started a study where we are looking at different soil amendments and fertilization strategies, but also different fermentation protocols to see how it affects the elemental composition of the cacao beans in terms of heavy metals and nutrients, and also molecular characterization of potential differences in metabolic or fatty acid profiles. And with the Cacao of Excellence lab of the Alliance of Biodiversity SIAD, we will also look at the sensory quality. So no results to present, unfortunately, yet, but this will be coming. So thank you very much for your attention. And yes, if you want to know more about the project that I presented, well, you can contact me, of course, but you can also check our website. Yeah. So thank you very much for your attention. I'm not really sure about the time and whether there's time for questions, but if so, then happy to take them.
Yes, thank you, Dr. Pullman, for the presentation. We're going to open the floor for questions. So if you have a question in Spanish, we will provide the translation. But here's a
Oh, no
question
worries. No need to translate.
Okay, perfect.
Yeah.
Hola, Miriam, Andrés Aramillo. So since I know you speak Spanish, I'll switch to Spanish. Okay. La pregunta es, ¿cómo ves la ruta de edición genética
of genetic addition to try to change the transportation center from the protein MRAM, the one that we are trying to explore now with Paul and his group at CIAT, understanding that it is the same protein that is in charge of transporting iron and magnesium. What we are trying to do is to create a mutation at the channel to make it a little bit more narrower, because cadmium molecule is a little bit bigger, is significantly bigger than iron and magnesium. The question is, how do you see that route? Do you think that it could work to create a new species of cocoa and maybe some other plants that have the same problem? Yes, that is a very good question. I think that you have more answers than I do, because I am not the expert at physiology and genetics of cocoa. I work in soils, so I can rephrase it in another way. How much variety has been promoted of cocoa? How many varieties do we have? And in the process of selection, do we have variables that are more prone or less prone to transport cadmium to the fruit? Yeah, it's very likely that it is like so. In the local climate, what we are doing in Colombia with Sagrosavia is different research strategies. So one of them is a survey of the commercial clones in the producer farms to check if there are some clones that accumulate more or less cadmium. But so far, what I understand is that we don't have very good results in that regard, because that work as the one that we are doing with agendas is very complex due to all the variability of the space that there is in the soil. So at times it's really difficult to tell apart the differences between cadmium in the clone only by the variations or in the soil, or because that's a genetical feature of the plant. So my colleagues are researching on that, so I hope that soon we are going to see some results with enough accuracy to have identified this clone. So it also could be that these materials do exist, but not of the commercial clones that we are producing right now. So probably we are working with the development of some markers, molecular markers, in order to screen the materials that we have in the genetic connections of cocoa. So we are working in different strategies, research strategies in this topic. In Climaloca, we are not working in that regard, but in the Alliance, they are. Thank you very much for accepting our invitation. There are a few more questions from the rest of the audience. Can you hear me well? First of all, I want to thank you for your talk. It is very interesting. My question is related to the topic of solutions. When we're talking about prevention and mitigation, my doubt is about the plants that are already planted and the cocoa that we are harvesting right now? Is there any way to treat it to reduce the cadmium levels? I understand the cadmium is accumulated in the inner part of the cell, so maybe in the milling part to produce to produce powder. Is it possible to treat those? What we are doing, but this is also work from other institutions and also with AgroSavia. I know that Roxanne is also connected, so maybe Roxanne, the next presenter, is going to be better at answering. But to reduce cadmium in after harvesting, what has been done in a university in Belgium and with AgroSavio and Casaluca is use different strategies for the fermentation with different temperature, different acidity levels, and with microorganisms that can. There's one paper, I think, that is coming up soon that shows that there is possibility to reduce the levels of cadmium. For instance, cadmium, which is at the center of the bean, can be moved to the outer part so that it can be removed when they peel the bean, at least part of it. So this one strategy that could be followed. Thank you very much. Thank you very much. We're going to continue. We don't have any more time. For this strategy, we have to see how the quality of the cocoa is affected, of course. We have to take that into account also. Thank you very much for your question. We're going to continue. Let's give a round of applause to our presenter. Thank you very much. Our next talks, phylogenetic characterization of Colombian biodiversity by Roxana Jotan. She's a doctor in evolutionary biology in University of Paris, where she studied the blooming of tropical plants. She is affiliated to AgroSavia, where she has a lead role in the development of potato and cocoa. His recent work has focused on improving the production of cocoa in Colombia using molecular tools to optimize the quality and the resistance of the crops. And she participates in the conservation of diversity, collaborating in different magazines and different international projects. Thank you very much. I think, please give me 1 minute because I'm not able to share my screen. We wait for you, don't worry. Yeah, it seems to be working now. Now, can you see it? Yeah, I think you can. Can you please confirm if you see my screen? I changed the title of my presentation because it's the characterization of preserve agrobiodiversity in our bank for the nutrition and agriculture. That is a bank which is preserved by AgroSavia, but it's a bank of the Colombian nation. I'm going to share with you the works that we have been undergoing to know this preserve agrobiodiversity and to get to know its the germplasm bank is preserving different centers that we have in AgroSavia, which are distributed all around the country. So we have a main center for seeds, which is in Mosquera, Bioitatá. We have another one in Palmira, another one in Rio Negro, Santander, Rio Negro, Antioquia, in Tumaco, Nariño, and the banana production zones in Magdalena and Puerto López Meta and Montería, Córdoba. In all these places, we preserve living species because it is not possible to preserve them as seeds. So what we do is we have these living collections that are located in these places. And we have about 50 researchers involved in conservation and curating these collections, in research and in documentation of these collections. Besides that, we have 275 species of plants which are preserved, some of them with more accessions, for example, of corn, potato, bean. The ones that we usually use in Colombia, we have many species, but for other species which are very important, for example, as Mauricio said at the beginning, we have very few collections, and the idea is to increase the number of collections. And we have 36,600 accessions, which are different samples, which are different, and that represents a line or something. So we have different systems to preserve these collections. We have in seed, in vitro, and in field. The seed collection is located in two centers, in Tibaitatá and Mosquera, close to Bogotá, and La Selva, which is in Rio Negro, Antioquia. Its curator is Manuel Guzman. In vitro, we have 1,471 accessions. It's mainly in Tibaitatá, and its leader or curator is Alba Lucia Villa. And we have in field 4,685 accessions of the species that cannot be preserved in seed or in vitro, or that we have not been able to preserve in seed or in vitro, and which are in 9 different centers. Its curator is Duber Cañar. The idea of this work that we have been doing, we have several groups that work in this bank, is to study and organize these collections, these plant collections. Not only is the idea to characterize these collections, but also to have a information system that allow us to know what we have, and to transmit that information in an information system which is a green global system. So this effort that started in 2016 has moving forward, because before we used to have the collections not in a system, information system, but in Excel spreadsheets or notebooks, and in that way it is really difficult to promote its use, to promote the use of these collections to collect for the producers. So this bank, this BGV, was created in 1948. In 1994, it was translated to the ICA administration, to Corpo ICA in that moment. And now we are called AgroSavia. Back in 2015, all this information that was stored in Excel, in spreadsheets, was then stored, and then the idea is take all that information, gather all the information, and put it in this information system called Green Global. So this is a very long process, but we have a significant progress in this process. What are we preserving in this collection? We are preserving generally most of the collections were gathered between the 60s and the 80s. And generally speaking, these are crops, these are traditional crops. And they become from producers, and we have been collecting from them. And we don't have that many wild material in the collections. After that, there have been very few collections along the years. And there are some introductions from other countries. For example, in the potato collection, which has been 72% revised, we have different materials. And most of them, they come from farmers. And there are some others that come from the market and from introductions. And very few come from wild material. Only 7% of that is wild material, 4% is improved material, and 89% is traditional crops. So the collections have been in the Andean region, as we can see here in these maps that I can show you now. After that, we have done an effort in this characterization of this germplasm. The idea was to implement a platform that allows to study and systematize that biodiversity of the collection that we have been preserving in our bank. And the idea is to be able to promote its use, make it visible, and foster collaboration within AgroSavia, but also with other institutions. So in order to do that, we made a selection of the species. As I told you, we have 275 species. We cannot characterize all of them at once, so we have to prioritize some of them. To start doing this, what we did was start with cocoa, then avocado, the musa species, banana and plantain, chontaduro, potato, and palm trees. So we tried to do that characterization to have the information about diversity, the footprints, the genetic footprints, the duplicates that we can have in the collections, take a look at the genome and understand its diversity to be able to tell the researchers as to the producers. See, these are different species that are diverse. You can start from there to understand the agronomic traits that might be important for you. Look, resistant to illnesses, the hydric stress, abiotic stress, productivity, and innocuity traits, as Miriam was saying, related to heavy metals, for example, low absorption of heavy metals. And then try, starting from those molecular data, try to have a selection program assisted by markers trying to compile most of the data so that we can have a genomic selection and achieve genic addition in these collections, and of course help improving the genome of these crops, as well as the conversation and its use and the use of these collections. This is the team that has been working in this characterization of the molecule on the molecular level of the genome. And we have several people here. You can read their names over there. It's a very strong team in the molecular level and bioinformatic level. So we have been publishing different studies, and we have had some different studies in cocoa, potato, avocado, banana, also in uchuva. I'm going to show you some information here because we don't have enough time to show you everything. Regarding genetic diversity of potato collection, this collection has about 2,400 accessions, but half of them are preserved in field that are in field all the time, and there are the 1,200 accessions that can be easily characterized because the rest of it is in seed, and then you have to do the preparations and propagate the seeds and be able to extract and evaluate in field. So this genetic diversity was made through some sneak arrays, specifically tailored for potato, and the idea was to identify which ones are the polyploids and haploids so that we can tell what was the diversity that we had in this collection. So what we did in our first effort, we studied the diversity of 89 accessions, and with some more recent ones, we increased the number to 480 accessions to achieve a total of 1,289 accessions, and we have different groups of this Indígena Pureja Papa, which are the local variety Chaucha, which is another one which is difficult to. It's called Chaucha and Guata, but actually they are the group of Indígena Pureja, Tuberoso, or some other ones without classification. The idea was to have this classification and see how this diversity was preserved in potato. At the end, we found in this genetic structure study three big populations, which are not pure. They are not only purejas or andígenas or the other type, which are guatas or chaucha. They are mixed groups, but that they have a big diversity between them. The most astounding that is that the purejas, which are diploids, are separated by their group, and the tetraploids are differentiated in two different groups, which are very diverse. The group A is the least heterozygous, and the indigenous is more. The two groups of indigenous are much more diverse among them. Since we had a collection of 1,200 accessions, we wonder how we can run a phenotypic evaluation that has different agronomic traits and that can be run in depth, because the problem is that when we do that, you go with that characterization, it requires a lot of resources. So the idea was to use this information to do some connections and tell the researchers, you have to be in charge of the evaluation. Look, use this collection of a smaller nucleus where you can do all the phenotypical characterization. So we did a study in which we used the genetic diversity at a molecular level of potatoes. We compared the agronomical traits, the few data that we have. We had to check if it matched this. So we divided this collection in different groups: 20% of the collection, 50%, 2%, 3%, to check if we were losing genetic diversity in that connection and not in the genetic diversity, not even in the phenotypic diversity we lost in any of the collections. It was created the diversity. So what we proposed,
10%, that is around 120 accession and a nucleo of 3% in order to assess this agronomically assessment. So this is more or less part of what we have been doing in the data collection related in terms of the potato. I will show you another aspect in regard to avocado. We have more or less accessions 140, and due to loyalties projects 240 locals materials about porta draft in order to see the genetic diversity in a global view according to what we have in Colombia. This is the layout that you can see in the area based on the accessions, and this relevant information was published last year. So we try to apply this information to see the source when the diversification occurred about the avocado diversity in Colombia took place. So we did this through GVS, genotyping by sequencing, and we obtained 4,931 markers for these accessions, and also we gathered genomic information from reference group related to avocado. So in a previous study and research of a Spanish group, they characterize other kind of materials in which they have a reference group, not only from Central America and Mexico and Guatemala, but the Antillas that are the ones recognized for avocados. We collected this information with our genomic information in order to see what was expected and what could we obtain. By non-parametric analysis that was performed, we obtained one genetic group, the Colombian one that you see there. And this group, we also did phylogenics, yes. And there are two groups that is supported. So this through the phylogenetic studies, because according to what we had, our Colombian passport, the data collection, according to that, in our Colombian, it has accessions that are pure natives, very Colombian ones, related to the Antillanas, then also to the Guatemala. We also wanted to see, by means of an estimate Bayesiana, we wanted to see what was the scenario, and that this Colombian population, this is a cluster that it is derived. So what we see is that there is a probability that the source of avocado was from the Mexican population, then the Guatemala, and then the Antillana and the Colombian one that are related among them. Also, we wanted to see what was happening with the rest of the germplasm banking. There were some of them that they were from Guatemala, 100% Mexican and 100% Colombian and Antillanas, but there was another from the group, 44.2, that has a hybrid ancestry. So they belong to these groups. 44.2% has a hybrid ancestral. The other thing that we analyze what happened with the other Porta inherto or rootstock. So that hash is something that is consumed, and there are rootstock that are native and adapted to the different ecosystems where avocado is harvested, because they are more resistant to diseases. So the cinnamomum spectactora. This destroys the plant if it doesn't have any tolerance to this disease. This 57.3 is Colombian, and they have pure ancestry. We wanted to see what happened through the time, and this program allows us to see when this Colombian population from the Antillanan population, I think that there is an estimate based on the studies, more or less 30,000 years was the divergence in Pleistocene, during the Pleistocene, and it was before agriculture activities started. This was many years ago, so the migration and the settlement and establishment of the agriculture in the American population. It looks like this diversion in the times of agriculture, of this divergence. It was before the agricultural activities started. Another thing that I wanted to show you here is about the activities that we perform in cacao in terms of the genetic diversity and also the genetic improvement that we have been done in agrosavia. So the main idea is to be able to respond to different kind of issues, which is considered a fine aroma, also but low production in our country, from 300 to 500 kilograms per hectares, because we have some constraints related to pests and also diseases, and we have unproductive areas between 30 and 40 years, and now auto incompatibility and the cadmium absorption and also exportation to different countries to Europe, and also climate change that is affecting the behavior of cacao crops. But we are focused on working on cacao particularly because the great number of farmers are small producers. There are families that they depend on this product. So since 2015, there was an establishment of genetic improvement related to cacao. We wanted to have it before, but this strategy, or this new strategy in terms of genetic improvement, this is led by the improver that is in the CIAT, Karen Rodriguez in Palmira. This is a recurrent selection strategy, is to make use of this genetic diversity that we have in the banks, in the gene banks, and to make use of that, and also to obtain improved material, and also to reduce this to that genetic improvement, that the idea is that it won't take longer in terms of 15 years. The idea and the focus is to have more productivity in this material, disease resistant and low content in cadmium. And also there are strategies that is not only about a monoculture, but also the diversification of these crops in order to have agroforestry systems, agro-diverse systems, in order to have to have the economic resources for the farmers and producers. In the germplasm banking of cacao, we have around 500 accessions. They have duplicates in Palmira and in La Suiza. And they are working in terms of genetic improvement and all the research work that is done at AgroSavia. There are 3 main centers that they have accessions, Palmira and La Suiza. And La Libertad, they have wild pair relatives. That is, I don't know if you have seen TB color, grandiflorian, and Herrania is like the sibling that has some characteristics for these crops. The idea is to foster and increase this accession in the germplasm banking of cacao in order to utilize it for the genetic improvement for cacao and the other species that can be cropped or harvested. So the idea was to perform a genetic diversity of the germplasm of cacao, and in a parallel way to perform a study of the materials in terms of monila, escoba de bruja, blood corn, all of those typical diseases that are more that have been identified, and also about pests that is affecting cacao, to see also the quality in terms of productivity and heavy metals absorption, particularly cadmium. And the main idea was to have a genotyping. At first we made use of GBS. Now we're moving to gene sequencing, and also specific features. We also transcriptomic in order to see interaction, plates, and heavy metals, in particularly in cadmium. The idea is to have this collection also for biodiversity, resistant to diseases, quality, productivity, low absorption of heavy metals, in order to have this collection for the proper use in terms of genetic use, and foster support in order to obtain information and have a selection. When identify these genes through transcriptomic and genomics, there will be a moment to do genes editing of some specific features. The genetic diversity of the bank, germplasm bank, Jaime Osorio and John Verdugo, they are the ones, and they did the study and also joined with the CAT, the total connection of 500 accessions. Then by Fluidan 96 SEMPs, we identified 2 groups, Colombian groups, public, which we have to continue seeing what we have, and we continue working on it in order that we can see something specific. For example, I don't know, better adaptation of these groups to a certain agronomic or agronomical features in order to improve the cacao crops and harvesting. We also did genomics association studies that were performed, and they were published in 2020, and the idea was the genotyping about for 29 individuals and incidence based on 3 diseases, mazorca negra, escoja bruja, and monilia, in order to associate this genomic information with this phenotyping or genotyping features. Also to see our genetic diversity that we have in the bank. This is a hybrid population that has different population or genetic groups. related to cacao. Also, we were able to identify 10 markers that are important, which has a role in terms of resistance to diseases and productivity. The idea is to continue exploring this. Currently, we are validating that these genes can identify resistant material to these diseases that were mentioned before and also productive material. The idea is to release these markers in order to select at an early stage, because cacao is very productive, more or less after 2 years of harvesting. The main idea is that at an early stage we can select so the main idea here is to be able to utilize these markers. So we are validating in order to release. Okay, I'm short of time, but the idea here is to continue studying. There was an index in which we did prioritization of 275 species of plants, and the idea is to continue working strongly in order to increase this collection for the germplasm banking, for the proper utilization for producers and farmers, and the relevant utilization in the whole country. Thank you very much.
Agradecemos a la
Thank you, Doctor, for this presentation. So, one question, if you have it, or anybody of you in the audience have any question for Doctor? Thank you for that presentation. I have a question. Because we have seen through different talks and the presentation, we have seen institutions that they also have the banks of seeds or reservoirs of different foods. If you share databases of the characterizations that you perform within the institutions here in Colombia and maybe around the world. The idea, yes, I mean, the issue is that the thing that is related to information is not to have it collected only in one place. For that reason, we have an effort in order to gather the information in the Green Global System in order to share easily this information with the different institutions that are interested and in general with the public, because this germplasm is from the nation. Germplasm banking is from the nation, so it's something that we consider is important to share the information and also to hand in that kind of collection if it's requested. So this information, I mean, some to a certain extent is public. Now at the moment the other part, it is compiled, it is gathered, okay, in the data system in order to be published. Thank you, Doctor. Because of time, we will continue. We want to thank you, the Doctor, with a round of applause. Thank you. The next, sugarcane genomics by Dr. John Jaime Riescos, who is the director of the problem of varieties. He has a doctoral degree in molecular biology, and he has been working in biotechnology and improvement of sugarcane. He has received an award executively, and he has led different projects financed by the World Bank and called Ciencias. Good afternoon, everybody. This is not the most interesting time of the day. I want to be brief and to tell you a story that maybe can keep you connected. I know the time. But according to the topics that we have been discussing today, this is about genetic diversity as a motor for sustainability and productivity in Valle del Cauca. The idea is to contextualize sugarcane harvested in Colombia. It is grown in 5 departments, very near from Valle del Cauca. We have Cauca, Valle del Cauca, Caldas, Quindío, and Risaralda. Here is focus the main sugarcane production for the sugar and ethanol. Let's not forget that still exists more than 250,000 hectares for the panela production. In these 240,000 hectares that we have for sugarcane and production for ethanol and sugar, we have 3 different mega environments that we have. The semi-dry, humid, and Piedemonte. The semi-dry environment receives rain throughout the year, more or less 1,200 milliliters. The humid, the rain rate is stronger, and the Piedemonte that is in a lower proportion. For about 4 years, the bioenergy factory plant, there is a production of sugar in order to produce ethanol, and we are talking about 20,000 hectares that are related to bioenergy. The sugarcane, in fact, is a sustainable crop. It is able to fix 80 tons of CO2 per hectare. And in fact, all of the whole process of transformation of the sugarcane can recognize within the circular economy. So maybe the products that you recognize from the sugarcane as such, sugar, ethanol, and energy. But in that process of production of sugar, we can see that honeys that go to the distillery and produce the ethanol, but the remaining after producing, it's called vinasse, and it's used then as compost. The waste, it is utilized for the cogeneration plants, but also there is an alternative use, which is for paper production, and also the cogeneration plants are the utilization of steam for the process that can be applicable within the manufacturing process. So within the sugarcane, nothing is waste, and in terms of the circular economy, it's applicable in a good way. In terms of the sugarcane harvesting is very special because the conditions that we have in the country for this production are good conditions. Colombia for many years has led the worldwide production per unit per area. In our country, for example, in 1 hectare, we can produce more sugar or sugarcane than any other country in the world. That is different. That it doesn't mean that we are the biggest producer because only we have 240,000 hectares, but Brazil, with 50% productivity, has 10 million hectares. So per area, okay, they can produce more sugar. When we compare Colombia with the other countries that produce sugar, regardless of if the sugar is coming from sugar or beet, Colombia is the first country worldwide. So the question that comes out is, what is the reason? What's the key? So there are a series of factors. I want to focus on a couple of those that we can say when we refer to the challenge that we have, that we can focus from the genetics. So in order to explain you a little bit what are those factors, the ones that I like to talk about, one has to do with the environment. In order to tell you about this idea, let me tell you that the sugar cane, the source is from the Asia, South Asia, New Guinea, China. But if you locate in this map, you can see we are located right here in this region of the country. If you see the latitude, I mean, if you make a horizontal line, the latitude is similar to the one, is the one that you are in the center of the source. So in Valle del Cauca and in Colombia, in the neighboring areas, we have climatic conditions similar from the origin of the sugar cane. And that is favorable factors in order that the sugar cane like to grow. That is one of the reasons coming from an improvement or genetic improvement program. In order to perform this genetic improvement in a similar area, we can obtain materials that are well adapted with high productivity levels for this area of the world. A little bit in order to digest this information to the country, I want to show you what is the composition. Since 1990, you can see that in that time, most of the varieties or variants that we have, that they were grown from any other improvement program. So there are varieties or variants that they were coming from Mexico, Coimbatore, from India, or the Java Island POG 2878 is very common in the different improvement programs around the world. And the variant was developed by the Mayagüez Sugar Cane Factory, which is called Mayagüez 74 to 75. That is not the current history. The current history is mastered by variant CC in Ceni, Caña, Colombia, CC. And this is an animation that shows what is the varietal change through the years. You can see how the variants have grown in the year 2011, 2014, and you can see with a 74% of the area. So the leadership to 1940 in 2011, it was a 42% of the area, and closing June 2024, where we collected data, based on the census, is 0543, is 62,000 hectares harvested of sugarcane. So not everything is something easy. So we want to talk about the challenges in order to deal in terms of we have to think about plant health. There are varieties for a country, so we are interested that our variants can be grown, but also we have seen that, let's say, there could be situations that cannot be positive when a variant is over-adopted in a similar area of the land. We can see the adoption of the variety 85-92 through the years in the 90s to 2025, and as I said before, the highest peak in 2010-2011 when they reached 75% of the area. The sugarcane in Colombia variants, they resist to the diseases. Important to mention that in these crops, fungicides are not utilized in order to treat diseases, but we do not select varieties resistant to that. We have a biological control. Yesterday we were talking about biological control, which is very effective, in which allows that it's not necessary to apply pest control or insecticides that is eating away the stem. There are some insecticides for secondary pests, but in a very low area. Having mentioned that, I want to tell you that the harm failure in coffee, you can see it was 0, but through the years that the variant was adopted, you can see that the harm trend, you can see 85, 92 is not in 2000 in hectares, but it was susceptible to the roya of the coffee. That is a topic that we needed to tackle from the genetic improvement. Another point that I'd like to discuss is about the effect of climate, which has an impact. We can see an animation how rain of the humid periods affects the sugar. So we can see through the line 2018, 2020. I selected both because you can see an example of what I wanted to show you. We can see what is the trend and behavior of sugar in this red line, or the percent of that, or sucrose, and we can see the precipitation in that line. So there is some inverse line. The more it rains, sucrose falls, and then when the rain falls, sucrose increases. So the rain doesn't impact. There are other variables related to climate change that affect. And here we can see other two variables, or another variable that is affecting the productivity in terms of accumulation of sucrose has to do with the mean temperature and median and minimum for the accumulation of sucrose. The nights are cold and the days are hot. Delta around 80 degrees is favorable for the plant of sucrose, but during the day is growing, so it can accumulate. You can see here those trends. The red trend, you can see the accumulation of sucrose. The blue line is the minimum temperature, the temperature of the night. You have seen that has increased above 18 degrees in the last few years, and the maximum temperature have increased a little bit, and this has helped the fluctuation. You can see within this range there is a down trend. You can see that the productivity in the last few years, there is a drop, and of course we have a lot of work to do. So how can we solve this issue? There is a series of action plans that we have to take on. We're talking about genetic. We need to talk about genetic improvement. This is the flow chart for the production of new sugarcane varieties. This is similar to the ones that is used in other improvement programs around the world. In order that you can take away this information, it's a long improvement plan to produce. It takes for about from 10 to 12 years. Our plan is one of the most efficient ones because in other countries it can take up to 15 years. Every year we start with elite parentals. We have a germplasm banking. The elite parentals is a subgroup of that big group, but the best parentals have been selected. There are crossings. They produce a progenies of clones that are assessed through the time. In sugarcane, we evaluate F1. So the first progenetics and different stages of selection, it makes that at the moment we're making process, we reduce the number of variants because they are selecting. This is a conventional process because most of the sugarcane is produced around the world. Brazil has 115,000 hectares of transgenic sugar, but we continue producing to a conventional way. But we are migrating in order to incorporate molecular tools in order to exploit a little bit that genetic variability that exists in the crops in order to improve those interest characteristics. When we started to try to exploit that genetic variability by using molecular tools, it already, I mean, this is a work that has been done through many years. We work with microsatellites and conventional markers. So these gave us some outcomes, but based on the new technologies, we decided to incorporate those technologies. And we started working with gene sequencing of new generation, but one of the issues that we run across is that we didn't have a sugarcane genome as a reference. So most of the sequences that we were producing, they were lost because they were in line with a phylogenetics of the sorghum, but we were wasting sequences because they were not linked to that genome. Efforts were gathered in order to improve the sugarcane genome. The first one was generated in 2018. In 2019, one complete genome of an relative ancestor is spontaneous and commercial variant. From this one, the complete one, and from this one was a monoploid, like a representation of the total genome. These genomes, they took time, so we did part of those initiatives, but we decided to start by generating our genome. We were lucky, more or less around 2016, 2017, back by they came out with a new technology, an important technology. So long sequences were produced with affordable prices, and of course we ended up building our monoploid genome, the variety 01-1940. We had found out 63,000 genes identified, but at the moment that we improve this genome, it will reduce to something similar that we found in spontaneous. We are talking about around 35,000 or 37,000 for sugarcane. And with that genome, we were aiming to exploit. So we produce population in order to characterize, and an example that I want to show you of the benefits that we have obtained from those exercises in terms of gene sequencing with those technologies. This is of genetic diversity, the same population, 220 individuals. It was characterized with markers or microsatellite markers that were produced for sugarcane, and this was characterized with GVS and RASIC studies. In fact, the characterization with markers that are amplified through PCR is less efficient, so we need more time, more work. So we have 5 markers, polyformic, that we always were utilizing, and they produce 70 SR alleles. Sugarcane is an organism that is octadecaploid. It may have among 8, 10, 14 copies of chromosome. It's not a traditional one as the one that we know. 5 markers or 3 markers. They produce 70 alleles, and here we have around 20,000 SNPs based on gene sequencing. You can see the difference between both endograms. You can see a group which is more compact. They are not separate in a very well way, but with the markers we were able to achieve a better idea of what was the genetic diversity in the genome of plants.
We have more than distances in this study that led us to conclude that the diversity that exists in sugarcane is more scarce than we were thinking, and that happens with the improved crops, but we have to have efforts to increase that diversity. That December presentation of the SNPs, which are a little bit more in detail, we can see that we not only did we tell apart the difference from, as you can see here, piscinatum, but we also were able to separate some hybrids produced in the improving programs, some hybrids in Colombia, Brazil, or even in some other improving centers, like for example in India, Australia, or Mauritius. We didn't stop there. We also created some association studies. So that means that the same population that we sequenced were characterized phenotypically. And we started associating, doing GWAS, the GWAS, the association studies, because we had a good number of markers. And eventually what we did was to leverage that out of the population that we have chosen, 220 individuals, we characterize them at a very good level with the aim to do all the possible association with those characteristics. And of course, it is important to mention part of this work was done in the ómicas project back in 2016 when I met Andrés Jaramillo, and we were able to incorporate part of the strategy of association in the ómicas project. Here we have some of the results of those association exercises. You can see there were many features that we characterized for these studies. We don't have all of them here, and the numbers that we see here are the set of markers which are associated with those interest features. Something that you can see in this graph is that there are some features for which we find a small number of markers, but whereas there are others with a bigger number of markers, and that evidently talks about the complexity that governs, that drives the characteristics. So disease resistance are features that are governed by few genes, oligogenic features, but productivity is more polygenic characteristic. There are more genes acting, and you're going to have a higher number of markers. So very quickly we realized that if we wanted to use markers to improve the genes, the classic strategy of trying to select the best individuals with the best markers was not going to work with that many markers. It is very difficult to tell apart the ones that did only a few. So statistically it is not feasible. It's when we started thinking about a different strategy. So we started we work with the concept of genomic selection, and that's the concept that we have been working on. Genomic selection, I am not expert in the topic, but you take into account all the molecular markers that are present in the genome, all the ones that have been extracted. Each marker has an apportation. It can be a positive or negative apportation with an interested feature, and at the end it's a sum of those apportations, plus, minus, plus, minus, and at the end you select the phenotypes which are more positive, but you use all the markers, not only a few of them. We started working with the features which is sucrose production, and we found that correlation, that prediction was very good, about 0.77 in those first studies. What does it mean? That when we predicted the sucrose based on the molecular base markers, and when we compared with the real measured sucrose, the correlation was 0.77, which is very good for an improvement program. we submitted we submitted that model of genome selection to test. We put it into test, and the state one, that is the first state of selection where you have all the varieties. We measured sucrose, and we compared that with what we were selecting for, or what was going to be selected with the genetic markers, and we saw that the correlation was 0.75, and that encouraged us to continue. You can see here there's correlation that what that it says is that the model is able to tell apart the families which have more sucrose and the ones that have fewer sucrose, less sucrose. So the farmer is going to be able to choose the ones that produce more sucrose and continue with the selection and production process. We wanted to implement this model in sugarcane, so we selected about 6,000 plants of those primary states, and we harvested them, and we put them in the freezer and sent it to a company that sequences them. That is state 1, 2025, because the improver plants them in 2024. The cycle is about 13 months. So the next year is where the improver is going to select the best ones. We already made a selection based on markers. We're going to see the selection by phenotype run by our improvers, and we're going to check if both selections match each other. To exploit the genetic diversity is a win-win game. We're doing this from a genomic standpoint, but that is not the only way we're doing it. Something that we were thinking, the improvers were thinking, is to test the idea that mixed varieties in field could be better in some cases compared to planting only one variety. This concept is not only applied in sugarcane, it has been applied to other crops. For example, we have wheat in this picture. In the north part of Europe, they faced a situation in which the more productive varieties were more prone to disease, and the more resistant variants were not that much productive. So they took in field and mixed and had a mixture of varieties in order to increase productivity and reduce damages. So following that concept, we started working with the mix of varieties. So that has been welcomed by people in the sector. This shows how mixture can be better. If we have an exercise, we have the one variety here. It was also planted with other varieties that give different features, like the ones that you can see here. And what you can see here, to understand this chart is a productivity chart. This is the sucrose productivity in the x-axis, and in the y-axis we have the cane production. And with these iso curves, what we have is the sucrose production per hectare. So this is a multiplication of sugar and hectares, and you can see that you can have similar tons, similar weights, but improving the amount of sugar of sucrose, which leads to more yield than planting only one variety. This strategy has been welcomed in the sector, and there's no coincidence that nowadays in that range of varieties we have a lot of hectares, over 1,000 hectares, but mixtures of varieties. That is very important because that is about 4% of the 240,000 hectares that we have in Colombia. Keep in mind that out of these improving varieties sometimes never come to the field, so 4,000 hectares is very important. To end, we were talking about this restoration project where we want to promote or foster the diversity in zones next to the plantations, because we have benefits that go beyond productivity but go to the ecosystemic services of the crops. We're working on restoration of biological corridors that foster the biodiversity of different species. This is a biological corridor. Here we have a sugarcane plantation. We are trying to diversify how this vegetation is. These biological corridors have many benefits which are associated with biodiversity. You can see them here, and these are important to these crops. By the protective, not only taking care of the environment with a sustainable focus, we know that this is good for everybody. With this, I end my presentation. Thank you very much. Thank you very much, Doctor. Do we have any questions? I liked your presentation a lot, and we have a question here. How do you deal with the problem of the polyploids when you want to determine the genetic distances? That is a problem which is very frequent in literature. Yeah, actually, it has been a problem we have faced, and we have had to work together, work collaboratively to understand this phenomenon. If you see the available software to calculate the genetic distances, they are designed for diploids. So at first we used the same software because that's what we had available, but very soon enough we saw that the results were imprecise. So what we are doing is assume the diploidity. We divide the results by 10, and depending on how the alleles are located, we can determine how we know that in this way we can have a more detailed information when we want to calculate those genetic distances. There is a package called GWAS-Poly, which was developed for potato, but it assumes tetraploidy, but it is better than the traditional software that assumes only haploidy. So we haven't solved this problem. We are dealing with it, and we have been able to find some improvements in the process. I want to ask this. Sugarcane, with the variety that we have in the extensions of plantation that we have, you can think about that as a biomanufacturing plant. What is the role of this polyploidity? Because you have to direct the metabolism of the organism to the generation of a compound. Do you see feasible sugarcane as biomanufacturing facility? I think it is. This is a step 4. It's very efficient in photosynthesis, which renders the biomass that is produced by sugarcane is the highest we can find in a crop. So I think this is a potent manufacturing plant. What we have to do is to make it produce what we want it to produce, because we know that it has the capacity to do it. In Australia, they use a transgenic to produce bioplastics. What happened is that the transgenic never came to life because the amount of bioplastic produced was not enough. So the potential is there, but we don't know everything. We still have a lot to learn in sugarcane, and is how we manage for this machinery to work for the purpose that we really need. John, thank you very much for your presentation. Very interesting as usual. I think that in the same line of the two questions, I think that the challenge is that it is a powerful biomanufacturing facility. I think that the exercises of the development of varieties is linked to a very mechanical process still, in which if we can do every study and try to determine the functions of the genetic annotation that lead to a particular function, the genetic edition is impossible. So we have to keep working with a mechanical crossbreed. So that's the most important, the quid of the topic. To a certain point, we have to continue improving with the conventional method. I think it's necessary, and it can be necessary in many other crops, because if we separate the addition, the genetic addition from molecular markers, in the case of molecular markers, what we are aiming is try to be more efficient when we select. But in the genetic improvement has to continue. So the process what has to do is tell the improver, don't take this material because they don't have the potential to produce what we want them to produce. Take this one because they once do. And the traditional work continues is necessary, and there's no other way to do it. In the case of genetic addition, actually we are facing bigger problems to any other crop, but we have the same problem. Genetic addition is only good to the knock down or knock up of change some cotton for another one and reduce the production of a protein in particular, or abolish it. And we do it, we have done it in sugarcane. There are some edits that are at the research level, but we find an additional problem with polyploidity. We have to make sure that you reduce the number of copies that you want to reduce. You have to reduce it only to a certain point, and that requires more specific strategies. But we have edited in sugarcane. Maybe they are not commercial so far, but we hope to do it soon. Another difficulty that I don't know if you share is what we were talking about in the break, and is there. I thought that sugarcane was that there was a huge diversity in sugarcane, and because of that, it was a crop that allowed biodiversity of ecosystem for growth, but apparently they don't. So what I'm seeing now here is that it is a crop that is being restricted to some conditions, to phenotypic, very particular phenotypic conditions for it to be productive. I know that it's planted in other latitudes, but to the lack of biological diversity, of biological expression, that limits you in the possibility to crossbreed with parentals which have a different exploration of that genome, which can be expressed in different conditions of the environment. So the question is if you have forced that genetic expression in the other conditions, maybe in controlled chambers where you can take the plant, you make it suffer. I know that those plants are more productive under stress. Have you taken those plants to more stress in chambers? And let's say that as any crop, when you are improving, doing genetic improvement, you have to analyze. If you improve, you choose what you want, and you're leaving behind something that you like, and that is diversity. And sugarcane is no different from any other crops. The climate nowadays have put us in situations where that we can say that we are evaluating differential conditions. And back to, we had a lot of rain lately from 2021 to 2023, and we saw how the commercial crops were behaving and the changes in productivity. We have experimented with some in a semi-controlled level. For example, for hydric stress, we have some structures in Seneca which are on the roof, which allow us to control that. When floods, we also have some pools, and we grow them in imagine like growing rice, in order to know those phenotypes that can produce under those difficult conditions. And we are also in the process of getting a phytotron. Can you think about that? A phytotron is not the same to have it for beans as to sugarcane, in order to get better results. So the idea is to try to know better a little bit in those not so normal conditions. Any other questions? A round of applause to the presenter, please. Thank you very much for your attendance. Now, due to the time, we're going to be here at 2:30 to continue with the presentations. Thank you very much.
She has played a key role in the development of standardized analytical tools for the analysis of food composition. Dr. Preni, can you hear us?
Yes, I can hear you. Can you hear me?
Yes, and we see your presentation as well.
Okay, so I go now? Okay, great. It's so weird. I can't see anyone on the screen. So thank you for having me here today and for allowing me to participate virtually. And as the introduction mentioned, I'm going to be talking about the Periodic Table of Food Initiative, what that is, and how we are specifically focusing our technologies on the development of standardized analytical methods for characterizing food compositions. So seemingly every day we see articles like these in the media that are highlighting the grand challenges we are facing as a global community around climate change and malnutrition. At the center of it all is food and the systems that surround the production, processing, transport, and consumption of this food that we all interact with daily and rely upon to survive. However, as important as food is to all of us, we actually know very little about it at the scale required to impact change. We do know that food is a complex matrix that includes endogenous biomolecules like macro and micronutrients that we're used to seeing on nutrition labels, and also the thousands of secondary metabolites like flavonoids, terpenoids, alkaloids, and steroids, many of which are known to be bioactive. And also, food also includes exogenous biomolecules like pesticides, herbicides, preservatives, and additives, which can enter the food at many steps during the production and processing. Conventional food composition databases track only at most 150 of these biochemical components of our food. And so as an analytical scientist, I know that this does not really align with the capabilities of our current or modern technology. So why are we not doing better? So one significant analytical or inherent analytical challenge is the chemical diversity that's represented in food, meaning that effectively capturing this chemical space requires many different analytical platforms. Additionally, we've had a historical reliance on quantitative data for food composition analysis, which necessitates targeted methods which are inherently self-limiting. Another challenge is that while there are certified or standard methods for macro and micronutrients, again, that we're used to seeing on nutrition labels and in composition databases, there's pretty much a complete lack of standardization in non-targeted or even large panel targeted methods that are required to capture this diverse chemical space. Importantly, there is also not a standardized approach to metadata collection, which makes it challenging or essentially impossible to compare or combine data sets across databases. Finally, the generation of data at the scale required to address these global grand challenges is a huge task and one that no lab or institution can do alone. So to address these challenges, the mission of the Periodic Table of Food Initiative is to build a global ecosystem and provide the data, tools, and training to enable us to catalog the biomolecular composition of the world's food supply. This knowledge has the potential to empower stakeholders throughout the food system to make evidence-based decisions to better address the health of both people and the planet. So the analytical tools needed to accomplish this mission are many and can be broken up into three categories. So the first category, or the top of our pyramid here, really encompasses those traditional nutrient components that are already being measured. Here we are adapting existing methods, creating standardization if needed, and really focusing on capacity building to enable deployment by our global partners. The next component, or the middle part of this triangle, or pyramid, is focused on all those compounds that we know are in food. There's lots of research out there, but these are not currently being measured in any sort of standardized or comparable way. And just going to this next step is going to expand the scope of food composition and what we know about our food from those 150 compounds to thousands and thousands of biomolecules. And then there's the bottom. There's these thousands or even tens of thousands of biomolecules that have yet to be characterized. And these have been referred to as the nutritional dark matter. So the rest of my presentation is going to focus on the tools that the PTFI has developed related specifically to the characterization of small molecule composition in food. So the first thing I'm going to do is start by walking through the steps a typical lab that's not currently within the PTFI ecosystem would go through to perform this type of analysis. So first, they would start by procuring the food, maybe from their backyard, a grocery store, or a farm. Next, they would prepare the samples for analysis. And this is typically done by extracting a select class or classes of molecules with structural similarities. Then they would collect the data. This is most commonly done using liquid chromatography coupled with mass spectrometry. And then finally, they would perform data analysis, which is where we can translate the analytical data, which is really just a bunch of peaks and numbers, to biologically interpretable information. This step is informatics driven and usually involves searching libraries of spectra and or chemical structures. This general workflow is pretty straightforward and used by lots of labs all around the world for analysis of food and all sorts of biological samples. But my purpose for walking you through this, this workflow is to illustrate that at every step, we are making decisions, critical decisions, that are introducing bias in our analysis. And they are ultimately impacting the results that are going to be important or reported. So to illustrate this impact in the analysis of food, we did a small landscaping experiment where 3 labs were sent identical samples and asked to analyze them using their in-house workflow. So here I'm highlighting the results that we received for the analysis of tomatoes. I'm showing you here some correlation plots, which illustrate the reproducible analysis of duplicate tomato samples analyzed within each lab. So here we have replicate 1 on the y-axis or the x-axis, replicate 2 on the y-axis within labs A, B, and C. So we can see that each lab is doing a really good job of detecting compounds in the tomato and doing this in a reproducible way. Importantly, we can also see that each lab is clearly detecting more than 150 compounds. However, when we start to look at the data reported across labs, we actually observe very little overlap in the named biomolecules reported in tomato. In fact, only 2 compounds were reported commonly across the 3 labs. And this might seem a little discouraging, but really was not surprising at all to those of us working with this type of data. And it's important, right, to say that these results don't imply that one lab is better than the other. Rather, it's an illustration that each lab is performing the analysis in a different way, which ultimately inhibits our ability to compare or combine data across these laboratories or experiments, even though these were the exact same samples. And this fact significantly limits the value of this type of data to contribute to the building a larger knowledge base of what is in our food. So to address this, the PTFI approach is focused on standardization and global coordination. This standardization approach includes an emphasis on the food procurement, including, importantly, the collection of key metadata, sample preparation and data acquisition protocols, the development of novel reagents that enable calibration and alignment of data across laboratories, and I'm going to talk about that more on the next few slides, the generation of a reference library based on the analysis of authentic standards, and the development of a standardized data analysis pipeline for processing of raw data. Okay, so now here's what the PTFI approach looks like in action. I'm going to focus this example on the analysis of gala apples. So following procurement, so in this example, the apples were procured by one laboratory. The apples were washed, the core was removed, and they were then frozen, freeze-dried, and homogenized into a fine powder. These samples were sent to 4 individual laboratories for analysis, and then each laboratory followed a standardized protocol for extraction and data acquisition. So the extraction step included the addition of the PTFI internal standard reagent. The labs then used standard liquid chromatography methods for the separation of the molecules, and then each of them used a different type of high-resolution mass spectrometer. The data was then processed to detect biomolecules from each analysis. And in this example that I'm going to show you, we are not looking at named biomolecules, but instead all of the signals that were detected in each lab. So here we're seeing correlation plots again for the duplicate analysis of the same exact apple sample within each lab. And so again, we have replicate 1 on the x-axis, replicate 2 on the y-axis, and we have now we have 4 labs, A, B, C, and D, and we can see within each lab, they're doing pretty good at reproducibly detecting the compounds in the apples, and also, again, detecting more than 150 compounds. So also recall that I said previously that all the labs use identical liquid chromatography conditions and separation. So this means that they use the same column, they use the same solvents, the same gradient and composition, and the same flow rate. However, even with this level of standardization, which is rare, there are still inherent differences in the analytical system that result in chromatographic variation across the lab. And so what we're seeing here is a plot of retention time difference. So the y-axis here is retention time difference relative to lab A. And the x-axis is the retention time of the method, of the chromatographic separation. And what this plot tells us is that even with really good protocol standardization, we're seeing up to a 40-second variation in retention time for compounds which were detected across labs, which is pretty significant. And this in and of itself would not allow us to align data across laboratories. However, I also mentioned that with the PTFI approach, we include the addition of a standardized internal standard reagent, and this is added to every single sample during that extraction step. This reagent contains 34 compounds that span the chromatographic separation that I've shown here. So here, each of these peaks represents a different internal standard compound. This is the retention time of the method. I apologize, now we're in minutes, and before we were in seconds. And these compounds are all readily ionizable and detectable using electrospray ionization and LCMS conditions. And they are non-endogenous to food. So they shouldn't be things that we would see in the food anyway. So the use of these internal standards and the fact that they're in every single sample that was analyzed allows us to align retention times across data sets by calculating retention index. And so after this alignment, we now see our plot of retention index difference across the whole range of retention index indices of our method. And we can see now that after this alignment, our variation is reduced to less than 4 seconds. And this is actually phenomenal. I also mentioned that each laboratory used a different type of high resolution mass spectrometer. And each of these have their own instrument-specific settings that cannot be easily standardized. And one outcome of this variation is the total number of features detected by each lab for the same apple sample. I want to note that the term feature is not the same as an individual biomolecule. We often observe more than one feature for each compound. But the specific features that we observe for a compound are definitely variable across different instrument platforms. So here we can start to evaluate the overlap of features detected across labs relative to the total number of features detected in each lab. So what I'm showing you here is these results relative to Lab B. So Lab B detected 5,616 different features, and of those, 57% overlapped with other data from the other laboratories. This percent overlap is going to be variable as we consider the results relative to each lab. And so we can see here's the results for Lab C, Lab A, and Lab D. And of course, it's smallest, the percent overlap is smallest for Lab D because they detected by far the greatest number of features from the apple. This trend was observed across all the food types analyzed in this experiment. I've been showing you only apples, but we actually analyzed additional foods, including potatoes. Oh, this is an apple. I think that's wrong, sorry. I'm not sure what this one is. Beef and onion, the mystery food. So these results show us that the PTFI standardized protocols and reagents, using them, we can successfully generate data in labs across continents and using different instruments that we can align and compare. And I really can't emphasize enough, like that's a pretty big deal. However, we also highlight the fact, right, that there are some inherent differences in instrumentation that we really cannot standardize. And these are always going to result in lab-specific signals. For example, the ionization process itself, so this is the process where the neutral molecules from our food become charged species in the gas phase so that we can detect them with our mass spectrometer. This process itself, which is fundamental to this type of analysis, is going to vary based on the design of the source and the voltage settings, and it's going to result in in-source fragmentation or attic formation that are going to be different for each molecule across the different labs with different instruments. And this really highlights one of the fundamental challenges, not just within the analysis of food, but in all sort of non-targeted acquisitions. But the flip side of this is that there is huge power in consensus, meaning that while there will always be lab-specific signals, the standardization and coordinated approach of the PTFI will provide increased confidence that the biomolecules that are reproducibly detected across laboratories and foods of similar type are real and confident biomolecules. And so putting this into context of our original goals, we have this small percentage of known biomolecules in food. And so here we're specifically focusing on the small molecule composition of food and not all the molecules in food. So using our standardized and global consensus, standardized approach and global consensus with the PTFI method, we can expand the chemical space we are characterizing. And in the future, we hope to expand the space even more by adding additional analytical data like collisional cross-section and MSMS fragmentation, along with the use or the development of new informatics tools. And ultimately, this approach is going to help us to distinguish those real signals, what are the real biomolecules in our food that we can be very confident in, from this lab-specific noise. So I'm happy to stay around and answer any questions. I want to quickly give a shout out to my team at Colorado State University. Also the Verso Biosciences team, which are leading the development of these analytical tools for the PTFI, as well as our many partners in specifically that have contributed to the development of these non-targeted methods for small molecule characterization in food. And I thank you for your attention.
Thank you, Dr. Brain, for the presentation. I'll open the floor for questions. If you have any questions, you can ask them in Spanish and we will translate. So, si ustedes tienen preguntas, las podemos traducir. Las pueden preguntar en español y las traduciremos.
¿Listo? Mi pregunta está relacionada con el experimento con las manzanas. Entendí que seleccionaron ciertas manzanas, pero quería saber si también tuvieron en cuenta como el estadio de desarrollo de la manzana, si era si no estaban suficientemente maduras o de pronto ya estaban llegando a podrirse. Sí. Sí. Sobre el experimento de las manzanas, mi pregunta es acerca de, en el proceso de escoger las manzanas, ¿tuvieron en cuenta el estadio del desarrollo del fruto? De pronto si era más joven o si estaba en una maduración óptima, o por otro lado ya estaba más allá de su estado de maduración.
Okay, doctor, can you hear me?
Yeah.
Okay, the question is related to the experiment that you did with the apples. So the question is more about the process to choose these apples. So did you take into account or into consideration the stage of development of this fruit in terms of ripening? So maybe was it in the early stages, or was already mature, or was over mature, or over ripe? That is more or less related to the question.
Okay. The short answer is no, because the purpose of this experiment was to evaluate the development or the method, or validate the method. And so our concern was really on having a representative set of samples that were all the same that we could provide to the different laboratories. That said, that what you described there is one of the potential applications, one of the many, many potential applications of these methods. This could easily be used to design an experiment to look at how the composition of the apples changed throughout development, or based on where they're grown, or across varieties, or any number of different things that you could look at. Apologies for my dog friend in the background.
Question? Any other question in the room?
Hi, Jessica. This is Andres. How are you?
Good, thank you.
How are things going? So just a quick question, and this is related to the annotation of unknowns. And I think a big portion of the work that is ahead of us is really beyond getting the quantitative information, be it relative or absolute. There is a huge space for data science to contribute in annotation of compounds. In that sense, that's one issue I think is critical. The other question that is more associated with this particular phase of the program is how do we address I think there's still a huge space in terms of differences between the instruments. And some of them are giving us enough tools to reach normalization, but from the results we have so far, that normalization is not, it's really on the low end percent when you try to standardize the methods across the board. So how do you think that is going to move forward in terms of PTFI attempting to escalate, the protocols beyond what we have so far, in order to make this really a huge global effort to try to characterize foods across the board? I think that is essential, and it still worries me what the path forward is in terms of, you know, the huge amount of unknowns that we have in terms of metabolites or lipids. It's a smaller set. Proteins, there's still a lot of challenge there as well. But in terms of annotation, it's a big problem in terms of the differences that we still see in the standardization, even though it's a huge leap what you guys did in terms of getting the standardized protocol to improve the level of coincidence between instruments. I think there's still room to improve. Where do you think that room might be headed?
Yeah, so great questions and very spot on comments. First on the annotation, I didn't talk about that in this presentation. I was really focused on the standardization of just the data acquisition and showing that we could align data. But the other aspect of what the PTFI has been working on is the development of a common data analysis pipeline. If you work in this field, you know that there's like so many different data analysis pipelines that you can choose from. I don't think anyone, well, I'm sure people would say one is better than the other, but in reality, maybe for certain situations or for certain types of data, one is better than the other, but they all work well and they all have pros and cons. And so it's really more about picking one that is standardized so that when we're, again, processing our data, we're comparing apples to apples and not apples to oranges. And so in that process, if we can standardize the whole workflow, then the annotation, we can do better with the annotation. One of the things that we're doing is we have measured a large number of authentic standards using the same method. And so if we can align the data across labs, we can also align this with our standard, with the authentic standard information, and we can start to make assignment annotations. And so that's where we're at right now. But the other thing is that now that there's still a huge number of unknowns. So that's spot on, right? Even with all of this work, maybe we're annotating 5–10% of the signals that the consensus signals that we're detecting if we're lucky. So there's still a huge unknown space there. And that, again, is where we're going to need to start leveraging those informatic tools that are out there, perhaps developing some of our own. But one of the things that we will have in our favor is the fact that we've now also aligned retention time. And so that tells us a lot about the type of molecules that we're detecting, even if we don't know what they are. And then your other comment about sort of the standardization or the lack of standardization across instruments, honestly, I don't know that we're ever going to get there, right? And I believe that that's one of the sort of inherent challenges of just doing this type of work is these lab-specific signals. And some of it is the instruments, but also some of it is just the environment of the lab. Our instruments are so sensitive, they can pick up things in the air, they can pick up like the perfume that we're wearing, maybe that gets pulled in and becomes a contaminant signal. The solvents that we're getting, if they're coming from different sources, they can introduce background contamination. So there's lots of sources of this lab-specific variation, some of which is instrument-specific, some of which is just environment-specific. And I think that there's a growing recognition that our estimations of how many molecules are out there or what this sort of dark matter space looks like might be a little bit overestimated because of all of this lab-specific signal. And so I actually think that we are going to get a long way, even if we accept the fact that there's this lab-specific signal and we focus on what we can see in consensus, that's going to move us a long way forward. Because in the end, I mean, especially between instruments of different vendors, there's really no way to standardize across them. Like even the same voltage setting on two different instruments means something different. And so that's my answer to that.
I agree. And you know, I'd like to publicly, you know, acknowledge the fact that you guys have made huge strides to improve on that, and kudos to you and your team for that. one final comment that we've been trying to address on our end, and it's the fact that it's not only the differences in instruments that we're seeing, even in the protocol that in sample collection, for example. And this goes back to the question that was asked before, and it relates to the fact that a living tissue will continue, enzymatic activity will continue changing its composition even after you sample it. So if you take a sample from a forage or even an apple, you pick an apple, it really depends on how fast you're able to flash freeze that sample or take a snapshot of it in order for you to get a unique or a reproducible mass spectra. So we've had to deal with, for example, forages that we've been characterizing at the metabolomics level. And doing lyophilization is completely different from doing heat drying. And we're seeing alternate compositions due to active enzymatic activity. And the question is, there will be these differences not only from the protocol itself, but also from the phenotypic expression of these organisms. You can, depending on where you pick it, how your samples are composed, if you do biological sampling, technical sampling, whatever you do, there will be differences. How can we, in terms of the biology, look at solving the issue of understanding the reactions that can still go on after you sample? Because there's no ideal way of flash freezing a composition.
I agree. And all I can say to that is that this point in particular really speaks to the importance of metadata, which is one thing that it kind of is like this not very exciting aspect of what we do, but is actually a critical part of this project and it being successful, right? Because we're never going to be able to control these things that you're talking about. Like, we know that these things happen. We've been primarily focusing on sort of following a collection scheme that would mimic if we were going to purchase a food and then eat it, right? So we're not like going to the farm and flash freezing it the minute we pick it, but we could, and that is going to give us a slightly different answer. And some people will, if that is their experimental design. And so we have included the capacity to notate that in the metadata, right? How were these samples collected? If they were collected as part of an experiment where we're interested in the biology of the plant and we're flash freezing them in the field, then that is included in the metadata. If we got them from the grocery store and we brought them back and treated them as a food, then that is also noted in the metadata. And so when we're interpreting this data across foods that are in the database long term, we can access that metadata to make the appropriate comparison.
Yeah.
If that helps.
Again, thank you very much, Jessica, for being with us.
Yes, it was my pleasure.
Okay.
Thank you, Dr. Prani. Let's thank the speaker once again. Continuamos con la ponencia Exploring Cultural Heritage in Foods Through Scientific Evidence por el doctor Andrés Jaramillo Botero. En Colombia es el director fundador del Instituto de Ómicas y director del doctorado de la Pontificia Universidad Javeriana. En Estados Unidos es director de la Unidad de Nanotecnología y Ciencia Multiescala de Materiales
Scale Materials and Process, same center, and also leading researcher and lecturer in the division of chemical engineering in Cali. He has been invited to the JPL in NASA and in the lab of mechanical engineering in the robotic division in the AIST in Japan. Good afternoon. I see people yawning after lunch. Okay. Today's talk is about the topic we have been talking about in the morning, and especially about what Jessica was talking about, Jessica Prenny. It has to do on how we can characterize foods, and in our case, we're talking about its composition, its molecular composition, to contrast them with the cultural heritage on food. I'm going to show you a couple of works. The first one is how we attempt to define the term food biodiversity, and our focus right now is nutritional composition. How we do it? In our institute, what we try to do is try to characterize the living thing in all its omic scales, which is explain how the organism is born, grows, and regulates in a particular environment. So probably you know genomics, you know proteomics, metabolomics, and the study of each one of these group of molecules is what we call omic sciences. In our institute, we are focused on using these tools of the science to understand how materials can grow from the bottom up. And the second one is how to achieve sustainability, environmental sustainability and productive sustainability. The third, the human health, diagnosis and treatment from the molecular basis, and at last the one which is nutrition and the relationship between health and what we eat. A particular topic here, a particular item, and this is a question for the attendants. What harms us as living things? What are the variables that attempt against our composition and structure? Do you know? Who can tell me? Any ideas? There is a hammer. Radiation, that's one. Especially high frequency, the one has enough energy to dissociate molecular bonds. UV is one of them. Gamma, X, all high frequency radiation. Yeah, that's cool. That's true, but it has to do with what? With the environment around us and the environment that feeds us. The ROS are species that, due to what we consume as food, create radical and they decompose us. It's only 3 of them. Which one is the third one? Pathogens. Okay? So be aware of little bugs. We're going to focus on the last one. I painted a chontaduro. This looks like a chontaduro. In several components, fatty acids, lipids, proteins, and metabolites, which is in colors. I'm going to state here a challenge for the ones that work in data science. Here we have a problem to which we face, and that's what we were talking about with Jessica earlier. We have a periodic table. 55 of them are natural, and you can find them on Earth. Out of those 55, only 22 conform a living thing. I have that vocabulary. I have this alphabet. It's very easy in biology if I start from genetics to understand what is a sequence or an assembly instruction. I have the nucleotides, only 4 letters. If you are DNA or RNA, it can be 5 letters. The question is, what starts happening after I climb up my scale? This is my base scale, but I do transcription, DNA transcription or messenger RNA. If I transcribe that to assembling matter to assemble proteins, I get to the cytoplasm and I find another language, which is synthetic, which is the amino acid language. I have 4 to the third, so I have 64 codons. A codon is just a sequence of 3 nucleotides or a code of a sequence of 3 letters. And I have a very defined, very well defined alphabet. I know what amino acids are. I know the synthesized proteins, but now we have the combinatorial explosion. And what it means is that if I'm going to check the set of proteins or metabolites that are derived from here, now the universe explodes in an illogical, difficult to grasp way. Everything that we have heard about these metabolites that we don't know is that explosion of from that chemistry in composition as in structure. Proteins are typically high, high molecular weight sequences of amino acids. Metabolites are low weight. We have a set of fatty acids, a set of lipids, which are formed by those fatty acids that add up to the total of molecules that we have to distinguish to be able to know the particular state of an organism. So the state, the health depends on all these molecules. So when you go to the doctor and you get a diagnosis, they tell you with a very reduced set of these molecules. Some of them proteins, some of them metabolites, but it's a subset that is not representative of the whole. The rest is magic or black magic maybe. With a vademecum, the story, the history that tells me if you have these conditions, probably the diagnosis is this or that. Okay, when are we going to achieve health if we don't understand this map? Not only statically but dynamically. You are constantly in contact with that environment. How can we follow up this? A mathematical exercise which I was telling you to another colleagues, if I take those 22 atoms and I form molecules, how many molecules could I form out of these 22 atoms? That is a very simple mathematical problem. If I was going to do only two atoms, molecules, it's 2 to the 22nd power, to the power of 22. If I'm going to do it with 10 molecules, 10 atom molecules, I would like to use 1.34 times 10 to the third to the power of 23. If I reduce that considering the valence of the atoms, the stability of atoms, and the available set of bonds, I reduce in that in 2 orders of magnitude. That is a huge space. So here is when we, with the same effort that they were describing in the former talk, to start talking about other living things which are foods which come from plants or animals with a composition, inherited composition, right? And now that we are in the state of the art, the periodic table of food initiative, we're one of the 9 centers of excellence in the world. We are handling the Latin American component, and I explained with this flow diagram. We first select the food, we do the procurement, or we go to the market and buy it. We go to the lab, we have to prepare the sample, and after that, for example, with fruits, we have to make 2 efforts. We analyze it and characterize it not only in the pulp but also the seed and the skin of the fruit. There are 3 different issues. What we have to do is decompose or break down that food in the thousands of molecules that I just mentioned to understand what it has, what is nutritive, what is its value, and what other molecular compounds could benefit us, or maybe that don't benefit us. We're not ever going to know that if we don't relate our genetics with the things that we are eating. Food is one of the things that harms us. What we do in the lab, we break down all the matter in the mass spectra, and those spectra are called the petrified effort. to achieve to this composition. And out of this, in the IOMICS institution, when we break down all the compounds, we have to know how those compounds interact with each one of the tissues that you have in the body. Heart, lungs, all the vital organs, even the brain system, the nerve system. And that is a very difficult work. It's going to take years, of course. What is the outcome of this instrument with which we break down the molecules? It gives us this, a chromatogram. This is the easiest way to conceptualize this. This pointer is not working, but okay, let's try. If I put in a solution and a flow in the solution of molecules, some of them are going to be lighter and are going to be able to move in that flow faster than the others. So the chromatogram is a race. The smallest molecules are going to move in that matrix, and they're going to reach the goal at the faster than the others, quicker than the others. So I'm going to get a plot like this. This is the first group. This is the second group to arrive. And then we go to another part of the instrument, which takes those molecules that are characterized by their rotation times and ionizes them, fragments them. And what we do there is distinguish each one of those fragments. And these peaks that you see here, which are the specific mass peaks for each fragment. Now we have to understand what fragments are those. So if you take something as simple as a molecule of glucose, 180 Dalton molecules, you can divide it into different spectra with different intensities, and that is the mass spectrum. What the equipment changes molecule by molecule and gives us thousands of those mass spectrums. All right. So what we're trying to do now is build a library of mass spectra big enough to understand what those foods are. I'm going to continue. So we start analyzing some fruits, a set of fruits, exotic fruits. We chose all of them. Maybe you know all of them. And we are also characterizing beans or grains of the world. SIAT has the bank that has 37,000 accessions of grains. We narrowed it down to 46 accessions, and we analyze them with all these tools. Where are we? This morning, Mauricio was telling you about the number of edible species in Colombia. We have above 3,800 edible species in Colombia. 24% of them are native to our region and are exclusive to Colombia. And now we have another topic, which is the topic of access and benefit sharing. And this is how these molecules that belong to our region are protected by us and are protected to the welfare of the communities that have been harvesting them for years. So we got maracuyá, tomate de árbol, chirimoya, mamoncillo, curuba, luro, maracuya, zapote, pitaya, dragon fruit, and chontaduro. Borjo, and there are more. We have 18 in total. And we started with this characterization, not only from the high level. In the high level, what we found is simple things like morphology. Take a look at the distribution of temperatures of growth. It's about maybe a little bit constant. The pH of the soil, you need probably a somewhat constant pH. The distribution of size of fruit, it's more dispersed now. We don't have any conclusive information about composition. And now we go to what we want to talk about today, is to understand how people have inherited the knowledge on these fruits. Chontaduro. What do you know about chontaduro? I only hear laughter. I can't hear you. What else have you heard from chontaduro? I only aphrodisiac. I hear only aphrodisiac. What about a grass? Is it good? Is it bad? What is it good for? When we talk about the cultural inheritance, it's what we have heard from our grandmothers. They say drink a cup of water with lemon for your throat. We heard of that. Even so, the determination of consumption depends on that inheritance. So take a look at the association, the cultural association that we have so far, and that is an exercise what we did in the exercise with several of the members and an anthropologist, because we needed to connect with the interpretation the peoples have with these fruits. You can see borojo, aphrodisiac, but immune system boosts energy, helps with anemics. Guanabana prevents cancer, and we have some claims, some associations, very eccentric claims with these fruits. All this range of interpretations of cultural information, how we match it with the reality that we see in the composition. Today we cannot go through all the space. These are the proximal properties. And this is general composition of those foods. Hydrocarbons. It's about 16% proteins, 1%, fat 1%, powders or ashes is 1%, and the rest of it is moist. Fruits as such is not a very good source of protein as such. Maybe guama, it has a 2% and it's well above the other ones. Chontadora is well above the other ones in protein. The rest of it is hydrocarbons. Take a look at the fats of chontadora, 7.6%. The rest of it is 0.7. So the question you should be thinking about is if that fat is good or bad. You have to understand what is a good fat and a bad fat. So we start analyzing the metabolites, the set of metabolites, which are the metabolites. They are not only the low weight, molecular weight, that are derived from the reactions in the cell. And low weight is under 1,500 Daltons. We got the 18 fruits. We use the instrument samples of those fruits. We got thousands of metabolites. What we did is try to understand what metabolites we have based on the spectrum. You have to change that spectrum into a chemical structure. Once you have the structure, you have to determine what is the function of that metabolite. Many of them are in databases that say that metabolite does this or that. So we got the exercise of getting all these metabolites of all these fruits, and we had an analysis of main component. That is a statistical technique which is not supervised, where you let them group based on the metabolites of each one of the tissues on a minimal set of groups. And what we got that guanabana has a composition of metabolites which is very different to the others, just like a grass or blueberry. So the compositions are absolutely different. So if we take guanabana grass out of that, we have some subsets or subgroupings which are clearly defined. Here we have chontaduro. Look, that chontaduro is next to sapote. Do you know sapote? Have you eaten that? They have the same color. They have the same similar textures. The flavors are completely different. Next, now we continue with an analysis of what is called a non-supervised analysis. I don't let so many groups to report. This is partial discord, PLS. This is a discriminatory analysis. It's a combination of analytical methods, and based on a score and a ranking, out of the content, we have all the fruits. I define which ones have a high content of that metabolite versus a a low concentration. See all the colors that we have. You cannot identify now. We zoom in on the first ones, and we start seeing stuff that has to do with what we were talking about earlier. Many of these metabolites that you can say with some very names, they are not annotated. We don't know them. We don't know what they are. Okay, maybe Jessica didn't mention that all of those unknown metabolites, that dark matter that we have in the foods, the main problem is that there could be a lot of toxins which we don't know, which are not good for health. Maybe from an apple, maybe a common food that we are ingesting all the time. We found the two metabolites that we could associate, so we know that, let's say, this particular tissue, Borojo has a lot of malato, also chontaduro, and we can associate that with that metabolite and see what function malato fulfills. It is associated with energy production, pH regulation, so you can start seeing what foods to consume to correct health states. Not necessarily it has to be a systemic correction. Maybe you cannot correct the complete problem, or if you have a pH problem in the stomach or in saliva, it probably won't correct the whole thing. Ácido salicílico. Chontaduro is the one that has more concentration of malato. And malate, salicylic acid is the one, Borojo is the one that has the higher concentration of this acid. And you can start seeing the functional association. We did the same with some of the other metabolites. There are some examples. I'm not going to go into detail. Let's talk about what we came. Chontaduro. Is it aphrodisiac or not? Personal opinions, I'm not going to mention anything. There is an evidence that comes from these results. There is indirect evidence. When I say indirect, it is that we are associating metabolites that have some indirect incidence on something that we call aphrodisiac. For example, vasodilation, libido. There are some associations on what being aphrodisiac in this set. There's nothing that creates a direct associations. There are a couple of associations with some molecules that are aphrodisiac. So, since we analyze the pulp and the skin and of seed, the higher concentration is where? Who eats the seed? No one eats the seed. Everybody consumes the pulp and they say that it's aphrodisiac. 2 minutes, okay. I'm going to try to do it. So don't eat the seed right now, because next we analyzed the content of amino acids in seeds. And we found something that you would expect to find, is that seeds concentrate a lot of the good things and the bad things also. There are a couple of fatty acids which are omega-3, omega-6, and omega-9. We have different chains of chemical chains. Omega is the last methyl that you can find in the fatty acid chain. The relationship that you would expect between omega-6 and omega-3 is omega-6 is pro-inflammatory, omega-3 is anti-inflammatory. You would expect the ratio between, you would expect 4 to 1. You can see that in seed you find 160 to 1, so don't eat the seed yet. There are other things that are contained in the seed. You can find heavy metals, for example. So what we are doing here is trying to work with all this set of molecules to determine its biological function, and of course we're going to determine some other functions for the available compounds here. Something important here is that this is metabolomics that gives us relative concentrations. the relative concentrations are very low. You saw the proximal table, the content of proximals is very low. The fatty acids content is relative to higher than the other fruits. Carbohydrates is tall, is high, but metabolites, we have not studied this composition very well. In beans, you can see the difference, the visual difference of these beans. We have a wide range. These are the geographical zones of data sessions that comes from the SEAT bank. And we analyze proximals first. There is no difference. There are three classes of beans in this case. The vulgaris class, common bean, lunatus, which is called the Lima class, acutifolius, which is in Spanish, tapering beans. Okay, then look that there's no difference here. There's no difference between the beans. They are to the eye, they are very different, but they are the same. They are an excellent source of amino acids, a very good source of protein. There is a superfood. Take a look at the composition of essential amino acids. It's about the same in concentration. If you can see the colors, which are the columns, it's about the same for each one of them. Where do we have the differences? If we do the same metabolite analysis, this is proteomics, we found three categories: the quality of the crop, nutritional quality, and response. 2 and 3, of course, of plant, how the plant uses those proteins to respond to stress, and how it uses the proteins to improve the quality of the crop. We don't find any difference when we go to main component difference. We see that there is a huge differentiation based on metabolites. If we go a little bit deeper in the distinction or the difference between the set of metabolites, we start seeing some bigger differences in proportion of some of those metabolites. Many of them are still metabolites that we don't know. But here is where we are doing that differentiation between the different classes of beans. The evident thing, if you see these mnemonics, is that there is a huge amount of mnemonics which we don't know. This presentation is still moving forward. Okay, it moved forward all the time. Okay. Okay, I'm going to stop here. I'm going to give you the final message, which is we are just beginning a revolution in food composition, because to what we want to go is, once we have everything annotated, all these sources of metabolites, proteins, fatty acids, you can be able to start definition personalized nutrition. How you can attain that personalized nutrition? You have to sequence the individual genomes and start relating the personalized genome of people with the food composition that we can derive from these sources. And I could choose the lipids from chontaduro, the proteins from beans, to start making up foods that are nutritional, low impact for the environment, and so on. That's the intention. That's what we are going to. The problem that we have now is the combinatory explosion of data. And I am honest about this. If you are not seeing how to learn statistical techniques, data analysis, do it, please. If you're not doing that, do it. And I'm not talking to the biologist or to the engineer. I'm talking to you all, because it's paramount. I'll stop here. I close by thanking my team. I'm the messenger only. This is my team. I just want to tell you, welcome to the ones who want to work with this. We need all the hands and the minds that we can possibly have. This is everybody's exercise. This is a universal challenge. Food security is not only about producing food to feed people. You have to make sure that this is a quality food and improves the health of people. I close. Thank you very much, and the ones who are in line. Thank you very much.
Excellent. Thank you, Dr. Jaramillo. So, any questions so far? Thank you for the presentation. It's a very interesting presentation. I have two questions. The first one, in that approximate how to control what it is considered in terms of noise by doing the analysis is of concerning. And the second one about the three species of beans, Acrotifulis vulgaris. To what extent have you had or have you contemplated the idea of handling the geographical variability where the exceptions are coming from? Of course, there must be a preference of some metabolites due to geographical location and soils. Yes, Christian, the two questions, I think they are good. Yeah, the first one, very quick. The threshold to sigmas, you can catch as needs it 2 blocks, but 2 sigmas is standard, and within the threshold, that metabolite is a real metabolite. Of course, if we had different tools or instruments where we get the same signal, it would be better, but these are expensive tools. Now what we're doing within the Excellence Center is start sharing information. That is something that we consider useful. Related to the second one, there is not a different solution about what Jessica mentioned. That is metadata, is to include metadata in the information that is recorded today. These instruments and tools are improving in terms of accuracy. We have a tools or instrument. It's a cutoff mass standard. Mass standard means that it's fragmentation of the molecules, and we can obtain more fragments, but we will have fine resolution. This instrument, 1 part per million, 1 particle per million, below 1,000 daltons. They are very accurate. The information, the phenotypic, or in terms of differential, the only way to solve it is that we have to do among excellent centers where we can see the differentials. Well, very interesting the information that you have shared, thank you. And I have a question. Those analyses that you have done in foods, in raw, but at the moment of cooking, for example, chontaduro must be cooked, and what about beans? So that information is changing at the moment that you have obtained in those analyses. That's my question. Yeah, please hold on, I solve to connect this in order to show you the outcome that you are asking for. If you are asking me about if you lose the aphrodisiac in the chontaduro by cooking it, the answer is yes. In chontaduro, we did the raw cook, the little about metabolite with indirect relationship with aphrodisiac. In the pulp, yes. Beans, we are with CIAT, they develop a protocol for cooking protocol for beans. So, believe it or not, it's a protocol that they have to develop a tool that they produce in order to sell. So they will be doing the exercise of cooking beans. We will send the samples. Metaboloid, we will do it. And at first it will change a little bit. For beans, the heritage and our mothers, they, for a while, they leave the beans in water from day to night. is to eliminate lectins and proteins to remove those layers of saccharides. So that is lost in the cooking process. So we can see that this is an exponentially more complex issue, and it's about recipes. So the way you cook or you prepare thermal chemical hammers, you are transforming those molecules of that food. I don't know if one day we can reach that point. I'm not sure, but the raw material, we need to know it. That is a good question. This is a question regarding the acquisition of information, data acquisition of metabolites. So I bring it up, some information that you mentioned about utilizing metabolites of this food source with these different sources have been, or have you considered in parallel, or have you explored the possible interaction of the proper work, adequate or non-adequate, of those metabolites? We are in chemistry, and you know that by the same exploitation or combinatory exploitation that I mentioned at the beginning, and the thermal hammers and the species that we use, the combinations are infinite. It hasn't been explored, the synergy. It's something that we haven't started to compose food from extracts, but it's something that is coming. All of us, we will end up eating through pills, or the habit, or it's a social activity by having lunch or dinner. But it is a great effort, something that we are doing with the Herm Pleasant banking with Peter Wurzel related to the beans that I mentioned. We did a proposal in order to improve the vegetable meat. Have you ever tasted the vegetable hamburger? Have you ever tried it? Oh, I didn't know a lot of vegetarians here. For lentils, in general, lentils is a grain anyway. That meat is suffering from two things. I mean, it tastes like chalk. It's a personal interpretation because it's a personal taste for me. But the reason is that it doesn't have the texture, nor the flavor, nor the taste. It has the protein content, but not in a similar way as meat. But there is lack of flavor and texture. And the only way to introduce is by bringing lipids that can solve both lipids and fatty acids. So this is an attempt of a proposal that we have in order to compose vegetable meat, taking into consideration not only that, but also the environmental impact where the source is coming from that compound. So we can start by mixing not only sustainable food, because we have the energy matrix to create a bean from the different variants, or to extract from chontaduro a lipid. The other question, excuse me, I want to take the opportunity, and by being aware about what we've seen in the morning, if we have the information about metabolites within the concept of the periodic table of food, what ideas can we build in order to say this food it's edible and with high nutritional content or quality? Yeah, that was my question, that they said that by heritage. Eventually we will have to reach a metrics that it will not be necessary to have toxins. It's a metrics of what is considered a good food in terms of quantitative, in terms of proximals, and also the molecules and the chemistry of the molecules. This is part of the optimization that is being developed. Yeah, of course, it will take years. This is not from one day to another, yeah. And we can close, or we still have time? At the back, please. Yeah, you have your hand raised for a while. Sorry. So from this effort of PTFI, Maria del Pilar, who is here, she's leading and effort good food fellows in terms of training students in order to learn these techniques in regard in applications for nutrition. You can raise your hand, Professor. They can know you. Hello. The first, thank you for the presentation. Very interesting. My question is about when you were talking about chromatography, liquid with mass. I don't know. I think about how to analyze a chontaduro through that technique, and above all, extraction and preparation, sample preparation. How is that part? Because we know that for liquid chromatography, the sample is everything. A good preparation of a sample, you can obtain a good analysis or fail. So I would like to know, how do you do that? Because you also obtain lipids, carbohydrates, fats. So for me, it's wonderful. Do you study here at the university? Okay. Anyway, you can visit us anytime if you want to see the process. But in short, for metabolites, the first thing what we do is the decantation of heavy molecules, long lipids, proteins. They are decanted, and then the remaining, it is extracted, and we have to pick out what are the solvents that are utilized for the chromatography column. We need to prepare the sample by utilizing oleophilization, flash freeze with liquid nitrogen in order to fix, or the snapshot, for example, that I mentioned before. It changes depending on the moment that I do it. That sample is flash freeze to the moment from the chromatographer. So the protocols are written, are implemented within the Excellence Center, within the PTFI program, are the ones that are applicable in the Fiji. And this is part of the process. So let's come to terms about some protocols that at least can give us the standardization of those procedures. They are written, validated. Welcome to the Institute if you would like to see how it's in practice. But you're right. Yeah, the handling of the sample is key, is fundamental. We want to thank Dr. Jaramillo. A round of applause. We continue with the next presentation. Amazonic PAM, Dr. Cardona, specialist in science and technology, food technology, master in technique and sciences, biodiversity in the Amazon, and teaching biodiverse processes for the environment. Her experience covers investigation processes, food chemistry and natural products, chromatographic techniques, and spectroscopic, and the development of natural ingredients. Good afternoon. It is a pleasure for me to be here in this auditorium with this interesting presentation that I expect can be complemented with this lipidomic from the Amazon, with lipidomic palm, which aims, according to what it says, lipidomic bioprospection of non-wood forest as a strategy of bioeconomics for the Amazon region. The question is, why too much Amazon region? Life has allowed me to be an adopted daughter of this region of our country. the Amazon rainforest that comprises 42% of our continental territory, that in fact keeps the 87% of natural coverage. Our country, in terms of agricultural fields, it's also forestry. It's important for us to understand our non-adorable forestry. All the biodiversity comes complemented with all of the people that live there. More than 1 million, very close to 1 million people that they know the area. Very exquisite knowledge. Populations, farmers, peasants, indigenous communities that from a scenery that is beautiful, that is breathtaking, that is lack of resources, that maybe we don't think about that in this comfortable auditorium. Why to go back and see the Amazon rainforest is to give them opportunities from the bioeconomics. That is why, as I mentioned, life allowed me to be adopted daughter of this region through the investigation research institute in the Amazon linked to the Ministry of Environment and Sustainable Development that aims the generation of knowledge, the development of technologies, and the transfer of this technology in order to give response to all of the social, economic, and environmental conditions that this region has. So this center aims to be an excellent center in research and to disclose that information about that biological social reality of the Amazon region in order to contribute to those sustainable developing process that 1 million people can live in a proper and adequate way with their biodiversity. And that has allowed us to be in the Amazon region, 42% that I mentioned before. You can see. So we only think about the Amazon, but there are more states or departments within Colombia. And the Institute has the advantage of having the headquarters here in Leticia, the capital of the Amazonas. And we also have subsidiaries in San Jose, Guaviare, Florencia, the new branch in Mitu, in Nerida. So we have is in terms of the herbal center, biological collection from the herbals, everything related with the fauna and microorganisms from the Amazon, and experimental station, which is called the Torno, located in Guaviare, in the small rural area in El Trueno, in Caño Trueno. And also it is constituted as our life lab, where we can do testings, non-timber products, something related to agroforestry, and of course, to mention about the rapporteurs they showed this morning about the loss of diversity, of monoculture. When we work on this retrospection and when we see all of this diversity, Dr. Mauricio Diaz-Granados this morning, he told us about the whole work, the Kew Gardens, what they did about aiming the useful flora. With that work precisely was achieved with them is to find that the Institute has the report of 2,286 useful species of plants. 96 of those are endemic, so useful not only for food but also for fibers, construction, for many other purposes and usage. And precisely to understand why they are called useful, we have dedicated to do the physicochemical analysis and biological analysis of these plants, and we have achieved the characterization so far 68 vegetable species, and to identify them as possible sources of natural ingredients. Later on, I will tell you why natural ingredients and no finished product. And from those 68 species, we may say that today we have 58 in low maturity level uh, in, in the lab. So how can we take advantage for with advanced maturity level? We are doing pilot testing and how to transfer to the community, and 6 in advanced technological maturity, which allow us how to make the transfer to organized communities that they want to take advantage and how to use those products and non-timber forest products. So in order to work with non-timber forest products as Colombian, as any Colombian citizen or any citizen, we may think that it's easy just to have access to the forest. Do you think that is so easy? What do you think about that? No. There is a legal framework because the diversity is part of our, as a state, as a homeland. From the Decree 2811 from 1974, they define what is the wild flora, that sustainable advantage of wild flora of the forest is a strategy of conservation and handling of the resources, because how I preserve. If I use properly, is a strategy in order that people can avoid or cut it out. For that reason, the economy is constituted as a strategy in order to tackle deforestation in the Amazon region. And then this decree 690, which identifies non-timber forest products as those goods of biological source different from timbers or from the fauna, and obtained in different varieties from the wild flora, including fungi, and they belong to the natural ecosystems. We don't know if we can eat or not to eat. We need to know that. Therefore, in order to be able to speak of those non-timber forest products, I wanted to take an example. The Mauritia flexuosa. Maybe you have had the opportunity to know it as moriche, aguaje, canangucha, buriti, or miriti, but it's the same Mauritia flexuosa. Were you familiar with it? I don't think so. Okay. When we see this palm, you can recognize and you can identify. Maybe have you bought some products, buriti maybe? Here, by presenting you the buriti or canangucha with the Mauritia flexuosa. This is a palm tree that can reach up to 35 meters high with diameter from 30 to 60 centimeters. And we can see subglobose and elliptic fruit up to 5 centimeters diameter. This is a dioic species, male and female, and also the health of the ecosystem depends on the balance of the population, where there should be 50 females per 50 males. This ecosystem is of interest because this is the humidity of the Amazon rainforest. This allows in order to have rain during dryness. So if we can preserve those humid areas according to the utilization, any canangucha palm tree can have from 4 to 7 bloomings per year. So that can reach up to produce 480 fruits. These fruits can be produced every 2 years per population level, because there are ecosystems where you can see a lot of palm trees, but in terms of population, okay, it can be very useful. So fruits, you can obtain fruits for months, and then we can see that they are coleopterous. When we take this canangucha fruit, we can see that the communities and part of the fauna, they can consume those fresh fruits. Also, they can make beverage, they make utilization of the seeds for aircraft, all those things. Also, the obtain of the pool for high level of carbohydrates. Also, for herbal teas, all those things, their fibers, you can use them for architecture. And besides, is the source to find the famous mojojoy that you can see when you go to Leticia and you do cheering, that you can consume that larva. But here, the understanding of the chemistry of the flexuosa. What is that? If you see the yellowish, what is the association with? What does it have? What do you think? We don't know? The carrot. What do they tell us about the carrot? Carrot for what? For the eyes. They tell us because it has carotenoids. Okay. Only the color is telling us good things or the goodness of the food. So when we want to understand the fruit, the first thing is to understand the macroscopic composition. So you say, but Juliana, but why? Because depending on this, I will be able to understand how much of this or each kilogram or ton. So how much can we take advantage? How much? So I here realize only 37% is only acid of lignin. It has a lot of metabolites that can be also aphrodisiac. We don't know exactly. That is another palm trip. But it has a hardness that is incredible. That for us, it also is a challenge to have a better understanding. What is the purpose of that seed? But the mesocarpio, as the pericarpio, this shell that you see there that looks like a snake. So you can realize that it has a color that is indicating that it has metabolites. Quite interesting. When I do the proximal analysis, the bromatological analysis that Dr. Jaramillo was mentioning about these fruits, sorry. So we can find that you can take a look. Okay, you can see here that the skin is fine and the mesocarpio is high with ethereal extract. That's why people like to do cosmetics with buritin. There is a high content of fats, but fats, when we see so close, it's a great amount of compounds together, triglycerides, diglycerides, carotenoids, sphingolipids. There is an amount of things to see inside. There is a universe of compounds that will tell us what this product is useful for, or this non-timber forest product. By this analysis, we can see the theanines, flavonoids, carotenoids, and therefore all of those are antioxidant compounds. That's why it's positive for capacity and antioxidant capacity analysis. When we do the quantification from chromatographic techniques of organic acids, sugars, carotenoids, doing a directed metaboloic is with succinic acid high in that, and also sucrose and total carotenoids. And this makes that this oil and the pulp of this fruit, it is not only good for or beneficial for consumption as well as for cosmetics, because these kind of compounds, along with other phytosterols, what allows is that it can be utilized in products after solar exposure or sun exposure, also sun protection. So then it starts making value or giving value to our forest. What is the extraction process? I will tell you as a story, very short, because that is part of that confidentiality agreement that I have with the associations that we work for or we work with. The Institute is working with 138 organizations, each of them to us in an area of the Amazon area. 4 of them, they work with this fruit, as mococa, agogonocacha, with Guaviare area. All of those dedicated precisely to preserve through the care of these humid areas. So in this way, we can see a cananguchal. I am a person that is short, so I see those 35-meter height palm tree. They are so high. We teach them how they can climb up. I mean, the indigenous people, they do it, but for the colonies of Colombia, they have to do another strategy. Through pulleys, they can take these branches. Okay, it's a small one, but normally the branches or the clusters, okay, they are higher than me, more or less 480 fruits, and in the field, they achieve what you see, which is called, that is taking out this fruit. In the process plan, what they do is the classification in order to understand what are those fruits that are ripe, which are not, which are in optimum condition, and then we do the hydration process. In the selection process, part of the work that we do as an institute is to achieve appropriation of the knowledge with those associations, as I mentioned before. We need to find some parameters in order that they can understand visually which are optimum for the process and which are not. And the question is why you say that is considered suitable or not. Because I'm looking for carotenoids, so you can take a look here that in fact by increasing the level of maturity, the carotenoid content increase substantially. So therefore it has a logic from the chemistry and biosynthesis for vegetable products. For us, it's quite important that these people can understand in a better way that they should and must do the processing in a ripe stage where carotenoids are mature and also etheria extract. It means the fats that I will extract from there. Additionally, we perform where we take the pulp, we drive, we press, and we obtain an oil of this color. So this is amazing, right? A huge amount of carotenoids in one wonderful and beautiful liquid. And when this chemical characterization is done from the canangucha oil, it has a high content of oleic acid. And if you see, there is a huge variability of carotenoids or total carotenoids. You can see that we can see a range within the 58 and 201 milligrams per 100 grams. Why would be that? So this is the answer that the whole afternoon you have been asking to the level of ripeness or maturity or genotype, environment, state or condition, and the type of extraction. And we can see the great challenges. Okay.
When you go in a more detailed lipotomic study with a technique like the one Dr. Jaramillo was describing, HPLC, coupled with an spectrometry high mass resolution, what we found is that most of the components of these fats is triglycerides, the glycerides and monoglycerides, and 1% has all the bioactive components, carotenoids, phenolipids, lysophosphatidylcholine, all these minor components which are the ones that give me the activity of this oil. What did we find? Most of these lipids have in their structure aldehyde chains. It's about 8 carbon chains, and above 90% of them contain 2 insaturations in their fatty acid chains. And that is very good for health, as Dr. Jaramillo was explaining earlier, our omega-6 oils. But makes it very liable to light and to oxygen. That shows us that, or it tells us how we should be storing this oil after its extraction. When we transfer this technology, it wouldn't be responsible not to think on the process, because as much as we think on the analysis, we understand that the process of extraction affects what we see in the chromatography. The extraction process is also going to affect what we are going to obtain and what we are going to give to our customers. So we also have to take that into account, take into account the extraction process. What we found is that when we use solvents as we would in the lab in a classical way, we extract more diglycerides and free fatty acids than when we do it in the mechanic extraction. So it is very important to take into account the way in which we extract the oils. Additionally, when we look at the change on the quality of the fats, which is the most important thing for my client, and we compare the two types of extraction, we can see that maybe there are no iodine index, but we find a huge change in the index of acidity, which we would expect after a lipidomic analysis. Also, we found that that relative composition, when we use mechanical method or approach, we go up to 73% of oleic acid, which is omega-6 and 9, and our linoleic acid is the most scarce, to 1.41%. So it has a very good content of fatty acids, as they were mentioning. There's diversity to stop buying olive oil. So we also find that this great oil has a high content of carotenes, alpha and gamma tecotrenol, which is vitamin E. It's very good for skin. Which are our challenges? What I was mentioning before. By the two methods of extraction it changes, what happens if I change the machine that I use to do the extraction? Is it the same with a screw than with a hydraulic press? No, it is not. And see that we have this new challenge here. Do they look the same in this picture? There are two different types in the same environment. Each one of them have different carotenoids and a different challenge in itself. Then, besides becoming a question about the content or the concentration of, it's an engineering challenge to be able to make sure that these communities obtain stable ingredients according to the requirements of the client, to create something so complex as their Buriti soaps in each one of their baths. Therefore, this only has sense is if as institute we only understand that the use of these products transcends, starting from this prospection, by prospection logic, it transcends what we can achieve with our communities, that they can understand how to leverage those products, how to obtain those natural ingredients. And for doing that, we need to transfer the technology so that they can do the primary transformation and secondary transformation. Because remember that we are talking about the Amazon in Colombia, where you don't have electricity or drinking water everywhere, but that allows to obtain natural ingredients which are stable if I do the transfer of knowledge and technology for other companies to go to the third transformation to a finished product that can then reach each one of your houses. I thank you. I finish with this. Thank you very much. I want to thank my team the intervention sustainability team and all the members of our bioeconomy line. Thank you very much. Thank you, Doctor, for your presentation. Do we have any questions here? Please. Hi. Thank you for your presentation. My question is about the last thing you mentioned about extraction if by solvents or mechanical. We could see that mechanical was better for triglycerides, and for using solvents you get another kind of compounds. My question is, how feasible is it? Would it be to use both techniques alongside each other? I'm going to give you the answer. First of all, it is better to have the triglycerides. They are more stable. Triglycerides have an OH group that to make them that reactive species. So when they have more triglycerides, my oil is more stable. Second, in the cosmetic industry and in the food industry, it is better. It is better paid everything that is obtained by green chemistry, and not only by the payment, but because it is better for our environment. So what we usually do is foster these mechanical extractions. Maybe a way to do it would be to combine both mechanical techniques, because one of them is more efficient than the other. All these types of work is what we do with our communities to achieve the most of it, because by solvents we could obtain more oil, but not with such a clean extraction as we would like. My question is, besides the technology transfer and knowledge, what other plans are you running to answer to those basic needs that the community needs to be able to achieve what we are stating here as it should be? Is your research being accompanied by something social? The intervention that we are doing in bioeconomy wouldn't be responsible if we don't think about this complete environment. So that's why we're required, as Dr. Jaramillo is an invitation for everyone to work alongside with us. Many institutions work alongside with us, international cooperation, the TIC ministry, Minagricultura, Minambiente, the different institutes of research, universities, because each one can give apportations for these communities. Some of them have some form, are very good at attending a business. There are some other indigenous communities which have different abilities and capabilities that maybe are not so good for in the business world, and this Western logic, maybe they are not so well adapted to that, and it's not easy for them. So that's why we need to accompany all of them, and not only giving them just a process and a plan, because we are giving also a problem if you don't do it in a holistic way. Juliana, thank you very much for your talk. I have two remarks and questions maybe. Yesterday, when we were in the other symposium, we were talking with Julian Colorado. He was presenting a proposal that we have already submitted to do phenotypification of moriche, understanding that this is a very, very complex thing to typify. We want to do it only with the canangucha, the expression of canangucha in situ. And I thought that maybe we can have a survey of free fatty acids in function of the ripening of the fruit. How much have you characterized canangucha in all its stages of production? And what is the quality index is free fatty acid, which is not good. This parameter, Dr. Jaramillo, is effectively. There is a publication maybe a few years ago in which we worked precisely during the growth and the development for acai, ceje, and moriche. This parameter, which could be a publication that we can share, and effectively I think that it is important to demonize that free fatty acid, because what we have noticed is that that effect of the type of extraction that is provoked by the type of extraction. And second, I wouldn't expect from biosynthesis those free fatty acids as a form of hydrolysis of those triglycerides that were formed. So I would think to complement that with secondary metabolites, maybe carotenoids. Yes, it was about the regional economy around oils, of good quality oils, and that implies not having these free fatty acids. Our effort for the phenotypification in situ implies the use of sensors that can extract the metabolite directly from the fruit, from the carangucha, that has a hard skin. So we are in that experimentation, and would like to know if that is a good indicator of if there are other alternatives that we can use without harvesting. Yeah, we would have to see. We have to check, because there might be other metabolites that could be a good indicator to do it in a parallel way. Okay, we thank the doctor with a round of applause, and please let's take a break, a 15-minute break.
Hi, Justin. How are you? This is Juliana. Can you hear me?
Oh, I can, yes. I'm worried there's been confusion.
No, sorry. We have a delay in the program. Sorry for that. We have one more talk left, and then it continues with you. We'll continue with you.
Oh, I at this point, I have a full calendar today. I have another talk I'm giving in about 10 minutes at this point.
Okay, so are you able to start now, and we can move the talk that we're going to have now? Are you ready?
I could, yeah, if that works for you, but I do have to leave at the top of the hour pretty tightly.
Do you have the 30 minutes now for you to start your talk?
No, I could do 15, max 20 right now.
Okay, yes, that will work. So I will let them know, and we'll start with you, okay?
Okay, that works. Sorry for the confusions.
No, sorry, because we had the program, but we had some delay.
I'll understand how those work. We've got a big event next week, so
okay.
I understand.
Thank you, Justin. So we'll start briefly.
Okay, our next presentation is going to be called Continue to Measure Proteomic Approaches to Biodiversity and Health by Justin Sigel, Associate Professor at UC Davis, specializing in chemistry and biochemistry. His research focuses on protein engineering and computational biology, applying these fields to biomedicine and biotechnology. He combines computational design with experimental methods to create enzymes with novel functions. In addition, he directs the UC Davis Food and Health Innovation Institute, driving sustainable innovations in food systems. His work promotes advances in health, environment, and technology. Justin, can you hear us?
I can.
Okay. Yes, and we see your presentation, so it's all yours.
The abbreviated time today. Sounds I'll have to get going soon, so we'll jump right on into it. Thank you so much for inviting me and having me, having me give this talk. It looked like a very interesting symposium, and I wish I could have been there for the full time, but glad I could hop in at least. So before I dive into proteomic approaches to diversity and health and understand why do we care about the proteomics and understand the molecular composition of the proteins that we eat, I think it's good to start off just thinking about what do proteins do on a daily basis for us in society. So a couple images here to convey that. If you think about the biopharmaceutical sector, the vast majority of new drugs over the last 20 years have been proteins. They're new proteins that these proteins provide functions, whether it's enzymes or antibodies or various other binders, or the GLP-1 like large peptides that modulate satiety. They're all proteins, and each of these proteins provide a novel bioactivity for our bodies that compensate for something that we need or want to mitigate. We use proteins every single day in laundry, and laundry detergent is another example. You ever cleaned your clothes and wondered why when you scrub with dish detergent, the stains don't get out, but you throw it in the wash with the laundry detergent, it magically comes out? Because your laundry detergent is chock full of enzymes and other proteins that break apart those tough to break down, those tough stains that don't traditionally break down. And even the foods you eat, practically every bite of cheese or bread that you've had, probably throughout your life at this point, for many of you, I'm assuming an audience here, so unless you're all over 60, let's put it that way, you know, has been, uses proteins. And it's not that I'm saying you're eating the protein like in a cheese, but it's actually using like the rennets or the various amylases and carbohydrate active enzymes to modulate the starch structures of breads to make them last longer and reduce food waste and increase shelf life, or make it so we don't have to harvest components from animals. We can just produce them recombinantly and produce the cheeses that we usually love and make. So I just described a whole ranging from laundry detergent to medicines to bread and cheese, all different things that proteins provide these functionalities. So quick dogma, because I don't fully know where the audience is coming from, so I'll not spend much time on it. But the central dogma of proteins is there's a protein sequence to a structure to a function. And a DNA encodes a protein sequence, which you can imagine a string of pearls, each different pearl being an amino acid. And those amino acids, the order and which amino acids are there, dictate the structure that that string folds into. That structure that it folds into provides the three-dimensional molecular composition that dictates a function. And that function can be an enzyme for catalysis, it could be a structural protein for your cell, it could be a binder like an antibody for recognition, or it can simply be for storage, to store critical nutrients and essential factors that our body need for various bioactive peptides to be released at a later date in a very rapid way instead of having to make them from scratch. So proteins provide all sorts of different functionalities. The challenge with proteins is that the space is massive. The space covers, as I said, it's a string of pearls. Well, the average string is 400 different pearls on a string. And there's 20 different amino acids. So that's 20 times 20 times 20 times 20 times 20 times 20. And it goes on 400 times. That's 10 to the 400th if you're doing the math really quickly. It's actually much bigger than that, but we'll round out to 10 to the 400th. The number of stars in the observable universe is 10 to the 24th. So the number of proteins that are accessible to nature, each one of which provide a unique function, is beyond astronomical. We would, in the field, often be very excited if we can narrow it down to an astronomical number of sequences. Luckily, what's happened over the last 20 years is this enormous space that's been very hard to navigate has become accessible. And so through advances in compute, analytical technologies, and genetic technologies, we're actually able to see and work with and understand the molecular compositions of our protein today in ways that are unprecedented in history. So for example, I think you've heard of, correct me if I'm wrong, but I saw an earlier talk was from the PTFI talking about probably the aspirational foods that were characterized. Well, part of that was proteomics. And so while there were 500 distinct different sources of food that had been characterized, each one of those has proteins in it. And we would normally classify those foods based on their taxonomy or genetic relationship, but we actually go down and look at their protein relationship, their protein-based relationship. We see a lot of similarities that are really interesting. So what I'm showing you here is what's called a sequence similarity network. It's a clustering algorithm where every one of these dots represents a protein, and every one of the little lines connecting the dots represents that there's a relationship in sequence, structure, and likely function between those proteins. And for simplicity, we said of those 500 foods, this represents about 80% of all the proteins in those 500 foods, this figure. And we just color-coded plant and animal just at a high categorical level based proteins. And what you'd expect is you see, hopefully you can see my cursor, there's clusters of proteins that are only found in animals, and there's clusters of proteins that are only found in plants. But what's, I think, surprising, but because of the way we think about these things, that there's a lot of commonality between plants and animals. And you can actually see a lot of these clusters have occupy both plant and animal proteins. So for example, If we highlight even further and think through, okay, shrimp, chicken, and beef, and we zoom into a very specific cluster, here's an example of a cluster. While it is mostly in animal-based proteins, corn, oat, celery, all exist within this cluster of proteins. And so there is relationships that we can leverage and utilize and understand how those proteins are interacting and delivering not only the nutrients, but the bioactivities we need to our bodies through these relationships and new sources of those proteins. So what do we do with this? I'll give you an example real quickly, given the time change. So here's another way of looking at the same data. In this particular study that I'll go through real quickly, we looked at 4 different burgers. We looked at a beef burger, a pork burger, a soy-based or a pea-based burger, and a soy or a tofu-based burger. I'm here showing on the traditional nutrient labels, and each one says there's 20 grams of protein, but in reality, in the beef burger, it's about 20% actin, probably 10% myosin, 5 or 6% myoglobin. We can actually break down the exact molecular composition by abundances of each of the proteins within this beef, and each one has a unique, distinct set of proteins. Those are those little dots on the constellation I just showed. And so we can start understanding the molecular compositions of these proteins in the foods that we eat. So the question really isn't, how does beef interact with your body? The question is, how does each of these proteins, regardless if it's from beef or pea or soy, interact with your body? And so we went ahead and did that. Sorry about that. My mouse got a little excited there and tried to jump ahead. So we did a clinical study, just a pilot clinical study, where we did 16 subjects, about half men, half women, where they fasted the night before and had a consistent meal the day before. They came in, we put a catheter in their arm, and then we had them consume a standard meal. Every subject consumed each of these burgers, and we put about a week in between each one, and with a little bit of water. And then we drew their blood every 30 minutes to understand how those proteins are moving into their body and delivering those essential nutrients. So, as expected, what you're seeing here is an average of all the subjects and looking at the essential amino acids and the change of essential amino acids in the blood. With the majority of essential amino acids, we can't synthesize them. So the majority of them are coming from that external source. Pretty well established in literature, the animal-based proteins are more bioavailable than plant-based proteins, and that's what we see here. Consistently, the beef and the pork, on average, increase to a higher level, and there's more total essential amino acids. Even though the amount going in the burger was the same, the amount accessed by the person was about 50% greater for beef and pork than pea or soy, and pea and soy behaving similarly. The thing here that's really important that I think is always missed in these stories, that it's really important to call out, is that that's the average, but none of us are actually average. Each of us are unique flowers, right? And there's pretty big error bars here. And so if you break those out and you look at each individual person, well, an example subject 4, was really good at pork. And actually beef, pea, and soy, they were about the same on. They were pretty similar as far as what they could access from that meal. Whereas subject 6 actually didn't access anything that well. It is a relatively poor, slow digester. You can see they actually never even go down. It's a very slow increase, consistently going up and plateauing after several hours, not even several minutes where you'd expect, around 90 minutes. So every one of us is different. So the averages don't paint the full picture. So I wanted to point that out. So now coming back to the molecule, that's looking at bulk protein, but what about the molecules of protein? Well, because going back to it, we have the absolute molecular composition of each of those burgers, and we know all the amino acids from the essential amino acids, the spectra. You can think of the fingerprint going into the burger. We can start comparing that proteomic profile to what we see in the serum and compare that essential amino acid fingerprint over time and how it compares to the essential amino acids found within each of those proteins, within each of those protein sources, to look for correlations, with the hypothesis that if a protein is highly correlated in essential amino acids, the new essential amino acids showing up in the blood are highly correlated to a specific protein, the chances are it's coming from that protein. Clearly, there's going to be some noise in this, but generally speaking, it's consistent with what we'd expect. And so then you can start actually getting down to every single subject and every single protein within each subject, and how each person is interacting with that food source. And the first thing that's striking, which is interesting, striking that, again, if you kind of look, the red and the orange are the animal-based, and the blue and the green are the plant-based. The red and the orange, you can see with the higher density, generally most of those proteins people are able to access better. But there are several red and orange proteins that don't look at all like what's showing up in the blood, suggesting that there's specific proteins that are recalcitrant to digestion. Same thing with the pea. There's actually some pea proteins that are the best digesting proteins. They look exactly like what's showing up in the blood, even though there's a lot of other ones that don't look at all like it and are likely harder to digest, which is consistent with how we understand digestibility for proteins, generally speaking. So now with this knowledge, we can start thinking about, okay, it's not just we need to digest protein better, it's specific targeted molecules within that protein matrix that need to get digested better. Some are fine and some need to get improved. And so I'll give you an example of what you do with that information. And this is one where we started a company called Digestiva that actually has tools to manipulate the molecular composition of those proteins in real time within our stomachs to allow access for those hard to digest proteins. So we target those proteins. We actually took the same pea-based protein, and I'm showing you the same data here. This is the baseline on the left-hand side, the amino acids in the blood, and it's the repeated data on both sides. If you use a tool that was developed to release those hard to digest proteins, now that we knew that they we were able to do increase the absorbability and the nutrients in the blood by 50% for that person after their meal. And so you can see actually the bar jumps up 50%. So it's like eating, if they ate, it's basically, you turn that plant-based meal into the equivalent of an animal-based meal as far as health and nutrition goes, which has massive implications for global health, with the thought that if you need to increase the protein to feed the world, you can either increase the mass of the protein or you can increase the efficacy of the protein. And this is a tool now that we understand those molecular compositions, it's a tool to allow the increase in the efficacy of the protein. You can think of it like a car. You either have a bigger engine or you have a more efficient engine to go faster. Instead of making the bigger engine, we're going to make the more efficient engine. And I'll wrap up there. I know I went really fast and jumped over a lot of detail, but wanted to make sure I left a minute or two in case there was a question before I ended. And clearly I don't do this alone. It's with a large team. The picture doesn't even convey half the team. I think a lot of these people are gone, so I put all their names that were on this specific project as well here. So thank you. Good. Yes.
Take the microphone for 1 second. Have you been able to identify if this is more related to the microbiome or related to the actual physiological ability of a person to absorb nutrients?
Yeah, most protein absorption happens before you hit the large intestine. So the vast majority of it's going to be driven by the straight absorption. There are separate clinical studies that have shown that, like, through gastronasal extraction, that it is more digestible in the stomach itself, and you are releasing it in the stomach itself. Generally speaking, if the protein makes it to the microbiome, the microbes ferment those proteins, and they don't release those proteins, the amino acids, into the blood. They use the amino acids for themselves.
Got it.
It's called, it's often in the literature, it's called putrefaction of the gut.
Yeah.
And you can tell when someone has a putrefied gut.
So how were you able to improve the efficiency? I'm still curious about that.
Yeah, so that one, the Digestiva company, now that once we understood the proteins that were recalcitrant to digestion through natural, like you can imagine, our stomach, we have pepsin, and then in the small intestine we have trypsin and chymotrypsin. they're generalists. They're, you know, we were made so you can eat pretty much anything and you get something from it, right? We're not actually great at anything. And so, which would make sense from an evolutionary perspective. Like, we never know what you're going to eat every day, so you got to be good at whatever comes by. And so, with that framing, that company was able to say, okay, well, here's the proteins that aren't getting digested. Let's go discover enzymes that are really good at digesting those hard to digest proteins. And we'll specifically find enzymes that are activated only in the stomach. So we actually can take that enzyme, it's like a few milligrams of that powder, put it in with the food, and then when you eat the food, that enzyme turns on in the stomach and acts just like pepsin, trypsin, and chymotrypsin to degrade your foods, but it does it for the hard to digest proteins.
Wow, fantastic. So it creates a whole new field also in the design of new enzymes, I guess.
Oh, totally. Yeah, which is really where my core expertise comes from.
Okay, yeah, fantastic. Thank you very much again. Let me give the phone to, probably we have a couple of more questions, or just one more question?
All right, I can take one question,
take one
and
more?
I really do have to go.
Great.
Is there a question in the room?
Yeah, over there. You can formulate, go ahead.
Hi. First of all, thank you for the presentation. My question is about, should we expect any difference in the protein digestion depending on the way the protein was cooked?
Yes, absolutely. It's a really important question, and something that, you know, we, in this particular study for these 4 burgers, and these are actual pictures of the actual burgers that we serve to people. They were just grilled, so their internal temperature got to the consistent temperature with consistent thicknesses, etc. But I am confident that different processing methods will impact the bioavailability and the way that these different proteins interact with our body. So I'm confident it will. We don't have the data to understand how yet, but that will be very exciting data to get.
Okay, Justin, thank you very much. We appreciate your time. Sorry for the delay, so let's thank the speaker once again.
I'm glad it was able to work out. Bye, everyone. Thank you.
Thank you.
Thanks, Justin.
Nuestra siguiente ponencia es Arroz y estrés por aluminio, un enfoque transcriptómico y epigenómico por el doctor Mauricio Alberto Quimbaya, quien...
With experience in experimental biotechnology as computational biology. He is professor and researcher of Instituto y ómicas in Universidad Javeriana, Cali. He focuses his research in systemic strategies to study stresses in plants and the regulation of plants in superior eukaryotes. He has research oncogenes associated to cancer, and he's studying methods to study genomic stability. Thank you for the introduction. It's a pleasure to be here. Thank you. It has been a lot of work this couple of days, but it has been very nice. So congratulations beforehand. I'm going to tell you about one of our favorite foods. We always have rice in Colombian plates. So the importance of rice is the topic I'm going to talk about. And one of the stresses that impact drastically the harvesting of rice, not only in Colombia but worldwide. So alongside with corn and wheat, rice is one of the most consumed cereals in the world. About 900,000 or 1 million people consume directly rice or grow it. And in some countries like ours, or even in countries like underdeveloped countries, other undeveloped countries, it represents about 30% or 40% of the caloric ingestion. For example, in Colombia or in other countries. And specifically in Colombia, we grow 591,000 hectares of rice, producing 3.2 million of tons a year for internal consumption mainly. And as you can imagine, like with the rest of the organisms in this time of change and the sixth massive extinction, rice is facing some challenges in a growing population: high temperature, high radiation, acidic soils with a high concentration of aluminum with low phosphate, new pests, and loss of resistance to some pathogens. And it is estimated that by 2030 we should elevate the production in a 95%, sorry, in a 25%. And in that moment, we're about to produce national sufficiency in the production of rice. That means that it is going to be cheaper to import rice from super producing countries like the United States and India than producing it ourselves. So we have to aim for strategies to secure that security, specifically in rice. I'm going to talk about the stress of acidic soils and its relationship with rice and how it is struggling against this stress. If we can see here in the map, acidic soils are about 30% of harvesting lands, and that's what we plant rice in the world. This is the distribution of these plantations of rice. Those acidic soils have a pH lower to 5.5, and what is happening there is that aluminum is solubilized like a Al plus 3, which starts being abundant in that soil and it starts having direct effects on the plants. So in the first stance is the roots. It's the roots of the plants, the ones who suffer the aluminum effect. So they are shortened and they lose density, and it impacts the later development of the plant, and it's going to be related to the production of grain, how the grain is filled and how much grain is produced. Those morphologic problems are represented by the molecular actions of the aluminum on the cells. And there have been described some mechanisms like the alteration of balance of ions within the cell. There is going to be reactive oxygen species, destabilization of the chromosomes, and there is a diminishing of the efficiency for production of proteins, and there is a direct damage on DNA. There are some species of plants which are naturally tolerant to aluminum, rice among them, and starting with the study of these plants, we have learned about the mechanisms of toxification that have evolved in these plants in heavy metals, specifically about aluminum. There are two systems of detoxification. The first one is exclusion system, in which plants synthesize and liberate organic that can lay the aluminum before it's internalized by the cell of the roots. And there's another one, which is the internalization mechanism, in which aluminum gets in the plant, but there are some specific physiological routes that take that aluminum to some specific organelles, the vacuoles, where it is neutralized by the organic acid or any other type of molecular chelants. Understanding those mechanisms and understanding that rice is a good model to understand how those processes have evolved to toxification by aluminum, we state these questions. How can we identify mechanisms of tolerance to the toxicity that have evolved in rice? And we decided to do it through an evolutionary scope, comparing two commercially grown species like Oryza sativa, which is the rice that we use a lot in Colombia, with some wild species that exist in our territory in Latin America and in Colombia, that are not similar to the rice that we use, but in the process of adaptation, have been able to take on that toxicity condition. So if we compare those mechanisms, we can possibly understand which ones have made able to domesticate rice. And under that perspective, in each one of the species, we compared contrasting variants who can take that stress. Here, I'm showing here that specifically from Oryza sativa we have a variant called Azucena, which tolerates aluminum stress, and Villa, which is susceptible, and Silvestre, which is Glomepatula. We have some genotypes that are tolerant, like Genome 97, OG97, and some other are susceptible. And if we contrast how that differential responses, we're going to understand what are the molecular mechanisms that which are activated in those tolerant phenotypes that allow them to be to take the toxicity, and in the other ones, the susceptible ones, to see what are the mechanisms that are reduced or stop acting and behave as such as susceptible. And within the different omic scales and the flux of genetic information, because this can be bordered in a different scale where we have the DNA. DNA produces a messenger. That messenger finally is the one that takes the instructions to produce proteins, and those proteins are the ones that perform the function, the molecular functions. We can try to understand the focus of how those mechanisms were developed, but we decided to board on the stage of transcription, where what we want to understand the canonic mechanisms of transcription. That means the ones that depend on sequence, how an activator or a repressor identifies a specific sequence within the DNA and how it is translated into a response of different messages which are activated by stress. Canonic mechanisms that depend only on what is written in the sequence, but also not only did we want to understand the mechanisms that depend on the sequence, but other mechanisms that depend on the accessibility to genetic material. Not only can we regulate those mechanisms by what is written on the genome, but because what is written is accessible for those machineries which are there, which are the proteins, and that's what we call epigenetics or epigenomics. And there are different levels to understand those epigenomic levels. We focus on the methylation processes, which are the addition of methyl groups in some specific regions of the genome, which enhances the chromatin to have some more transcription, or on the other hand, it can have hypermethylite regions wither that process. So we try to focus from both points of view. So from how much is transcribed in the genes, by the specific relationship with the sequence. And we did some experiments of sequencing of the RNA and how methyl groups can restrict those transcription processes, and we did it through bisulfite sequencing. How we did it? We contrasted within two species, Sativa and Glomepatula, those genotypes which are tolerant and susceptible. We grown them in controlled environments with aluminum. We had some adaptation period, and then we changed it to the control media with those concentration, and they were evaluated that phenotypic response and the genetic response after 10 days of being submitted to these stressors. So we established some mechanisms for each one of the approaches, and we sequenced the RNA. We designed some scripts that allowed us to identify the quality of the sequence, align them with the genome, count the transcriptors, and finally evaluate with those countings how the aluminum changed the levels of expression. And with the bisulfite sequencing, we did the same quality controls, and we used other software that allowed us to, at a genome level, identify those regions too. And the CPG islands, where there is an abundance, and where there are some markers, epigenomic markers represented by those groups, methyl groups. What we could learn, we learned a lot of things. I'm going to focus specifically on what, for me, is the mechanisms. And first, for the transcriptional part, there were some differences in the expression. Some genes were overexpressed, other of them were subexpressed in each one of the genotypes. About 3 to 5% of the genes that were coded responded to stress, but the interesting thing is that independently of the genotype, before the stress by aluminum, we identified 28 genes that always, and that always were genes that always responded to stress, and that respond some central machinery to response to stress by aluminum. But we also found, for example, these which are the genomes or the varieties, the tolerant variants, some genes that are overexpressed in those tolerant variants and that represent mechanisms which are characteristic for those variants, and also the same with the susceptible ones. We found some genes which were subexpressed in those susceptible variants that can represent all of them, that machinery that are compromised and inhibited, and it's translated into those susceptibility phenotypes. What we found when we talk about these variants in Orysis sativa and Glumepatula, we found that some genes which are associated, as the ones I mentioned here, some genes which are associated, related to the process of internalization of aluminum. MRAT is one of them, which is a aluminum transporter within the cell, and there's LS1, which is implied in the transportation of aluminum to the vacuoles where it is chelated. And finally, the transcription factors, which are associated specifically to the induction of these two genes, are underexpressed. So, somewhat, we could conclude, starting from this observation, that those phenotypes of susceptibility could be associated to an inhibition in the internal way of toxification of aluminum. We also found that those genotypes, which are the tolerant genotypes, transporters of citrate, for example, which is an organic acid that chelates aluminum before it gets in the cell. And we find these two genes, OSTAR1 and OSTAR2, which are implying the formation of glucose and transporters of urapyrglucosa, which are like a barrier that blocks the entrance of aluminum to the cell. So we can conclude that tolerance phenotype could be associated to an efficient way to the exit way of aluminum. We found other aluminum mechanisms associated to some genes. We found
in the auxin production, and these are hormones that are implied in the whole cell, and they are inducted anti-stress response. Also, we can find genes that are involved in the response to reactive species of oxygen, which are induced to transport aluminum in such a way or in a significant way. Now we have also found out other carriers, such as cadmium carriers, that the stress, aluminum stress, that are present, by suggesting that they may have a relevant role in the toxification due to aluminum. We have found other genes, they have not been implied in this inclusion route or exclusion route in order to protect from aluminum, but they are implied in other kind of stress. We have found genes that they have been induced in that central mechanism associated with lack of nitrogen, with salts stress, and lack of sulfur, or by induction. So how is the relationship with the epigenomic? For those that you have seen cellular biology or genetics, okay, I can remind you that the promoters are the ones of receiving the transcription factors will induce the triggering of some established genes. What may happen here at the moment that aluminum is induced over those promoters, methyl groups are added. If the transcription is active, then the adding of those methyl, the transcriptional machinery will not go to the DNA, and there is a gene silencing. But it may occur on the other way around. There are some epigenetic marks in the gene, so there are gene silencing, and through aluminum stress it is removed, those methyl groups and the genes that were silent, and they can be triggered or activated in such a way that we should expect a correlation between those methylation and the expression of the genes in such a way genes that are associated in terms of tolerance of those varieties that are tolerant, we can find promoters without these marks of methylation that are correlated with transcriptional processes. The other way around, in the genotypes that are susceptible, we may find promoters, genes, highly methylated, correlated with the inhibition in terms of the genetic expression. Of course, in order to do that, the first thing we need to do is map out or lay out how those methyl groups are distributed in the rice. And also we disclosed, and as a result for the scientific community, the detailed map in these genotypes related to rice. And also we found other genes. Well, the pattern continues. In terms of tolerant variants of varieties, we have those hypomethyl promoters, and then with the N expression, then in susceptible variants, we have hypermethylated and correlated with a reduction in the expression. Those genes are implied in what? Sub12 is an analog gene. It doesn't exist canonic caspasas that are known in humans. So this is the holy grail to find it. And subtilin are proteins that they tend to be analogs related to the caspasas tend to behave in human. In degradation of several components, ionized, such as aluminum, we may find proteins that are perform sulfotransferasas that modify proteins associated with auxins and ethylens, and they activate these response ways, anti-stress, and of course, against aluminum, and we also the hydrogenase mitochondrial associated with the production of those reactant species. As a conclusion of these activities, the takeaway is that there are similar mechanisms of tolerance that have remained throughout the domestication and evolution process of rice, and there is a central mechanism, very punctual, that regulates the response to stress by aluminum, asynchronic. This is very punctual, a core of genes that are related to response to anti-stress. The phenotypes of susceptibility may be associated to an inhibition through the inner way of detoxification, and the tolerance may be associated to exclusion mechanisms. And not only exist these two mechanisms, but also there is a cross flow that they relate reactant species that are physiological processes that are connected with those phenotypes response. And finally, we can find evidence of regulation epigenetic associated with the response to stress by aluminum. This is a very dedicated work. The team members that I mentioned, it is worthwhile to thank them. Isabella, now she will start her doctoral degree in the United States. Ana Maria continues working in CIA. Sebastian started his master's degree in Mexico. And Jenny, right now, is postdoctoral on the PTF5. And based on the results, this is thanks to this dedication to the team members and the interest by generating and share knowledge and all the work done with the team members and the OMIC team. Thank you.
Muchas gracias, Dr. Quimbaya.
Thank you. Any questions in the audience? Hello. Thank you for the presentation. Very interesting topic. In fact, rice is one of the greatest consumption and energy and all of the nutrients related to rice. And one thing that is of concern and I'm curious about susceptibility that it has, and in terms of being vehicle of fortification in Colombia for being one of the foods in terms of consumption in our country in Colombia. I don't know at the end of the world what would be the recommendation if the government would like in order to be involved or establish that is necessary, okay, for the Colombian consumption. So, I mean, the question is if what would be the recommendation from the work that you have been doing with your team and the people in regard to rice as a possible vehicle in order to do biofortification? Yes, as you know, there has been the golden rice or dorado rice in terms of fortify with different vitamins or with different nutrients for the rice. So I think that before reaching the stage of biofortification, I think that we have to start by solving the issue that currently we have. It is the one about rice production, that we now have lack of rice, or we are getting short of rice, or start importing rice from the United States and from other countries. That is something that we need to start focusing on and giving a solution, and after that continue with the next step, that is could be biofortification. My speech was in how can we be tolerant or make rice tolerant, and in fact currently I don't know if in Colombia we are doing attempts and trials in order to improve rice consumption and turn it into a more nutritional vehicle or career. We know with the dorado rice, with the golden rice, one of the main or the points is to improve the rice is about the conceptualization, because at the end the improvement that would be, it would be a genetic improvement for rice. We know the issues of genetically modified organisms, GMO, and even if it's improved by these ways or fortified by these ways, how to convince the community, even if we're talking about GMO in terms of modification, demystify everything that has to do with that. So let's say that it's something that involves a whole community, not only scientists that they are trying to achieve in order to be more nutrient, but on the other side, what it has to do with the demystification, how to achieve the genetic modification. We are making progress, we are evolving. Genomic edition is something that we have been doing, but if there is a tool, new myths that try to see about how powerful that tool would be. But of course, in countries in Colombia that we consume a lot of rice, one of the options would be the modification in order to turn it into a more nutrient product. We want to thank you, Dr. Kimbaya.
Para darle cierre a nuestra...
To close our presentation, we have Plaza Colombia, Platform Monitoring and Tracking of the Food System by Dr. Eduardo Gonzalez, Doctor in Environmental Sciences, Master in Economy, and Economist. His work involves climatic changes in South America, Central Africa, and Asia, analysis and development of technological applications related to food systems, monitoring and tracking of environmental network in Colombo-Peruvian Amazonian rainforest. Among others, Doctor, can you hear us?
Voy a compartir pantalla. Si están viendo pantalla completa o la...
En este momento te estamos viendo.
Please let me know if you can see the presentation. We don't see the presentation yet, but please. Can you see, but you don't see it on presentation? want to thank you. I will not take longer. I know that you have an agenda, but the idea basically is to tell you a little bit about the exercise that we have been doing with several partners in the development of platforms in order to monitor the food system. My name is Carlos Gonzalez. I work for the SIAD Bio Alliance in terms of economic area, but also people in terms of consumer behavior, others in data science. Without further ado, let's start with has to do this topic that you have been discussing. I was listening to the presentations, very interesting topics. I don't understand too much about other topics, but I'm not an expert, but the point here is how this food is put on our table. So for us, there are different milestones that are key within the research process in order to understand the food system. And one of the milestones is the table where people have dinner, where people get together. And the question is how all of these food get to that point. And in fact, to understand the whole process that is behind the production and the distribution and all of the elements that are associated with this is kind of complex because of the interaction of multiple elements among them and the components. For example, we can see about the lentils. This is one of the most traditional Colombian food and dishes, but when we've been exploring a little bit each of these dishes in food, we realize that specifically lentils is not produced in Colombia. This is an imported product from Canada. So reflections like this on the table is quite important in order to be more aware and to understand the difficulties and the challenges to take this food system, agro-food system, to your tables. So this is a question that we wonder, and what is a food system? What are the food systems? And we had found that many of the answers have to do with the component in the middle about production, distribution, the supply chain. But as a system which has a different connotation and complexity, there are other elements, other areas that are affecting the supply chain, such as demographics, about migration, technological, environmental matters, climate, and also political matters. So that this production system for food that has been affected by all of the components above, it's having an impact, in sometimes hidden impacts of the food system, such as nutrition, waste handling, and management. In several times when we talk about food policies or interventions in the food system, normally, so the traditional is to start working to produce and produce, but that is not is the problem or the critical point of the whole system as such. So basically, this is an example why our food system is failing. And we can see that in terms, if we compare the percent with food safety, we're talking about 56%, more than half of the population in this area. They are expressing vulnerability or food safety. And the population with overweight, more than 50% of people, we can make the comparison Cali with the Pacific area, and basically about food waste. We're talking about huge amounts. There are a lot of discussions about the measurement, how to measure waste, and what is the focus point, if it's in the at the gate of the farmer or is in the distribution process. We think that this is wasted more at the gate farmer, of the farmer's gate. And also, it's important that the amount of food that is wasted, basically, we are also wasting nutrients, energy, all of the topics that you have mentioned, which is interesting, and it may mitigate everything related to food safety or food unsafety. So our food system is not sustainable. And we need to understand and we need to study a little more about that. About these discussions within the alliance and with other partners, they said they started to profile, to create, to map out all of the components, to do the mapping out of the key elements of the food system, the green part that you saw at the beginning, and also the factors related with measurement. And there was a study, a pilot that was done where data was collected. All of the available data. The first message of this exercise that in fact Colombia, different from other countries, has a huge amount of data, so they are broken down, but it is important in such a way to integrate it in order to have a very powerful narrative in regard to the food system. And the message here, when we have data, we can tell stories, but the lacking data is something that we cannot tell. So there are some topics related to food system in which there is a gap that maybe we can improve a little bit. So as a result of this exercise, it was also applied in Africa and Asia with very good results in order to understand the food system and also the building of that. Based on that gaps and the data collected that are spread all over in different places, we decided with different partners, and I want to thank to the Faculty of Economy, the Faculty of Nutrition, our partners in the Javeriana Universidad del Cali, with other partners, we created the first monitoring system for food system in Colombia. With all of the possible challenges, but also integrating different visions, data analysis, perspectives, and insights, we were able to put it in different layers of the layers in order to build to have good, consistent narratives and integral. This platform that you can visit through the QR code or by just typing it, there are several advantages. The first one is that it simplifies in terms of surveying when you're looking for data. If somebody would like to surf or browse about supply, you have to download teras of information and process that. So we are talking about for the decision makers, not only for the private and public sector, they don't have enough time or resources, etc. So what the platform does, this is escalated. They offer simple data and comprehensible and understandable, and something is integrate different data sources. So that is something that I will show you later on. We know that the agro-food, they have more than 200 KPIs. It's difficult to reproduce them, but it is key and fundamental also to generate some KPIs which may have an analysis and interpretive capacity in order for the decision making. We also have a powerful one that I will show you later on in order to identify in terms of stress points, situations of something that is blocked, in terms of strikes related to impacts on supplies or climate change. We have created so far 13 boards. We will release and update it more. These boards are related in the same structure of the food system, so you will see associated to food production, associated to consumption, associated to the impacts, and also boards associated to the drivers. So the platform as such is an exercise for visualization. What it is behind the platform, there are a lot of data that we are processing in order to be easier to browse. So one board that you will find are the determinants. You can see a study, the alliance with the Universidad de los Andes about the coherence of policy in food systems, if the policies are coherent internally or the policies within ministries and governmental agencies, do they have or they have a level of adjustment of that they are in line based on the guidelines in terms of food system. So you can browse, you can see information, and you can find an assessment of more than 30 policies and so on. In terms of food production, we have gathered all of the data. So there is a cross-checking with Finagro, and also we have defined boards of information. We can have visibility, what are the indicators or the key indicators in terms of performance, output, production, and in another board you can see how we cross-check the areas of the productive area with the assignment in terms of credit for per crop. If it's a farmer, high, medium, and low scale, you can find that in the platform. This is what I mentioned before. You can see the assignment of the credits, the type of farmers or producer, okay, per state, and also municipality. The idea is to go to the geographical scale the lowest possible in order that for the decision makers can be easier as well as for researchers. In terms of food distribution in the platform, you can have the possibility of selecting, in this case, for example, Cali, and according to the data, these are data that we have validated with other centers or supply centers, and in general, they have a good representation of the distribution trend of food, so we can understand the amount of food, the food origin, the sources that they reach a city. So this is something key in terms of understanding what kind of food are coming, about food sovereignty, the circuits, and all those things. So the platform, with a couple of clicks, you can browse and see data. For us, it's quite important to understand what is the length or the distance based on the first slide, how food is put on the table. The geographical distance in terms of route is something fundamental in order to understand what is the distance, what is the length, what are the products that are taken longer distance. For example, in Santiago de Cali, we had found supply distribution centers of 1,300 kilometers, and we have done computerized analysis and algorithms in order that this analysis immediately it can have a representation of mobility in the area. This analysis in terms of food mobility, this is classified per groups, per food groups, and you can be explored by subgroups, which is broken down, and then according to the convenience of the data that you're looking for. The platform also allows to make a comparison between capital cities, and you can see relevant information. For example, we have Barranquilla and Santiago de Cali. We can have an 11.3%. This 11.3% is basically the food that are getting to Barranquilla from municipalities that they belong or very near from their or the department. It means the neighboring cities or neighboring towns. For the case of Santiago de Cali, it has a 40.5%. So this is an estimate based on 2022. So if we would like to see about food sovereignty, short cycles, etc., and also the neighboring cities or towns in terms of lack of supply, I think that Santiago de Cali is located in a better place compared to Barranquilla. So also in terms of intervention in order to improve food safety and all those things. We also have layers where we can see the amount of cows based on KPIs. Number of cows per farm, the geographical distribution. This is powerful information because you can see this layer of information, the localization of the cattle raising and the farmers of the cattle raisers. It may generate more information for the people who are interested. We have published an article that has to do with emissions and the mobility in terms of how the cattle is transported. In terms of consumption, this is one of the KPIs that we feel proud of because it's powerful and useful. This was developed with the team members from the Javeriana University and with the Economy Lab and the faculty of is to analyze the minimum cost for a diet enriched in calories, a healthy diet, a nutritional diet. You can see this is based on the GABA's diet. This is a minimization model, and with our partners at Universidad Javeriana, we did the estimates according to the nutritional composition per area and per age. So the result is a cost in order to have access per day in average. In this case, for example, a person of my age, around 39 or 45 years, it would be 10,000 pesos. It's for a healthy diet. This is quite interesting because this information was taken in September 2022, where in terms of inflation, there was a process of increasing prices. Based on this analysis, with this R code can be reproducible through the time and also to have a monitoring on real time and to be compared to other regions. This is another extension of the analysis. Here, for example, you can see the distribution in the sales per expense per household, and the green line, the darker one, are the expensive or monthly expenses of a healthy diet. So in the seal number 7, it means that the household in Cali, they can take or they can pay or can be affordable a healthy diet. It means that more or less 30.2% of the house, they can have a healthy diet. When we did this analysis, we found out, for example, that for the case of survival diet, which means rice, only 5% of households in Cali may have access to a survival diet. This is disastrous, which means that there's a lot to do. We need to work on in terms of the gap of data, in terms of consumption, diet, at least to continue with the research. We have also collected data from the studies and research in terms of food consumption, diets, and waste. By understanding that the government is not measuring this matter because of the cost that it implies, within the partners, we have found out how they can take part with the data. We also, this research, WW, it was done, different surveys were performed in different areas based on production, procurement, storage, and all those days. You can browse, okay, you can see the results. You can also filter out the result, strata, gender, occupations. Also, you can see how many times per day you eat based on the filters. 42% they say they eat 3 times. There are people that they eat more times, 28%, etc., and so on. And the group of foods that may be included in your diet, also eggs is the leading one, with a 92%, then fruit and vegetables, and so on, down the list. But also, you can browse through the waste component, production, procurement, and storage in Colombia. We have the results of a second survey that is ongoing in order to upload it here. In terms of impacts on the food system, there is an impact. It's about health. This is something that it is important to establish research processes where we can connect cost of the food system, and one of these is related to health. We have data about weight, obesity, max index, so you can see the comparison in different regions in Colombia. And this is data about emissions per mobilization of food. So as you know that there are greenhouse emissions by production of food, for example, also there is a contribution in terms of mobilization. Given the fact that we have a transportation system that make use of biofuels, about the route, the distances, etc., here what we have applied is a little about mathematical equations, and we were able to understand that according to the parking lot of the number, access, and the volume, what are those foods at the moment of transport are generating emissions, and what are the big cities, or depending the size, of the food that they receive, Bogota as the capital city, where we can see that the plantain, the onion, and potato are the top three in terms with the highest contribution of emissions compared to Medellin, the second city of Colombia. We can see papa capita, rice, among them 27%. So the distance of mobilization of these two products to take them to Medellin. There are some environmental implications which are consistent and strong. There are a lot of things, of course, in the platform that you can find, more analysis that we are developing, seminars, meetings, etc. And this is something that we have been doing with a community in practice about food system. All of the people that are interested in joining to our meetings, you are openly to come. There are some logos that are missing, but anyway, we have, for example, an event in Cartagena, something that we developed, management system for food system in the Caribbean region, that we do believe this is one of the regions that is still lack of understanding the connection and the food system. Also, we have other tools that we have developed along with the Javeriana University, that this is a RNA package. It's called food price, and you can see the estimates and the calculation of cost per diet. So I did it fast because I know that you're short of time and you're tired, and there is a taste activity that you will have later on after this presentation. So I want to thank you for your time.
We thank you, Dr. Gonzalez, for your presentation. We open Q&A. Carlos and Jaramillo, thank you very much. Very interesting presentation on what you have developed in Plaza. My question is about the platform, of the capability of the developer to handle real-time information and georeferenced. You showed something about environmental impact of animal agriculture. We have a program of emissions in which we are aiming to georeference the emissions, the real-time emissions of different kinds of animals, bovines and also crops. The question is how open is the platform for us to automatically report, since COT system information in real time, and that it won't be, the best question would be, are you fitting this manually, or do we have systematized ways to do it? Well, this is a very important topic because we have a volume of data we are updating periodically. But what we are migrating is the link with some applications that allow us to automate the process. That's what we are doing right now. So for now, it is not a manual task. We are doing it in an automated fashion now. But we are migrating to a platform with APIs for the system to be completely automatic. But does the platform have an API? No, the platform doesn't have an API yet. It's because visualization space, all the analysis is underneath that on the servers, where we take care of the results of the information, and what we do is migrate the results to the dashboards alongside with the university. So we are trying to build some APIs, and we try to link it with other APIs for more automation. There are some others because there is not worth to do it because of the source. And we have some sources with very low frequency, maybe some that are a little bit late. According to the topic, it would be nice to consider automation to check that, but so far it's a very good picture of the information that we have at a municipal level. And we are doing some engineering tricks to have quicker information and real-time information, but so far that's what we are doing. The cattle, are you doing it manually? Those are databases of the ICA. And that's important. It's important to mention that that 90% of the data that we have comes from public sources, which allow the replication of the information. But based on those public data, we can extract additional extension of research, and we are already doing that, for example, with the cost of biodiversity. Thank you very much, Carlos. Any other questions? Okay. Thank you. A round of applause, please. Bye. Thank you. Before the tasting, we have some words by Andrés Jaramillo. I think everybody is tired. I want to thank the ones who came here, and of course the ones that are online, and to everyone, every one of the people who had a presentation. It is something very interesting. Somehow we got some convergence in the topics that were discussed in this day. We started with Mauricio about inventory, about botanics, that opens our eyes to the diversity of our country, and with some talks that start talking about how to characterize that diversity, to Justin's talk that talks about composing foods and start doing the task of being more efficient, having a more efficient consumption of foods because of the biological effect of absorption. And with Carlos' talk about the platform plaza, which I think is an excellent start to keep an inventory of how the food industry moves in Colombia and how the consumption of foods is happening with that in Colombia. I want to thank you again, the staff, the logistics staff. I don't see them around. Maybe they are gone now. No, I can see them. Yeah. Ariadna, thank you very much. Our Master of Ceremony, Diego, who has been around all the time, both days in both symposiums, to our institutions, our collaborating institutions. Yesterday and today, we received Best Air Fund, the CIAT, the Rockefeller Foundation, PTFI initiative, and I think that this sets on the table the idea that we cannot work we have to open to other disciplines within the university. That is difficult to do. Sometimes just by the pressure of time, but if we open to what everybody else is doing, it's better for everyone, right? At last, the support staff, thank you very much. Up there in the table, translation personnel, and everyone, logistics staff. Let's taste some chontaduro.